So, good morning, everyone. I'd like to welcome you to the 9th Immuno-Oncology Symposium we are doing here under the umbrella of the Comprehensive Cancer Center Zurich. My name is Markus Manz. I'm also chair of the Comprehensive Cancer Center and of the Department of Medical Oncology and Hematology. And I had the pleasure to organize this 9th symposium together with Professor Chiara Magnani here, Tobias Weiss, and Dominik Schneiderwind, who will arrive soon. He's still taking care for patients, I assume. There he is, Dominik. Good morning. So, and that with the help of the Comprehensive Cancer Center, so mostly Silke Uflacker, taking care for all the organization and also taking care for the organization with the speakers. I think we have a fantastic program. It's also a very packed program. We have usually 20 minutes talk time, 5 minutes discussion. We have 4 sessions with some thematic grouping. I really would like to encourage speakers to stick to their time so that we do have the time for discussions, which is the great thing when you have a physical meeting. So I heard that the registration for the meeting was about 340 individuals and 157 for at-site participations and the others online. So when we do the Q&A, it's also important that you get a mic. People will walk around and hand mics, and we will also read the questions from the chat. I don't know whether you follow that, but of course we were very pleased to read that again, University Hospital Zurich is 10th best hospital of the world, at least by an evaluation of Newsweek, and one might dispute how that is done. So we are very proud of it. We know that it's probably mostly for the clinical service. This symposium is really about bringing innovation, and you might know that Switzerland is also very high when it comes to innovation index, also in the world ranking. However, we do have somewhat a problem, at least in my view, and in view of many colleagues, in terms of really translating all the innovations into practice, and that is particularly true also in medicine. And all the speakers here are really drivers of innovation that go into translation, and we are very, very fond of that. And we hope that we in the future will also there make great strides towards translation. So finally, I should say, of course, these symposia are also only possible if we do get the respective support from our industry partners. It might be streamed here in the breaks. Yes, here we are. So we are very thankful that this support is coming, and we are able to do these symposia. Thank you. Okay, so I think without further ado, we should go into the first session, and it's my pleasure to announce Pietro Genovese, coming from Harvard and the Dana-Farber Cancer Institute in Boston. He is a very visible person when it comes to the genome editing, and recently he really made important contributions in terms of immune epitope shielding, and he will tell us what that is. Maybe I should add here, we invited speakers and proposed titles And most of the people said that they would respond. They didn't change the title. And I thought, wow, are they really giving all these titles? And my expectation would be that most of the titles will be changed once people come down here and give their talks. Anyway, and that might be the case here as well. So, Pietro, please, the stage is yours.
Thank you. Thank you very much for the kind introduction, and again, thanks for the invitation with both you and Chiara. Indeed, it's my real pleasure to be here and present you some of the most recent updates on different projects. For this reason, I changed the title that at the moment running in my lab in Boston. So, as you may know, I've always been interested in developing genetic technologies on hematopoietic stem cell and T cells for finding new therapeutic strategies for human diseases. These are just my disclosures. And when I start my work in Boston, I decided to try to solve one of the major problems in the development of cancer-adoptive immunotherapy for acute myeloid leukemia, which is actually the absence of a real actionable target for an effective immunotherapy. Because despite several studies investigating at the transcriptional and molecular level the differences between leukemia and healthy hematopoietic cells, many or actually all the possible suitable targets for a cancer-adoptive immunotherapy are often shared with the normal healthy hematopoietic stem or progenitor cells. So this actually imposed the limitation that any type of immunotherapy has to be confined in time in order to avoid incompatible hematological toxicities. And this often is not enough for achieving complete eradication of the leukemia. Recently has been proposed a very interesting approach that is based on the fact that since AML patients, especially the high-risk patient, normally underwent to allogenic stem cell transplantation as a standard of care, we can possibly engineer the allogenic transplant of the healthy cells in order to knock out the target of your immunotherapy, such as, for example, the CD33, that is one of the first targets that has been used with this approach. So in this manner, you can spare the toxicity on the healthy cells and allowing now full activity of, for example, a CAR against CD33 that is now recognizing and killing only the leukemic blast. I think that this approach is, of course, very nice and it surely is elegantly showing already some level of efficacy in a clinical trial that is ongoing in both therapeutics in Boston. But it's still the fact that you have to perform knockout of your target impose again a limitation that only few targets can actually be knocked out without affecting the normal development of an hematopoietic system. So the number of targets are few. Moreover, if you can perform knockout without any significant functional impairment, this means that these genes are not essential for the survival, neither for the stem cell, but also in particular for the leukemia. So the leukemia can easily possibly generate immunogenic escape variants just by downregulating the expression of the target or losing completely its expression under the pressure of the immunotherapy, like, for example, occur with the CD19 in some CAR-T cell trial. So our idea was that instead of performing knockout of the gene, since we can be very precise with our gene editing tools, we can actually just modify the epitope that is recognized by the therapeutic antibody, and so the CAR-T cell, in order to avoid the binding of the antibody, but at the same time preserving the full functionality of the gene. So in this manner, we can target also genes that are pretty essential for the survival of the leukemia stem cell, such as the cytokine receptors that we select as first candidate for this approach, so FLT3, CD123, and c-KIT. So to develop this strategy, we actually optimize a screening platform for first performing epitope mapping. So we took advantage of a library that contains several mutations in the extracellular domain of our target receptor. And then by screening with a therapeutic antibody and another relevant antibody, we can select the cells that are negative for the binding but still express the receptor. And then by performing sequencing, we can identify which are the actual important amino acid residues that are important for the binding. And to our surprise, I admit, we actually realized that for all the 3 targets, a single amino acid substitution was sufficient to completely abrogate the binding of the therapeutic antibody, while of course fully preserving the normal expression of the receptor on the surface, and importantly, without affecting the binding capacity of the receptor to each specific ligand. So thus, after exposure of the ligand, the normal intercellular signaling transduction pathway can be activated. So since a single permutation is sufficient to provide these features, we decided to develop a gene editing approach based on the use on the most recently developed adenine-based editing tools. So these tools, for those of you that does not know the system, are based on the use of a Cas case that is fused to a deaminase domain. So these enzymes are actually able to introduce a base conversion into the so-called editing window of the target site on the human genome. So of course we need to precisely position this editing enzyme on our locus of interest. So for this reason, we test a different version of the Cas enzyme with different PAM specificities in order to identify the best one that can fit our mutation. And we select the best combination that allow us to introduce with high efficiency this epitope edited mutation into the target cells. Then, by exploiting in vitro transcription of a messenger RNA and electroporation of this editing enzyme into primary human CD34 positive hematopoietic stem progenitor cells, we found that we can achieve a very high editing efficiency in these primary stem cells in all the targets that we tested. Indeed, we reach more than 90% for FLT3 and more than 70% for the other 2 targets. And importantly, also by analyzing the different subpopulation that constitutes the heterogeneous hierarchy of stem cells in the total treated CD34 cells, we found that the editing efficiency is very high also in the more primitive progenitors, thus possibly allowing the editing of long-term repopulating progenitors. Then, as I mentioned, the fact that all the adenine that are present in the editing window can be edited actually intrinsically generate the possibility of introducing what we call bystander edit. So we edit also not only our target site, our target adenine, but also the one that are there nearby. In some cases, this generate non-silent variants. So in order to understand whether the receptor that will be generated after this mutation is actually functional, we screen also all the possible combination of bystander mutation and site-specific mutation. But in all the cases, we confirm that the resulting receptor is fully resistant to the binding of the therapeutic antibody. It's also at the very high doses that we use in our assay. And again, importantly, there is no differences in the affinity of this edited receptor to their specific ligand, so that's confirming the functionality of the receptor at the molecular level. Of course, we then want to confirm that the cells are not affected at all by the editing procedure. So after editing the hematopoietic stem cell, we run several functional assays to try to understand whether there is an impairment in the functionality of this receptor after exposure with their ligand. And so we run transcriptomic analysis, of course, growth rate in presence or not of the cytokines, and even phosphoproteomic studies. And with all these type of assays, we do not observe any difference within the edited cells and the control cells. And importantly, there was no counter selection of the cells that have been edited, thus further confirming that the edited cells are fully functional, like wild-type cells. We also perform, of course, transplantation in the mouse model to confirm long-term engraftment of the edited cells. But then we move even further to try to develop a more advanced in vivo mouse model in order to test if the cells are actually really resistant to a CAR T cell therapy when the CAR T cell are actually killing a leukemia. So for doing this, we took advantage of MBSGW mice, and we first engraft them with the edited hematopoietic stem cell or control cells. And then after hematopoietic reconstitution, we challenged the mice with the patient-derived AML xenograft, like mimicking a kind of relapse post-allogeneic transplantation. So after leukemia establishment, we treat the mice with CAR-T. In this example, it was the FLT3 CAR. And after 2 weeks, we check for the presence of leukemia. And as you can see, this in particular with FLT3, but also the CD123 CAR were very, very effective in controlling the leukemia that was almost completely eradicated from these mice just in 2 weeks after treatment. But as expected, the use of FLT3 directed CAR T cell actually generate toxicity on the healthy hematopoietic cells that normally and physiologically express the FLT3 molecule. Indeed, we lose almost completely the presence of the granulocyte monocyte progenitor, the lymphomyeloid progenitors, or the pre-B cells. While on the contrary, the mice that were transplanted with the epitoperitic cells normally display this population and were fully spared by the toxicity of the CAR T cell therapy. Importantly, we also made a very interesting observation that not only the epitoperitic spared the toxicity to the stem cell, but also has some positive effect also on the CAR T cell per se. Because indeed, since now the healthy hematopoiesis is not expressing anymore FLT3, there is an overall lower antigen burden. So the CAR T cell now, they can concentrate their activity only on the leukemia. And indeed, we observe that in the mice treated with, and transplanted with the edited cells, there was an overall lower level of CAR T cell expansion. And these CAR T cells express a lower level of activation and possible exhaustion markers, thus suggesting that this can potentially benefit also the long-term persistence and reduce the exhaustion or cytokine release syndrome of the transplanted CAR T cells. Then, since base editing actually is a very simple but also not so dangerous gene editing procedure, we can easily perform multiplexing by editing not only one target, but even 2 or even 3 targets we try on the same cell. And by running this experiment with the improved protocols, we actually realized that the editing efficiency is very similar, even if we target only 1, 2, or 3 targets. So we don't lose efficiency just by performing multiplexing. So we can easily generate multiple edited cells that are now resistant to the bispecific or even trispecific CAR T cells. And this can result in an improved combinatorial CAR T cell therapy that can better control also heterogeneous leukemias that express the target a different level of expression. For example, we were able to eradicate also a leukemia that was not possible to be eradicated only with single CAR T cell. So in conclusion, I think that this strategy can actually provide a new way for exploiting a novel and more specific target for acute leukemia. And indeed, we are also happy that other groups independently develop a similar approach, for example, targeting CD45, such as the group of Sargil in UPenn and Lucas Checker in Switzerland. And we think also that this strategy can easily move towards clinical testing because, as I mentioned before, allogeneic transplantation is a standard of care for a risk patient. So we can theoretically perform the editing of the allogeneic graft and then treat the patient at the moment of relapse or MRD positivity. And in this manner, provide a way to control also the very nasty relapse in this type of disease. But of course, once we can confirm the safety of this approach, we can even think that we can exploit the use of CAR T cells that deplete the hematopoiesis to perform a kind of, to anticipate the treatment even before the transplant and use the CAR T cell per se to perform a deep conditioning of the patient, so thus removing both the leukemia and the healthy cells and thus allowing the engraftment of an edited hematopoiesis that is now resistant to the CAR T cell and can allow the CAR T cell to persist and perform long-term controlling of any type of relapse. And as you can imagine, this type of strategy is also very suitable for other for other application, also in non-malignant disorder. And very briefly, I will mention the possible use of this strategy for improving some of the emerging non-genotoxic conditioning strategies that are currently under testing in order to allow hematopoietic stem cell transplantation without the need of genotoxic chemotherapy regimen. So the idea behind this approach is that you can use antibodies directed against the stem cells in order to kill the stem cell directly in the bone marrow niche, but while sparing the toxicity of the chemotherapy to the healthy bone marrow niche. So in this manner, you can avoid the off-target activity of the chemotherapy, but also enable a faster engraftment and hematopoietic recovery in the treated patient. So some of these approaches, especially targeting c-KIT with antibody or antibody-drug conjugates, are currently under clinical testing for some immunodeficiencies. But this approach is still actually limited by the pharmacokinetic of the antibody, because the antibody, of course, have to be fully diluted before you can perform the transplant. Otherwise, you will kill the transplanted cells. And moreover, you have also to use antibodies at a really high concentration in order to completely deplete the bone marrow niche. And this can actually generate off-target toxicity, especially if your target is expressed at low level in non-hematopoietic system, or if you use an antibody-drug conjugate, you risk to oversaturate with the drug that is conjugated to the antibody. While on the contrary, if you can perform a transplantation with the epitoperitic cells, now you can perform the transplant at the nadir of activity of the antibody, because so at the nadir also depletion of the bone marrow niche, thus facilitating the engraftment of the transplanted cells, at the same time possibly anticipating the hematological reconstitution of the patient. Moreover, and also very importantly, you can perform also multiple administration of the same antibody, because now the cells are forever resistant to the antibody, so you can also further expand in vivo the graft till you reach the therapeutic level for of engraftment that is required to achieve therapeutic benefit. So we decided to exploit this approach with in collaboration with the group of Dan Bauer to develop an autologous gene therapy strategy in which we aim to edit the KIT receptor to provide the resistance to the KIT antibody, and the erythroid enhancer of BCL11A, which actually, after disruption, allow the restoration of the expression of the fetal hemoglobin, so we can possibly develop a strategy for treating hemoglobinopathies. So also here we took advantage of electroporation and base editing, and we can achieve easily, I would say, very high percentage of editing, in this case triple editing of the hematopoietic cells, with both KIT and the two erythroid enhancers. And importantly, by performing the editing of two enhancers instead of one, we can also further increase the level of fetal hemoglobin expression. So that's possibly providing a better way for controlling the hemoglobinopathies. And then when we test in the mouse model, we try to, again, exploit the real possible, to test and challenge the real advantages that we can get with the epitope. And so we tested different antibody conditioning regimens. So we compare the more conventional, what we call intensive treatment with antibody, with a more diluted antibody regimen, in which actually we use the same cumulative dose of antibody, but distributed in much more time after the hematological constitution. So we ran this experiment by transplanting in competition edited cells and wild-type cells, and then we measured the engraftment first longitudinally in the blood, and then the sacrifice in the bone marrow, by looking all the different hematopoietic lineages that develop in the xenotransplanted NSG mice. And so, as you can see here, despite we transplanted only a small fraction, 20% of edited cells, here marked in blue, After the treatment with antibody, we can achieve a significant twofold enrichment of the edited cells in all the different hematopoietic population. But importantly, by prolonging the antibody exposure, we actually achieve an even better and more sustained selection of the transplanted cells, reaching more than 80% of engraftment that is now deriving from the transplanted cells. And importantly, at the genomic level, we can confirm that we can, by selecting for KIT editing, we can co-select also for the edited cells, so the therapeutically edited cells. And so we think that this strategy can actually provide some benefit for also application in non-malignant disorder, in particular autologous gene therapies. And I didn't have time today, but we also exploit a very similar strategy by taking advantage of the prime editing strategy that allow us to have even more flexibility in the selection of the mutation that we can insert for conferring resistance to the therapeutic antibody. And I think I'm running late, so I would like just to finish the presentation, acknowledging all the people in my lab that actually ran the experiment, and in particular Gabriele Casirati, who lead the development of the PITO editing, then supported by Andrea Cosentino, all the other lab members, our collaborators, the funding agencies, and of course all of you for your attention. I will be happy to take any questions.
Thank you, Pietro. Fantastic talk. Are there questions from the audience? Maybe as the microphone is distributed, I start with one. So when you select the target antigens for the epitope editing to do post-transplant continuous pressure, for example, on the AMLs, in how far are you, when you select the antigen, in how far are you considering on low-level non-hematopoietic antigen expression, which will not be edited outside of the hematopoietic system, and where the CAR T cells could, in the long run, maybe cause some trouble as for KIT expression, FLT3 expression, IL-3 receptor alpha expression?
Yes, no, this is a very important question. Indeed, for the KIT and for KIT, indeed, we envision that maybe a CAR T cell approach is not the ideal, exactly for the reason that you say. There are possible also low expression in non-hematopoietic tissues that is something that you don't want to have. In case of the FLT3, which is actually our first lead candidate for our first clinical testing, we think that the expression of FLT3 is pretty much confined in the hematopoietic system, so it should be pretty safe to move forward with this target. CD123 is still controversial. There is some expression in the endothelial level, even if in the original CAR T cell trials there were no toxicity observed. Of course, we want to monitor this, also take advantage of the use of suicide gene, possibly to control the response of the CAR T in case of adverse events that are not hematological toxicities. But at the same time, we also would like to have at least some level of antigen that is still present in order to support the engraftment and persistence of the CAR, because otherwise they will not expand or survive. Have to be tested.
Thank you. Chiara?
Thank you, Pietro, for the nice presentation. I have a question on what you show about the possible translational approach that you envision for your strategy. So you mentioned two options. The first one will be to proceed with transplantation and then treat at the moment of the relapse with CAR T cells. The second option instead are using CAR T cells to deplete the tumor, but also as a non-genotoxic condition in the gene. And you also mentioned that in the first option, basically you will reduce the amount of antigen that you are targeting and preserving a little bit then the T cell fitness without having exhaustion. is something that you also evaluate in preclinical model. What is your understanding about a possible dose that then you are going to use in these two approaches?
Yeah, so of course, all that slide were pretty much just a simplification of the possible approaches, but our idea is that with the pitope editing, we can really increase the therapeutic index. So what we would like to achieve is actually using a higher dose of the immunotherapy, but also possibly the CAR-T cell, in order to really achieve therapeutic efficacy. Because at the moment, many of the CAR-T cell trials in AML actually do not show neither efficacy, but also no toxicity. So if you don't have toxicity on hematopoietic target, this means that you cannot have efficacy. Well, what we would like to achieve is increasing the dose and increasing at least the way we can treat the patient to have a more effective immunotherapy. Of course, this can be a problem if you have only minimal residual disease and non other antigen, because the CAR-T cell will not expand. So you have to play with the dose or use antibodies or antibody drug conjugates instead of CAR-T cell in order to perform the therapy. If you want to instead use the CAR-T cell before, of course, then there is the issue from where you took the CAR-T cell. So, as you know, as we discussed with several, also with you, one idea would be to use a donor-derived CAR-T cell, and in a patient that has already underwent a first stem cell transplantation, so use the same CAR-T cell from the cell donor to control the leukemia and then retransplant with the same allogenic donor.
So I do not see further questions, and I believe also in the chat we don't have questions. So thanks a lot, Pietro. Great talk. So it is my pleasure to announce the next speaker. It's Christos Georgiadis from Grand Ormond Street, the Children's Hospital, and he is a senior research fellow there, focusing also on base editing technologies to generate off-the-shelf universal CAR-T cells. And the floor is yours.
Thank you. You can hear me, right? Yes. That was fantastic pronunciation, by the way. Thank you very much for the invitation to this beautiful city. It's absolutely fantastic. And thank you for the introduction. So this is our city. This is probably an AI reimagining of what the weather would look like in London. It doesn't really look like this, but we are very close to St. Paul's Cathedral there. And we're quite uniquely positioned at the interface of academic research at University College London, that you can see there on your left, and the Institute of Child Health is the new building that we're based in, which is actually part of Great Ormond Street Hospital. This is what it will look like in a few years after construction's finished. But we have our outpatient department in our GMP facilities on the 4th floor, and us researchers are in the basement. So it's a hard act to follow after Pietro, so I'm going to take you a bit back to basics. I see you're a mixed audience. I'm going to sort of describe the concept of universal CAR-T cells, then talk through some of our trial data using CRISPR for the development of the B.A.L. therapy, and then our most recent base-edited therapy for T-cell acute lymphoblastic leukemia and some of the first treated patients. So it's undeniable to say that in the past 50 years, the outcomes in the treatment of childhood and adult leukemia have been absolutely impressive. So you can see the chance of survival in 1970 was below 40%. Now we have almost 95% of patients having an option to be treated with conventional therapy, stem cell transplantation, and modern medicines. However, there is a fraction of these patients, and it varies between B.A.L. and T.A.L. and AML, which have really lagged behind compared to B.A.L., where you have refractory relapse patients. Now they will have failed induction chemotherapy, or they will have failed the first or second transplant, and then their prognosis is dismal. So the chance of survival falls below 20% in some cases, with the only options being palliative care. So the past decade has seen an impressive array of new therapies that have come into use. So you have your bispecific engagers, you have antibody drug conjugates, but what's quite remarkable is the progress that we've seen in the CAR T-cell field over the past decade. And I'm sure the majority, more than 90% of you here, will know what a CAR is and how it functions. But I thought I'd introduce you before the next 20 talks, which we'll be talking about CARs. But you basically fuse the antigen recognition domain of a single chain variable fragment from a monoclonal antibody. This could be one that's available in the clinic, let's say rituximab for CD20. And then once you have that antigen binding domain, this is fused to the co-stimulatory domain in the next generation CARs and activation and signaling domains of a T-cell receptor. So this offers you an MHC unrestricted way of targeting and killing a disease that you could otherwise not have, such as B cells with T cells. And the poster child of this therapy is Kymriah and Yescarta, and you will all have heard of it. You will have used it here in your hospitals. It's commercially available. And this has really shown remarkable responses in the treatment of these, what were considered untreatable diseases. These are all focused around CD19, ubiquitously expressed B-cell targets. And what you can see in these relapse refractory patients, we have now managed to increase the chance of survival and long-term survival. These are being tracked for well over 10 years now, above 40%. So these all operate on a second generation CAR architecture. So this includes a co-stimulatory domain, and they're typically delivered to cells, to T cells, using a viral vector, typically lentivirus or retrovirus. So there's 6 products, half a dozen products now commercially available for the treatment of BALL and multiple myeloma from these different companies that you see here. So it's just important to explain that these are autologous products. So these rely on obtaining the cell material from the patient, who will have undergone quite significant cytotoxic therapies ahead of time, so the quality can be variable. These are bespoke products. They have quite a high price tag, but you don't have the limitations of trying to match the donor. The responses have been quite impressive, though. So, as I said, we are quite uniquely positioned in that we have a lot of prior background in transplantation and treatment of cancer at the hospital, but we also have in-house GMP viral vector manufacture in the academic institution. We have GMP suites, in-house release testing, and a good collaboration with the MHRA and the regulators for approval of clinical trials, and a point-of-care manufacturing and delivery of the product to the patients at the hospital. So this was the perfect sort of breeding ground to develop these off-the-shelf CAR T-cell therapies. And that's been made possible from the recent developments in genome editing, which have allowed us to use unmatched healthy donor cells as a starting material, modify these ex vivo to remove alloreactive markers, deliver a CAR, and expand them, qualify them. So we do all the quality release, bank them, and then they can be administered to patients at the time of need. So that greatly reduces costs and increases the accessibility across patients. So how would you go about overcoming the alloreactive, the HLA barriers? So there's two parts to this puzzle. You involve preparative conditioning for the patient. So this would be using lymphodepleting, debulking agents such as fludarabine, cyclophosphamide, and alemtuzumab. So alemtuzumab targets CD52, a lymphocytic marker, and can really convey quite deep conditioning to the patient. And that would allow the CAR T cells coming in to not be rejected. So on the other hand now, you also have to, using genome editing, disable a few markers on the cells, one of them being CD52 to avoid them getting cleared by the residual serotherapy, but also the T cell receptor, which is the key mediator of graft versus host disease. So we've shown that by eliminating the TCR, we can practically eliminate GVHD effects, and we haven't seen GVHD effects in these patients. There are a number of other markers that you could target, such as the class 1 and class 2 molecules. There's work ongoing with these. They're being tested in trials. Our products don't have these implementations yet, but we expect future ones to. And how would you go about doing this? So you have a whole host, an arsenal of these editing tools. So we started off with a protein-guided system, zinc finger meganucleases, talons. And now we have newer technologies, easier to design, cheaper to manufacture, the RNA-guided systems based on CRISPR, which first came about in 2012. But our first experience with genome editing was using TALEN technology. This was back in 2015, where we had a multiplex edited removal of TRAC, the T cell receptor, and CD52 in CAR-19 cells. And this was for BALL. It was a clinical study run at Great Ormond Street Hospital. And the next one followed with an implementation of the CRISPR system. So in that initial study, it was the first universal CAR T cell study. We were able to show that the TALEN-edited cells that were disrupted for TCR and CD52 could be infused into patients that had been pre-treated with alemtuzumab, and they could achieve molecular remission within a period of 28 days following infusion. Now, this was a bridge to transplant therapy, so the patients, after achieving molecular remission, would go on to receive a matched transplant to allow for immunological rescue. And this was then taken over by Servier, and they run a larger study with manageable toxicity for patients who didn't have an option for autologous CAR T cell therapy, and the results are published across adults and pediatric patients. So in 2020, we adapted this system for the use of CRISPR. So we developed a new lentiviral vector, second generation CAR contained within it. But we positioned a dual targeting cassette for the same targets, TRAC, T cell receptor, and CD52 within the long terminal repeat, 3' long terminal repeat of the vector. And we deliver our nuclease, the Cas9, by electroporation. So this is transient to ensure that it's not causing long-term effects such as off-targets. But the editing is permanent. By combining these vectors, we are able to couple the CAR expression to the knockout, which means that we can enrich quite significantly for the CAR T cells. And this is where we started working with Miltenyi to develop a pipeline using this semi-automated manufacturing device, CleanMax Prodigy. I'm sure a lot of you will have seen it or even used it, where we could start with 100 million healthy donor T cells, and then following transduction, electroporation, expansion, and finally a TCR alpha beta depletion step to remove any residual TCR alpha beta positive cells, we could get close to 3 billion cells back from it that we could cryopreserve into doses. That's enough to probably treat over 20 patients from that single initial donation. And as you can see, due to that vector design, after depletion, we can get our cells homogeneously expressing the CAR construct, all TCR negative and pretty much all CD52 negative. So of course you need to do a lot of off-target analysis. Everyone brought up the complications of using nucleases to disrupt the genome. You can increase the risks of off-target editing. So we look at the molecular level, and we can look at the on-target analysis using something basic like Sanger sequencing before moving to in-depth NGS analysis or whole genome sequencing analysis. And for off-target effects, because you could have similar sequences targeted across the genome, in this case, we use a technology developed in South Korea called Digenome-seq. So this is an in vitro cleavage analysis, so it's a bit more robust than your in silico predicted targeting. and then we can interrogate those sites with next generation sequencing. Importantly, we didn't find any off-target effects. We can also look at the translocations. So these are reciprocal translocations and karyotypic abnormalities. When you hit two different chromosomes, you have the potential for fusions and translocations. This was seen in the TALENT products. Up to 40% of the cells did have translocations that went into patients. However, they didn't have any significant effect in them. And we look at this with ddPCR technology. So following this, we set up our clinical trial. So this is for pediatric patients aged 6 months to 16 years old that had failed prior therapies, including prior autologous CAR-19 therapies. And the patients were screened, lymphodepleted with fludarabine, cyclophosphamide, and alemtuzumab, and then they received a dose of between 1 and 2 million CAR T cells per kilo. They were tracked for 28 days, and depending on the results, if they achieved molecular remission, they would proceed to receiving a matched transplant, and then they were monitored after that. So the toxicities with this study were very similar to the previous ones reported. Very low levels of CRS, no reports of high-grade GVHD or ICANS. And out of the 6 eligible patients that were treated on this phase 1 clinical study, quite a few of them achieved flow remission, but only 2 of them achieved quite clean flow and molecular negative disease. And these 2 patients are here on the right. So they're both alive and well, and it's more than 4 and 3 years that these have been tracked now, and we're carrying on tracking them. So now moving on to TALL. So unlike with BALL, the same level of progress has really not been achieved with this disease. It's a very aggressive disease. It operates in quite a different way. And it's also due to some inherent challenges which are associated with the disease. So one of these challenges is fratricide. So this is self-targeting. So this is the presence of shared antigens on both the healthy, malignant, and effector T cells. And there are clever ways that you could go about removing these from the surface. So one of these reports involves a protein blockade where you can trap in this case, CD7, because CD7 is very highly expressed both in TLL and on T cells and T cell precursors. So you could trap the CD7 in the endoplasmic reticulum, avoiding it from being expressed, and you could proceed with your manufacture. There are additional complications with T cells. So unlike the B cell therapy where B-cell aplasia can be treated with IVIG treatment, there's no really an alternative solution for T cells. So you'll get these protracted cytopenias and T-cell aplasia, which could, of course, increase the risk of infection. However, we feel these could be mitigated by a time-limited exposure to the therapy followed by immunological rescue. And in the autologous setting, of course, this is not necessarily relevant to us, but it's important to understand that you can have product contamination if you use patient material to develop this therapy, because you could very easily transduce your malignant blasts. So we received funding from the MRC and the Wellcome Trust. So this was a 2-pronged approach because CD7 is also expressed in AML. So the idea is that we could use a single product to target both diseases separately. So looking at the first one here, so we used a technology that had been recently described by David Liu, base editing. So Pietro mentioned this before. So this is a near seamless approach. So compared to your nuclease double-stranded break induction of CRISPR, you now have a nicking and then seamless nucleotide conversion at a precise location. So this is a really powerful technology. So we use this in this setting because we now had to add an additional editing step. So we had our TCR, we had our CD52, but now we had to disrupt CD7 to prevent fratricide. And we also have to change our timelines because this has to happen now prior to transduction. So to reduce the risks of additional translocations with the 3 edits, we now switch to base editing as an option. And the idea is that we could have these triple edited cells, again, with a transient delivery of base editing mRNA transduce them, and we could then target CD7 on the cells, on the malignant cells. And using base editing, you can achieve the same form of gene disruption by targeting nucleotides. The following conversion could introduce a premature stop codon, or you could target the splice donor or splice acceptor side, therefore disrupting your gene transcription. So this is quite an effort to obviously assemble all the materials needed for the GMP manufacturing. We worked with a number of European and international suppliers to be able to design these GMP products. But we managed to get high quality products to use in our trials. And this was, again, using the Prodigy device, but with an altered protocol where we have to deliver the editing material ahead of transduction to avoid the fratricide. We manufactured banks of these cells. You can see here just there are some R&D banks, but looking at the GMP banks, so high transduction efficiency, practically no TCR residual expression, and very high levels of knockout across the 3 targets. Looking again at the molecular, we interrogated these cells. You look at these cells quite differently to the ones that have double-stranded breaks. So here we're looking for nucleotide conversions. So as I told you, some of these guides deliver a nucleotide conversion to create a premature stop codon, and others to disrupt the splice side, but can achieve near 100% conversion at those sites. We had to do off-target analysis, so we picked the top 20 in silico predicted off-target sites for each of these guides and run next-generation sequencing on them. There were negligible, if any, off-targets picked up. And also, as the editor is known to target RNA, we also wanted to assess whether there were any RNA changes. And we specifically focused on the lentiviral scFv, because if that was to be modified at the RNA level, you could inadvertently change the targeting. So what we saw was that across time points looking at the RNA, there were no obvious changes. I apologize for the quality of the image. I don't know why it's so blurry. We did in vivo functionality before moving into a clinical trial. We also compared this to an SPCas9 made batch alongside, and we didn't see any difference with the base edited cells. Functionality was not impaired in any way. However, you can see here, when you now look at the potential for translocations across these sites, if you're to use a double-stranded breaking nuclease, you would end up with a relatively high percentage of translocations, whereas these were avoided with the base editing. So we set up this clinical trial using very similar routine to the ones that you saw earlier for the BALL trial, with lymphodepleted patients receiving a single infusion of the CAR-T cells. If they achieved molecular remission, they would go on to have a stem cell transplant. So this is the first patient ever to receive base-edited CAR-T cells. I should just say now she's alive and well. She's a fantastic person, and I've got her invited to speak at my public engagement conference in a couple of months. So she had relapsed. She was refractory. She would have gone into palliation. However, following a single infusion of the CAR-T cells, we were able to eliminate the disease. And by day 28, she received a matched transplant and had immunological recovery. And we could pick out the cells throughout the whole period of treatment. And when we pulled these out, phenotypically, they were identical to the product that went in the first place. And throughout treatment, she had 100% chimerism from the CAR. And then following the transplant, the transplant donor then took over. But it was molecular remission by flow and PCR after the day 28. We treated 2 more patients with this product. The first patient, unfortunately, passed away on day 33, even though they'd achieved flow remission. However, there was an Aspergillus infection that led to their demise. And this is one of the complications with obviously having quite a CAR that causes quite a bit of cytopenia. It's very hard to fight off infections. Patient 3 is alive and doing well. So they had CNS involvement, but we were able to clear the disease. and they are now close to 2 years post-treatment. So just in the final couple of slides, just say, so now we're also focusing on AML. As I said, 7 can be used for the treatment of AML. It's a very heterogeneous disease, as Pietro alluded to earlier. You can't really manufacture an autologous product because the host genome is very much at risk. And what we think is a combinatorial approach might be better suited for this. So there's all these targets for which we have CARs developed against. The one that we're taking forward and have taken forward is the 33 CAR. We've also knocked out 7 on there to make it compatible for combinatorial infusion with a 7 product. And we're working with Hanover University as a combined trial. So this is open at GOSH, and we are treating patients. However, I can't share any data on this at the moment. So in summary, I can show you that T cells are attractive for gene editing, and we can create these universal banks of T cells that can be ready-made and readily available. And we've now advanced genome engineering to the step where we can have accurate, reproducible, and traceable edits. However, long-term follow-ups would be needed to really assess the safety of the products. And it's a risk balance of risk benefits between the short-term and long-term risks. So we need to manage our expectations and manage the products and tailor them to deliver the level of lymphodepletion, the level of toxicity that may be required for the treatment of the disease. And you know, it's a rapidly evolving landscape. What Pietro was describing earlier with epitope shielding might be the best way for immunological rescue for patients such as those in AML, but we have other ways of editing molecules on cells to make them fitter, resistant to exhaustion, more potent. So with that, I'd like to end and just thank all the people that have been involved in this work, and thank you for listening.
Thank you, Christos. Are there questions? So again, I might start off. This is really impressive, and the use of allo-CAR T cells in this very immunosuppressed situation of acute lymphoblastic leukemia is easy to understand and likely working, as you showed, very well. Do you see a role of allo CAR T cells beyond such heavily immunosuppressed states, and what would that mean for the recipient in terms of maintaining the allogeneic cells?
Yeah, that's quite important. So we are obviously there's other studies that are looking at using these at earlier stages, so not in those highly immunosuppressed individuals, but for allo-CAR Ts are at the moment the last resort therapy just because of the structure of how you receive the therapeutics. But in terms of your other question, we are trying to modulate through editing and editing class 1 and class 2 and being able to reduce the required lymphodepletion both pre and post. Whether it's a good idea to leave these allo-CAR Ts in the patient long term, we're not sure yet, but obviously you can have suicide switches that you can turn off the expression. We don't have very many well-working ones at the moment, but yeah, so for the primary concepts, we could use something like what Pietro was describing, have an edited transplant that wouldn't be affected, that would allow for immunological reconstitution rapidly, or modulate the T cell to make it more invisible, therefore requiring less initial preconditioning.
And their class 1 editing is the primary way to go?
We're an editing lab, so I'm biased, but I'm sure there's other clever techniques out there. But it's a very powerful technology if you have that initial concept. Yeah.
Okay, Christian Pellegrino.
Hi, thank you so much. It was a very interesting talk. I was wondering about the patients which you showed a response and really long persistence as well, since it's a long follow-up as well. was wondering if you performed any sequencing on the population of allogeneic CAR T cells which survived, and whether you could see any common features between the patients and maybe isolate a potential profile for your allogeneic CAR T cells.
Yeah, so that's a great question. And we're obviously quite limited by the period of time that we have to work with, so 28 days. And while you can pick up the copy number, maybe the number of cells is quite minimal. However, for most of the patients who are now on the TVT trial, the T-cell trial, we've treated a number of patients who are almost at the end of our phase 1 study, and we have some external adult patients who are treated off study. So wherever possible, I did capture cells for single cell sorting, so they're all ready, waiting for me to analyze. But yeah, that should uncover potential changes and maybe identify a signature that may be of interest to follow up on. Phenotypically, as I showed you, the cells that we pick out of the patients may be increased slightly in their CD8 ratio. However, phenotypically, with the basic markers that we would use, they don't seem unchanged to the original product. But no, we haven't done any further integration site analysis because by day 28, the cells wouldn't have changed that much. Yeah. If we were to see something months after, which we wouldn't expect, of course, we would sequence it. But yeah, single cell should confuse things even more.
Thank you so much.
Yeah.
So I do not see further questions from the audience, also online, which in a way is good because we are a little bit over time. So thank you very much again, Christos. And our next speaker is Reddy. He's from the ETH Zurich in Basel, from the BSSE. He's a professor there, but he also is co-chairing the new or relatively new founded Botnar Institute for Child Health in Basel. And he will talk about engineering T-cell specificity for cancer immunotherapy. Please,
yeah. Thank you, Markus, for the introduction and the invitation. It's a pleasure to be here, especially among all of this great clinical researchers. My work is much more on the engineering side and technology side, so I will not have the such compelling clinical data that we've seen from these earlier presentations, but hopefully I can show you some work we do on technology and platform development that maybe can have an impact clinically in the long term. Okay, my disclosures. So I think that this audience is fully aware of engineered T-cell therapies and their potential to have a major impact for cancer therapy. Now, if we really focus in on TCRs as one of the possible modes of treatment, therapeutic TCRs, it's clear, would have to be both potent, so some kind of high level of activation and targeting of a peptide MHC target, as well as safe, meaning they would have to have a fairly low amount of cross-reactivity. Now, in the natural T-cell environment here, you can see T-cell development. T-cells undergo this process of thymic selection, meaning that as they get to become higher affinity, I can see here, yeah, I have my arrow here. So as they become higher affinity to a self-antigen, they would undergo clonal deletion. Clearly, because if you had high affinity TCRs to self-antigens, you would end up with an autoimmune reaction. Intermediate affinity TCRs will could remain and become part of the regulatory compartment, but most of the conventional TCRs that are produced will be quite low affinity for obvious reasons. And when we are now thinking about tumor-associated antigens, so today I'm really going to speak in the context of tumor-associated antigens, not neoantigens. Tumor-associated antigens are essentially self-antigens. So people would only make low-affinity TCRs to such TAAs. So in the past, what has been attempted is to engineer a TCR, so enhance its affinity. Quite obviously, there are very powerful protein engineering technologies like phage display and directed evolution that enable the possibility to enhance the affinity of a TCR. And so when this has been done in the past, one of the most notable examples is a clinical study done by the company Adaptimmune. And in this study, they engineered a TCR by phage display for enhanced binding to this MAGE-A3 peptide pictured here. And when they incorporated this engineered TCR into a cell therapy, so a TCR T-cell therapy, autologous, and administered into patients, unfortunate consequence was that only within a few days, several patients died through cardiogenic shock. And retrospective analysis revealed that the engineered TCR that they performed by phage display ended up having a cross-reactivity to a peptide derived from the protein titin. And titin is a highly overexpressed protein on cardiomyocytes. So essentially, this engineered TCR had that cross-reactivity and attacked cardiomyocytes, leading to this adverse event and unfortunately patient death. Now, this had, of course, a pretty big impact in the TCR engineering field, really sort of slowing down the concept of making synthetic or engineered TCRs. So you saw at least attempts to move into this field were more captured by, just not engineering TCRs, but TCR discovery. So just finding patient TCRs, but there's a lot of caution in the idea of using any kind of protein engineering to manipulate a non-natural TCR because of that risk of introducing cross-reactivity. Now, of course, one of the issues is why did they miss this off-target? And there's different reasons for this. And so what we thought is, in my group, is we really wanted to explore a way to improve TCR engineering using some additional and newer tools. And so a few years ago, I, along with an excellent postdoc in my lab at the time, Rodrigo, we developed a platform that we call TCR Engine, which is really a high throughput platform for functional TCR engineering. And the difference here is that essentially it's going to consist, instead of doing a binding assay like one would do with phage display, we actually engineered a mammalian cell line that could express a library of TCRs, and that it can be screened. And this TCR library could then be screened both by binding-based assays, which can always be done using peptide HLA dextramers, but more importantly, it has a signaling pathway of activation, so we could do co-cultures with the TCR library and an antigen-presenting cell line. And then this would give you also a signaling functional readout. Our hypothesis was this was going to be much more sensitive than just a biophysical binding-based screen. And then we couple this to a lot of techniques we've used in the lab over the year, including deep sequencing and bioinformatics. And I'll show you essentially how the outcome of this. So first, we had to build this screening platform. And we did this by essentially performing a multi-step immunogenomic engineering process. And there's a lot of steps here, but I'll just briefly walk you through them. First, we incorporated a constitutive Cas9 into the genome of our cell line. And we used the Georgiadis T-cell line, which I think many of you are familiar with as a common cell line for T-cell, human T-cell study experiments. And then this SB-Cas9 is integrated into the genome. And then additionally, we end up adding basically a reporter of T-cell activation here. So that's a GFP linked to the NFAT. So that's a TCR signaling pathway activation detection. We knock out the CD4 gene. We want this actually to be a CD8 T-cell platform. So we knock that out and actually introduce CD8s. We knock out the alpha locus. I'll talk about that more in a minute. And we also knock out the fast gene to have to reduce activation-induced cell death. So this is kind of the sort of long list of genomic engineering steps we did, which was a pretty significant effort. It probably took about 2 years to finally make the cell line platform. And we call them TCR accepting T cells or TNT cells. Now, importantly, TNT cells, we've also removed the endogenous TCR, so in the cell line, which means that we can now go in and put in a synthetic TCR or TCR library. And we do this again by using CRISPR-Cas9 and homology-directed repair. Because the cells have an endogenous Cas9, we also have very efficient and simple genome editing, because we only have to electroporate a guide RNA and a template, because we don't have to worry about the Cas9 because the cell's expressing it already. And so the way we introduce a synthetic TCR or transgenic TCR is we actually put in a VJ alpha chain with the constant region of the alpha chain separated by P2A, and then the VDJ beta region. And importantly, this goes then into the actual endogenous VDJ beta locus with a guide RNA and appropriate homology arms. We maintain the splice acceptor site, which means that when you do this, only if it's integrated in the correct locus will you actually get functional TCR expression, because if it's an off-target integration, there'd be no way for it to splice with the beta chain. So essentially, only correct integration will lead to functional TCR expression. Because we don't have a beta chain included here, it's from the endogenous genomic locus, including splice sites. Okay, so then what this results is a pretty good platform to, as you can see here, we have the TNT cells with no TCR, and then we've added a new TCR to it. I just want to point out that it's very difficult to measure TCR expression with antibodies because of different germlines. So we use the CD3 co-receptor, which if you knock out a TCR, you would lose CD3 co-receptor expression. If you reintroduce a TCR, as you see here in orange, you see CD3. And in this example, we also had the cognate peptide MHC dextramer, and you show binding. And you can see here also when you've co-cultured these cells, this was a 1G4 with NY-ESO-1 peptide. When you co-culture that with an antigen-presenting cell line, you also see this very clean NFAT-GFP expression showing activation of the TCR. Now, we also can use a CD69 on the surface of Jurkat cells as activation. This ends up being a little bit more sensitive, and also CD69 has very low expression on Jurkat cell lines. And these are examples, again, of different peptides and TCRs and controls, which show quite a clean activation. So then the next thing we can actually explore is start to make libraries. And in this case, we make mutagenesis libraries of the CDR3 beta chain. And we did this on the DMF4 TCR, which is specificity to MART1. I'll get through some analysis of that in a minute. But what we tried initially was to make what's called a deep mutational scanning library. And that just means you introduce an NNK codon. So you introduce a mutation in each position of the CDR3 beta region that can code for any of the 20 amino acids. So it's a relatively small library because it's just the number of positions, which is 11 here times 20, so about 180 variants. And then the thing is, once you've introduced this into the cell line, the advantage that you have is you can actually screen it for both binding to the peptide MHC dextramer, but in parallel, you can also screen it for activation by just co-culturing it with an antigen-presenting cell line and the cognate peptide. So you can actually directly compare signaling and binding. And what comes out here, this is just a heat map that sort of is a way to visualize this. This is coupled to deep sequencing, as you see here, of the activated or binding fractions. And the heat maps, you can look at is binding on the left and activation on the right. They look pretty similar, but there's some differences here. It's easier to actually plot it like this, where you look at the enrichment based on sequencing on the X and activation on the Y, and you can see that if binding and signaling were completely correlated, you'd have something very close to this diagonal line, but you don't see this at all. So it actually shows you that these binding-based measurements are not as informative or as correlated with signaling. And of course, when you think about a TCR and you think about its function, signaling is a more closer physiological representation. Just to show you, these are single point mutation variants of this DMF4 peptide. And you can see this is the binding on the top and the activation on the bottom. And what's really interesting here is you can have examples where you see a MART1 where you see basically no binding by this peptide MHC dextramer, or very minimal binding with this mutation. However, when you co-culture that with the antigen-presenting cell line, you still see pretty significant activation, again, revealing there is this discordance between binding and signaling that can occur. This is not a completely unusual result, because work from Christopher Garcia's lab at Stanford has shown the opposite effect, where you see activation, where you see binding but no activation. So this is the kind of a reverse phenomena. Okay, so to kind of use this platform, we then went back and we used, we went after the same target of that clinical trial that was unsuccessful, and we used the exact same TCR that had been engineered by phage display. It's called an A3 TCR, and it's against the MAGE-A3 target on this HLA. And we applied essentially this workflow, which is using our TCR engine platform, but it's using multiple different steps. So it's activation steps, binding steps, but also counter selection steps against the Titin antigen. And this is always coupled with deep sequencing. Eventually, we used all of this data bioinformatically to select specific candidates. And what you can see here, I'm just showing the binding data of engineered TCR. So this is A3. And this is actually A3A. That was the engineered one by phage display that led to the cross-reactivity in the clinical trial. And then these are, of course, these are now our engineered variants. And what you see is certainly we have improved the affinity of all of our engineered ones to the MAGE target compared to the starting point of A3. But we also have many of these candidates, because we've counter-selected them, that don't show observable binding to that problematic off-target Titin. So we have some candidates to explore further. Now, in order to understand more, I mean, we only looked at one off-target, but there could be many more off-targets, obviously. We've engineered this TCR. So we did a peptide scanning assay, meaning we took our TCR cell line and we co-cultured it with an antigen-presenting cell, but with peptide variants of the target MAGE-A3. So basically, we make every single amino acid variant of the single position variant of the MAGE-A3 peptide. And then we do this co-culture. And this is actually the A3A that went into the clinical trial. So you can just kind of look at this. All this is basically saying is in position 1. You can tolerate a few mutations here, but it's pretty conserved to this, E residue. And, these positions here actually can tolerate lots of mutations. So now these are some of our engineered variants. And you can see by first glance, one advantage is immediately what you can see is that there's less green, which just shows you we've somehow, it appears in a very general estimation, reduced off-target, potential off-target cross-reactivity. But more importantly, we can use this motif to run basically a search on the human peptidome, for example. And what's really interesting here, in black is the A3A TCR, and the #1 hit in terms of the activation is the MAGE-A3 peptide, the target. But what was really fascinating is we actually uncovered that the second most activating peptide was Titin. Even though Titin is not in this assay, because Titin has 4 mutations and we only have single point mutations, you can sort of recover that motif by doing this analysis. And that means if we had been able to use this assay, it would kind of be like a stop sign that would have said you shouldn't take this TCR into the clinic, because clearly this assay uncovered Titin as an off-target. So anyways, going back though to the more important point is, what about new off-targets that could have been generated? So we then ordered another 350 peptides that are predicted to potentially bind or be activated by any of these 3 TCRs. And then we screened those again. Oh, sorry, I don't know. We screened those again, and what you see here is basically the peptides here have A3 on top, which is that clinical trial one, and ours. And the good observation is we have less off targets. We do have some that are remaining in all 3, like FAT2 and PLD5. We also discovered new off targets that were never reported for A3A. And more importantly, we did remove Titin. So there are still more off targets that we may have to explore, and whether those are clinically relevant. So we would still look into those. And then lastly, we put these TCRs into a human primary T cells into the track locus, made TCR T cells, and did activation assays as well as killing assays. And in both cases, you see our engineered variants show good activation without Titin cross-reactivity, as the A3A did, though. And we showed tumor cell killing in vitro at the same level as A3A. And then in a human tumor xenograft mouse model, in collaboration with Reddy and Heinz Laubli's group at Uni Basel, we show that one of our engineered TCRs can potently, kill tumors in this mouse model as good as A3A. So with that, we I do want to mention we started a spinoff company with Rodrigo as our CSO. And one of the things is, of course, the TCR or just the cell therapy market in biotech has shifted a lot in the last couple of years, making it very difficult to advance cell therapies, at least from a funding perspective. So Engimmune has shifted to TCR-based T-cell engagers, so soluble TCRs, which essentially are like similar to bispecific antibodies, and that they would target a peptide MHC here, but then they'd have another ligand like a CD3 engager that would drive basically T-cell activation. So it's like a T-cell engager or bispecific, but using a TCR on one side. And there is a clinically approved molecule like this called Kimmtrak from Immunocore. Now, one of the challenges of doing this is that it actually requires extremely high affinity because you don't have that many peptide HLA molecules on the surface of a cell. So you need much higher affinity than you would for a cell therapy, a TCR cell therapy. And so what we did is we first solubly expressed these TCRs and measured their affinities. And one of the interesting things is here's A3A. Its affinity is around 1.4 micromolar, but our engineered variants are, in the 6 or 10 micromolar range, which is actually fairly high affinity for a TCR. TCRs have much different affinity ranges than antibodies. But if we need to make this a picomolar binder to be relevant as a protein therapy, that's a huge, it's a million-fold improvement in affinity we need to achieve while not introducing cross-reactivity to other peptides or to the peptide or to the MHC region, which obviously would be a huge problem. So with a lot of engineering efforts from the team at Engimmune, my lab does a lot of machine learning guided protein engineering. And so we developed a workflow that is basically specificity guided engineering using machine learning workflows in this process. And what I just want to show you is these are some of the campaigns. Sorry for the awkward names, but essentially these are new engineered variants of those TCRs I showed you earlier. And then even on a lymphoma cell line that only has 2 to 3 copies, one of these engineered variants like Gliptodon-2 is showing very strong, is showing the ability to cause activation. This is also using an effector cell in the T cell in the co-culture. And then also using selections here, what I'm showing you is a great example here of Gliptodon-2, where it can show activation with the cell line that is expressing the cognate target peptide. But this is now a beating cardiomyocyte that would have titin present, and it's not showing, really any substantial activation. So this kind of demonstrates that we've been able to really engineer potential candidates in this format as well. Now, very briefly, I don't know how much time I have left. I think maybe 3 minutes. I didn't keep track of it, but I want to see if I can sneak in one more quick story. So I've explained to you, obviously, what I think most of you know what a bispecific T-cell engager is. So instead of a conventional TCR, or what I just showed you with the TCR, it'd be more like a conventional antibody. And there are many molecules on the market like this. Now, one of the interesting and important things that's being uncovered around using these T-cell engagers is really pretty interesting. And it's about functional T-cells. And this is a study from G-Red, Genentech, from a decade ago. And what they're showing you is this mosunetuzumab, which has since been clinically approved. It's a CD20, CD3 T-cell engager. Is that the number of CD8 T-cells, and then, of course, with the bispecific, correlates, the number of T-cells correlates with tumor cell killing, in this case, CLL, which is sort of obvious. More T-cells, the more killing. But in this study, what was really fascinating is they had patient samples, and they used the endogenous patient T-cells, and they showed actually very low levels of killing here when it added with the bispecific. But only when they added healthy T-cells from a different patient, those are these darker lines, do you start to see enhanced T-cell killing, suggesting maybe endogenous T-cells in some patients aren't particularly functional. And then this is another very good study that came out recently using a different bispecific T-cell engager, but showing that essentially patients who respond to this treatment versus patients who are non-responders showed differences in their T-cell phenotypes. They did single-cell sequencing. And what you see here is the non-responders have a much higher propensity to have T-cells that are exhausted or non-functional. So again, it seems somewhat logical. You need a functional T-cell for the mechanism of action. And if a patient has a reduced number of these or a lack of them, one might see a reduced amount of efficacy. And then we also know that there are a significant fraction of patients that don't respond. This is a different T-cell engager glofitamab from Roche. So we did a very simple study in collaboration with Masha Binder at Uni Basel. Super, super easy. First, we just took PBMCs from CLL patients, and we just counted the number of T cells present in the PBMC fraction, which was substantially lower than what you could expect. Or at least these are some of the estimates that are ranged, depending on how you do some of your gating and counting. And then when you actually add different concentrations of this glofitamab, you can see essentially that there's very little activation, right? So you don't see any killing of these CLLs when you're doing this co-culture, okay? So that really makes it see that it's very hard potentially to get patient T cells to be potent when you have a bispecific antibody. So in this project was then we set up a new concept with this using working with a former PhD student in my lab who also has an MD, Edo Kaptanovic. And what we developed is an idea called allogeneic engineered decoupled T cells. And the idea is the concept is basically, can you provide a patient with healthy T cells from a non-related donor, basically an allogeneic therapy? Obviously, if you were to do this, the biggest problem is, one of the biggest problems would immediately be that the donor T cells would have an HLA mismatch and cause graft versus host disease against healthy cells. So what we tried to come up with was a molecular engineering approach where we could somehow shut off TCR peptide HLA-based signaling while preserving CD3 signaling, because CD3 is the arm that is still engaged by one of these antibodies. And so this was a pretty difficult study because the TCR CD3 signaling complex is very complex and difficult. And just if you tried to do something like simply knocking out the TCR, because that could be a possible way, is you can't really preserve CD3 surface expression. They're co-receptors. So simply just knocking out the TCR wasn't going to be a strategy that would work. So instead, what we did is we did a lot of sequence analysis, a lot of screening, and a little bit of structural analysis. But there's a motif that's highly conserved in the J chain across almost all TCR human germlines, as well as many other species. And so we made the hypothesis that this could be a motif that's important in TCR signaling. We tried many different motifs. So I'm showing you kind of the result of success. It wasn't like we just got it on the first try. But anyways, we made 2 mutations in this motif. And then we actually introduced this into first a Jurkat cell line to test it. And what I'm showing you is basically the mutated version of this is called AED. And this is the wild type version using that DMF5 TCR. The DMF5 TCR clearly still binds to the peptide MHC here on the x-axis. And so does our new engineered TCR called AED with those 2 mutations. However, our engineered cell doesn't get activated by peptides of different MART1 peptides unless you go to very high concentrations, but it still gets activated by bispecific blinatumomab. So it means that it has much attenuated activation through TCR, but preserves most of wild type signaling through CD3. So that essentially allows us to potentially remove that allergen graft versus host disease mechanisms. So then we went forward, and this was a set of experiments in collaboration with Elisa Riccio of EPFL, and I'll just kind of fast forward. Basically, the idea is we were able to show that our engineered AED TCR in combination with blinatumomab could result in tumor cell killing in this mouse model. While the wild type obviously also works, but we show that without the blinatumomab, there is no tumor cell killing, and the mice are pretty normal, meaning there was no graft versus host disease or other type of response. And then the last experiment, okay, I'm an engineer, so I can't show fancy clinical data, unfortunately, but this is at least an interesting, very simple experiment, is that we went back to those CLL patient samples, and then we added our AED T cells to the co-culture. And what you see is you actually do start to see tumor cell killing. So while the endogenous patient T cells are not successful, just adding these AEDC cells maybe. And so this, we hope, is someday can be a pathway towards a way to enhance bispecific T-cell engagers by adding these healthy functional T-cells. There's a lot of other challenges we need to address before we would ever get close to the clinic, though. I do want to mention we do a lot of work on what I would call next generation protein engineering, which is combining, like I showed you here, a lot of tools like CRISPR-Cas9 and deep sequencing, and also machine learning. We've applied it for a number of different applications, as well as some things in CAR-T, intracellular and extracellular domain engineering. So always be interested in talking to you about using these platforms in more exciting and interesting translational applications. With that, I really want to thank my group. Highlighted Edo and Rodrigo were really the ones who led those studies, but there was a great team of others who've been supporting me over the years, and our collaborators. And thank you for your time.
Thank you, Sai. Are there questions? Yes. Parrish.
That was really great, thanks. So when you use machine learning to affinity enhance your TCRs, how straightforward is that, and what sort of affinities do you achieve?
Okay, great question. To kind of get you to the technicals, machine learning isn't going to change anything outside of your training set. So it's not going to necessarily allow you to get much higher than what your training data is, which is the experimental screening. But what it allows you to do is to build classifiers. So I can screen against the target, but I can also screen against the off target. And then both of those classifiers allow me to classify a much bigger number of variants that I could have screened experimentally, looking for alternative candidates that bind to the target, but don't bind to the off target. So it just adds many more candidates, because if you just rely on experimental screening for a selection, counter selection, you'll end up with a small number of variants. And if one of those if some of them have another off target or some problem with expression, it's very hard to keep re-engineering. So this just adds more candidates. So it's not magic, it's just allowing you to screen more sequence variants or variants than you could do experimentally. Because there's also a mammalian cell line. We screen libraries in the hundreds of thousands to maybe a million, which is a lot for mammalian cells, but not anywhere close to what you can screen with, phage display type platforms.
And maybe another question to that. So you showed how that would be possible on a plethora of peptides expressed, for example, by tumor cells. But these engineered TCRs, they also then are looked at in the context of the plethora of the HLA class 1 in that case, right?
In this class 1, yeah.
So when you generate one, it's always one class 1 restricted, yeah, and one peptide restricted as this bispecific molecule that's on the market in melanoma, right?
Yeah, so it's always, very narrowly defined as the TCR and cognate HLA and peptide. We do have, although it's not like something we've done a lot of work on, we're still developing it. We've developed an antigen presenting cell line that allows you to incorporate a peptide library genomically. So others have done this. You may have seen work from Stephen Elledge's group, something called T-Scan, and they have related platforms. But what we do, instead of putting a genome library, we can put like a large synthetic library, and again, use machine learning on the peptide part that's compatible with our TCR. Because the reason why we need to do that is I showed you peptide scanning with chemical peptides, array-based screening where you need to, we screened 300, but that was a lot of work, and it's very expensive to order synthetic chemical peptides. In this, with this new system, because it's genomically integrated, you just order DNA oligos, so you can make library sizes in the millions, and the readout is sequencing, so you can do a pooled screening rather than arrayed. So that's the way. We still have this challenge that if you're going to engineer TCR, you need to screen much more off target. So that's why we're trying to develop new tools for that.
Thank you. I do not see more questions, and I'm afraid we have to move on. Thanks again. Fantastic technology, fantastic talk. Thanks. And the last speaker of the session is Chiara Magnani from our department, and she will give a short talk, and afterwards we have a coffee break. So Chiara, please.
Thank you very much. So good morning to everyone. It's a great pleasure to discuss today about how the acquisition of immunosuppressive microenvironment after CD19 CAR T-cell therapy drive T-cell dysfunction and resistance. But before talking about this project, I would like to mention that I developed and have been using the Sleeping Beauty transposon system to generate anti-CD19 CAR T-cells for academic clinical trial. And basically, this is a non-viral vector technology that allows us to reduce the cost and regulatory compliance associated with CAR T-cell manufacturing, but still is achieving stable gene transfer. And with this technology, my former institution, we successfully treated 45 patients with B-cell acute lymphoblastic leukemia. And the results of the phase 1, which was the first one demonstrating potent anti-leukemic activity with this technology, were published in 2020, and the results of the phase 2 were recently accepted. But today, I would like to talk about something that we realized while we were treating our patient. So we know that in B-cell acute lymphoblastic leukemia, more or less 40% of the patients treated with CD19 CAR T-cell relapse. And the relapse is associated, may be associated with CAR T-cell dysfunction. And BLL, as you can see here, is a disease that resides in the bone marrow microenvironment. And as you can imagine, CAR T-cells do not act in isolation, but they act within the tumor microenvironment, which is quite immunosuppressive. However, the respective role of the tumor microenvironment in influencing CAR T-cell therapeutic activity is at the moment undefined in this disease. And something very interesting came out while we were analyzing the hematologic laboratory data from our patient treated with commercial or our investigational CAR T-cells. So we know that normally CAR T-cell peak after infusion, and then you see this contraction phase after 3-4 weeks. But what we found interesting is that we see a similar kinetic in lymphocyte and monocyte, suggesting that CAR T-cells may elicit the bystander activation of an immune response involving both innate and adaptive immunity. And importantly, this innate and adaptive immunity activation was not only mirroring CAR T-cell expansion, but also contraction. And therefore, we hypothesized that the tumor microenvironment was reactive to excessive CAR T-cell mediated inflammation by contracting the immune response through the activation of an inhibitory pathway and molecule. And this could be a potential mechanism of tumor immune escape. And to verify this hypothesis, we perform a single-cell RNA sequencing and also spectral flow cytometry-based analysis of patient sample treated with CD19 CAR T-cell post-infusion compared to pretreatment sample. And the idea was, of course, not to characterize only CAR T-cells, but to look at endogenous T-cells and also the CD3 negative immune niche encompassing the myeloid population and NK population. And you can see here that after CAR T-cell treatment, we observe a strong remodeling of the tumor microenvironment in red post-infusion compared to violet pre-infusion. And particularly, we did see an increase in myeloid cells. Therefore, we analyze myeloid cells, and within this cluster, we observe upregulation of gene pathway that are associated with the immunosuppressive function of myeloid-derived suppressor cells. And this is why we apply a score to our data set, and we demonstrated that myeloid cells post-infusion have a high resemblance to myeloid-derived suppressor cell compared to pretreatment sample. And in parallel to this observation, it was very nice to see that post-infusion, we have an increase in exhausted T cells, NKT cells, and type 1 regulatory T cells. And T cell exhaustion was associated not only to CAR T cell, but also to endogenous T cell, meaning that this mechanism is possibly leading to widespread immune exhaustion. And this was verified in 20 pair patients. Importantly, the PD-1 expression on endogenous T cells was associated with a lack of durability of a response in our cohort of patients, indicating that this acquisition of exhaustion in an endogenous T cell could be clinically relevant. And then we decided to analyze the intercellular communication that can happen between the tumor microenvironment by using this tool called NICHNet. And as a sender, we check the whole cellular component of the bone marrow microenvironment, and as a receiver, myeloid cells here, HAP and CAR T cells. And it's very small, but you can see that we observe a upregulation of hypoxia, TGF-beta signaling, and interferon gamma response, and they seem to really play a key role in the intercellular communication. And we think that this leads to a chronically inflamed hypoxic microenvironment that possibly drives T cell dysfunction. And to verify this in vivo, we generate a combined HSPC humanized turon-bearing model by intrahepatically injecting human CD34 positive cells together with a B-ALL tumor. And in that model, treatment with CD19 CAR T cells verify the upregulation of myeloid-derived suppressor cells and also myeloid cells expressing hypoxia marker. And this was associated with also exhaustion compared to, of course, control mice treated with untransduced T cells. So to conclude this part, we think that excessive CAR T cell inflammation drives the acquisition of an immunosuppressive microenvironment characterized by myeloid-derived suppressor cell expansion, endogenous T cell exhaustion, and CAR T cell dysfunction, and this can possibly antagonize the effect of the therapy. So we believe that we have really to design a CAR T cell that is smarter, that achieve potency without having this mediated inflammation that we know that in the clinic affect the toxicity, but can also lead to exhaustion. And as a final slide, I would like to run through our program here at the University Hospital for treating patients with acute myeloid leukemia. So over the year, we developed a strategy to target the CD117 or c-KIT. We already heard about that today. And the idea is to eradicate the AML leukemia stem cells, but at the same time to allow for a non-genotoxic conditioning regimen before allogeneic hematopoietic stem cell transplantation. And this strategy implies termination of CAR T cell activity to prevent subsequent graft rejection. And we achieve this by using a platform that we call adaptor-based anti-FITC CAR T cell. And as you can see here, basically, CAR T cells are only working with the presence of small molecule adaptor. And in this, and therefore we achieve a precise on-off control. And we are really, I think the clinical data, the preclinical data are really promising, and this approach will be translated hopefully next year. And with this, I would like to thank all the people of my lab, Professor Manz, the director of the department, our collaborator, the funding agency, and all of you for your attention. Thank you very much, and I'm happy to take questions.
Thank you, Chiara. Are there questions? Yes, let's go with the chat.
So the question from the chat is if the signature of the myeloid-derived suppressor cells in your single-cell RNA-seq data resembles published tumor-associated macrophage activation states, yes or no, if there's an overlap or if you check this.
So we haven't check this a lot in our signature because, of course, what I show only as a differentially expressed gene is a limited number of markers, but could also possibly that we have activation of macrophage and with this. But in theory, in the bone marrow, this is the source of monocytes. So what we think is that there is more a conversion toward a myeloid-derived suppressor cell. Of course, in bone marrow, we have also some macrophage, but those should be limited. And thank you very much for the question.
Okay, so if there are no further questions, I think that closes the first session. We are somewhat late, and I'm afraid we will have difficulties to make that good again through the day. So we do a 15-minute break and then come to the next session. Thank you.
Okay, I think we can start. And I would like to give you a very warm welcome to the second session about cellular immunotherapy. And as a first speaker, it's an honor to introduce John Haanen from the Leiden University Amsterdam. And he's going to talk about Claudin-6 CAR-T cells combined to mRNA vaccine for solid tumor. Thank you very much, and the stage is yours.
Thank you very much. Thank you very much also for the kind invitation to be here today to speak about a study that we started a couple of years ago together with BioNTech and a couple of German centers. This morning we have heard a lot about the treatment of patients with heme malignancies. Today I will talk about patients with solid cancer. So here are my disclosures. So one of the problems, if you compare heme malignancies to solid cancers, that CARs have not been very effective so far in that setting. And we know that the tumor microenvironment is quite hostile. We know that there's a problem with the persistence of the cell. Sometimes there's a lack of infiltration, which could be because of physical barriers or some other barriers. And T cells develop rapidly in an exhausted state, which makes them also dysfunctional. So there's quite a number of things that we need to improve in order to make CAR-T cells or any cell therapy work in solid tumors. So one of the correlates with response, at least in heme malignancies, is that there's a correlation with the persistence of the CAR-T cells. And that is probably because these T cells, these CAR-T cells, encounter their antigen very rapidly in the circulation or a lymph node or where the tumors are residing. But it's very different in solid cancers where these cells do not persist, probably because it's more complicated for them to be continuously in contact with the antigen. There's a lack of peripheral antigen there. So how can we improve the engraftment? That's one of the biggest questions. And in order to do that, one could try to induce the expression of the antigen in, for instance, in lymphocytes or primary lymphoid organs, and by that increase the expansion and persistence of the CAR-T cells. So the provision of accessible antigen. So in this study, Claudin-6 is being targeted. It's an oncofetal antigen, which is a tight junction associated antigen, which is expressed during fetal organogenesis, but then epigenetically silenced, but can be overexpressed again in many solid tumors. It's not expressed in healthy tissue, so therefore it's a really ideal target for a CAR in a solid cancer. So this BNT211 is an investigational therapy where the Claudin-6 targeted CAR-T cell is combined with a mRNA vaccine, abbreviated CAR-VEC, which encodes the full-length Claudin-6 protein. So BioNTech developed a Claudin-6 CAR-T cell, which is a second-generation CAR-T cell with a Claudin-6 specific single-chain variable fragment and a 4-1BB CD3 signaling domain. And then it's being combined with a liposomal formulated Claudin-6 encoding mRNA. And then the combination is given to patients. And to the right, you can see that especially testicular cancer and ovarian cancer have very high expression of Claudin-6. So in this case, almost 100% of the cells are being expressing Claudin-6. And what's interesting that not only primary tumors do that, but also in the metastatic setting, these metastatic lesions do express this antigen to a very high level. Now, if you give the CAR-VAC, the Claudin-6 encoding mRNA vaccine intravenously, this will be then going to the secondary and primary lymph organs where it's being expressed in the presenting cells. And by that, the idea is that when you give a high dose of CAR T cells alone, you will have a rapid decline in the persistence of the cells. But if you give them a lower dose and combine it with the CAR-VAC, you can keep the cells within an optimal therapeutic window. That's the idea, and in the mouse models, that works perfectly well. So BNT21101 is a phase 1 first-in-human open-label dose escalation study for patients with relapsed or refractory advanced Claudin-6 positive solid tumors. And we first performed a dose escalation study with a cell product that was generated with a manual manufacturing process, and that was published already, I think, 2 years ago. But then, because of the demand of cell products, the company went to an automated process for production, and that changed the cell phenotype slightly, and therefore we performed a repeat phase 1 study now in 78 patients that I will show you today. So the key inclusion criteria of this study is that the tumor should be, of course, Claudin-6 positive in more than 50% of the tumor cells to high extent, so IHC score 2+ or 3+, measurable disease either by RECIST or by elevated tumor marker, for instance, alpha-fetoprotein or CA125 or ovarian cancer, and a good performance status. You can see here the dose escalation scheme. So either the CARs were given as a single therapy, so monotherapy, or combined with the CAR-VAC, and you can see the different dose levels starting from dose level 0 to dose level 3, and also with the CAR-VAC, we started dose level 1, 2, and I will not today report on dose level 3 plus CAR-VAC because that part is still ongoing and we haven't analyzed all the data. The primary endpoint is safety and tolerability, the maximum tolerated dose, and if it's possible to come up with a recommended phase 2 dose, and secondary endpoints are overall response rates, disease control rates, and also duration of response. So here you do see the schemes. So patients are being screened for Claudin-6 expression, and that's being done centrally. So we just send a couple of tissue slides to a central lab where the Claudin-6 expression is being studied. And if the patient has a high expression, then this is a possibility. And of course, these patients need to have progressive disease on a prior or bridging therapy before they can be enrolled in the study. Patients will undergo leukapheresis, and then the CAR T cell is being manufactured. And prior to infusion, patient will receive lymphodepleting chemotherapy consisting of cyclophosphamide and fludarabine, followed by adoptive cell therapy with the Claudin-6 CAR T cells. And then a DLT period of 28 days follows. In case of the CAR-VAC combination, a patient will receive the first CAR-VAC at day 4 following adoptive cell therapy, and then there will go an interpatient dose escalation from 5 to 50 micrograms to 100 micrograms every 3 weeks for another 4 doses, and thereafter the CAR-VAC will be administered every 6 weeks. So when we look at the baseline characteristics for the safety set, this is a very dense slide with a lot of columns. Maybe I should explain it to you. So on top you see first CAR-VAC only. Maybe you can better do it on the slide. I hope you can see it. The CAR T cells only, and here to the right you can see CAR T cells plus the CAR-VAC. In total, 78 patients were treated. You can see that there's another dose level here, dose level 1.5. Because when we were treating patients with dose level 2, we started seeing toxicities, which were then possibly attributed to the CAR T-cell dose. So we lowered the dose in order to see whether we could limit the toxicity that we were seeing. And in the combination, you can see that we also some dose levels with lower lymphodepleting chemotherapy, so 50% and 75%. Also, that was done because we started seeing toxicities, but it turned out that with lowering the lymphodepleting chemotherapy, the persistence of the cells really hampered. So we stopped doing that and we went back to the 100%. The median age of these patients was 48 years, so quite a young patient population, probably mostly because of the testicular cancer patients and sarcoma patients that we treated. We were usually very young patients. About 46% of the patients were female. You can see also Claudin-6 expression was very high, so the median was 90, with a range of 50 to 200. The majority of patients that we treated were either testicular cancer patients, so almost 39%, and almost 40% of the patients were ovarian cancer patients. But we also treated some sarcomas, non-small cell lung cancer, gastric and esophageal cancer, and some other cancers. The median prior lines of treatment was 4, ranging from 1 to 11, so heavily pretreated patient population. When we look at the safety sets, and again, as you can see here, there were a couple of fatal treatment emergent adverse events, and 3 of those were considered related to the treatment. 1 patient died of a sepsis, 1 patient died of a fungal pneumonia, and 1 patient died of a hemophagocytic lymphohistiocytosis syndrome, which we call now immune effector cell associated hemophagocytic syndrome. We also saw patients that discontinued the treatment. There were 2, and there were in total 7 DLTs. 4 of those DLTs were, again, hemophagocytic syndrome, but there were also patients with pancytopenia, liver toxicity, and a patient with CRS. When we look at these side effects, you can see that this is very in line with what is known for CAR T-cell treatment. So patients will develop CRS, mostly grade 1 to 2. There was 1 patient with a grade 3 CRS, 1 patient with a grade 4 CRS. We did see in 8 patients a grade 3 or higher hemophagocytic syndrome occurring. And when you look at patients with ICANS, we did only see that in low grades, so a couple of patients may have had grade 1 ICANS. And so therefore, we think that this is manageable treatment, despite the fact that we did see quite some toxicities, but did not disturb the dose escalation scheme that we had, and many of the side effects that we did see were in backfill cohorts. So now moving to the efficacy and looking at the best overall response for the total patient population that was available, 74 patients. The overall response rate was 32.8%, with a disease control rate of 67.2%. In blue, you do see the patients here on the waterfall plot that were treated with dose level 2 or higher, with 100% lymphodepletion plus or minus CAR-Vac. And if you didn't look at the 41 patients that were treated, you can see that the overall response rate went up to 51.5% and the disease control rate to 84.8%. So quite promising. Now, looking at the different patient population, and especially the ones that were treated in this study with testicular cancer, again, 27 of these patients were evaluable, an overall response rate of 24% and a disease control rate of 56%. But again, when you look at those patients that were receiving dose level 2 or higher, plus 100% lymphodepletion, plus or minus CAR-Vac, again here in blue, and also in the spider plots here in blue, then their overall response rate went up to 41.7% and the disease control rate to 75.0%. 2 patients had long-term complete remission following a resection of a remaining lesion lasting over 400 and 500 days. Looking at, when we look at ovarian cancer patients, so we had 24 patients with ovarian cancer, overall response rate of 33.3%, disease control rates 75.50%. And again, looking at the blue columns and lines here, you can see that then their overall response rate was 58 and 83.3%. So quite promising results. Now, what was the effect of addition of CAR-vac? And now we only look at patients that received dose level 2 CAR T cells, because that were the most of the patients, the largest cohorts, I must say. And we can compare dose level 2 alone with dose level 2 CAR T cells plus CAR-vac. And what you do see is that you see a decrease in the decline of the T cells in the circulation, adding several weeks of persistence of the cells compared to the CAR T cells alone. And when you look at for instance, day 50 with CAR T cells alone, here 100% of the patients that received CAR T cell plus CAR-vac were detectable above the lower limit of detection, where this was 75% for patients that treated with CAR T cells only. But if you look at day 90, there was only 14% of patients that had a detection above the lower limit of normal, whereas still 75% of the patients were detectable when CAR-vac was given, clearly indicating that you can improve the persistence of these cells. So to conclude, I think that what I've showed you is that it's possible to give Claudin-6 CAR T cell plus or minus CAR-vac, although we do see a toxicity occurring, but these toxicities are manageable and are consistent with what has been previously published using CAR T cells in hematologic malignancies. And we don't see that CAR-vac administration results in increased toxicity. We have seen encouraging signs of anti-tumor activity across all indications, especially in patients with germ cell tumors of testicular cancer and ovarian cancer. And among those who received dose level 2 or higher with 100% immunodepletion, plus or minus CAR-vac, the overall response rate was 41%, 41.7% for testicular, and 58.3% for ovarian cancer, with some durable responses lasting over a year in a couple of patients. CAR-vac improved the CAR T cell persistence beyond 100 days, and the study is still ongoing, and the follow-up of this study as well. And we are currently evaluating those patients that were treated in dose level 3 plus CAR-vac. And hopefully I can report on that later this year. So I would like to, of course, to thank all the patients and families to participate in the study. The research group, so all the investigators and all the different centers, mostly in Germany, but also in Australia and in Sweden, and of course the BioNTech team that made all this work possible. Thank you very much for your attention.
Thank you very much for the great presentation. The talk is open for questions.
Markus.
Yes, so it's very, very impressive data. I have a question for understanding. So these particles deliver mRNA that are preferentially taken up by the antigen-presenting cells, but
yes,
also macrophages.
Well, they go at least to the secondary lymphoid organs. So if you do let's say a PET scan, right, following this infusion, you can see that especially in the spleen and lymph nodes, you get up expression of the mRNA vaccine if you use
and then they produce this claudin, and this is going to the cell surface as a full molecule.
Yes, that's the idea. That is expressed
so
as a full molecule.
MHC restricted peptide expression.
No, it's not, as far as we know. It's expression of the antigen at the cell surface, which is then being recognized by the CAR. Whether it's also expressed by class 1, I don't think we have any idea about that.
So which leads then to killing of these.
I'm not sure that these Cs are being killed, right? Because similar things happen when you have recognition of T cells, or let's say an activation of T cells by dendritic cells.
In the context of MHC, then
less sensitive to T cell killing. So we don't know, but that's clearly not a major effect of the treatment. And mind you that these patients have been lymphodepleted, right? Also, so you have also an impact on some of the cells there. Thank you.
I have also a small question.
Yes.
So it's very intriguing to using a vaccine to increase the potency of CAR T cell, but you also show in your clinical experience that you also see a lot of toxicity.
Yes, we do. Yeah.
Exactly. So what is your learning experience after this clinical trial in terms of also calibrating the dosing of the vaccination during the days in order maybe to skip this first peak of CRS and HLA, if you can a little bit talk
yes,
about that.
So we have tried several things because usually when we see toxicity occurring, it occurs quite rapidly. So days after infusion of the CAR T cells, we see the CRS, which is also typical for heme malignancies. And then usually around day 7 or 8, we start seeing the HLH, which is really probably more common seen in this treatment than we see in heme malignancies. So we have stopped the CAR-VAC infusion when this happens. But for those patients we continue with the CAR-VAC, we didn't see more CRS occurring. So we have not a clear indication that the addition of CAR-VAC really adds to the toxicity that is being caused by the CAR T cells. It could be that the toxicity is prolonged by the CAR-VAC. So we usually, depending on whether the patient develops toxicity, postpone the CAR-VAC infusion or not if they don't have any toxicity. So we try to titrate, but it's complicated to see what the effect on the toxicity is. Yeah.
Thank you very much.
Yeah, there's one question in the chat. How do you balance the need for antigen exposure versus T-cell exhaustion?
Good question. I don't think we have all the answers to that. So in the mouse model, of course, that's easily being done. In a clinical trial, that's much more complicated. We do see that the CAR T cells may start expressing PD-1, but at the moment we have no idea how to balance that at least not in patients. Yeah.
Okay, thank you very much for the presentation and the discussion. To conclude this session, we have a short talk from Cristian Pellegrino, who is working in our department. And Cristian is talking about adaptor CAR T cells in solid tumor.
Thank you, Chiara, for the introduction, and I really appreciate the opportunity to show some of the research we're conducting here in Zurich. And I will be talking more specifically about the adaptor CAR T cells, but just one step back, we saw a few talks on the solid tumor microenvironment and how this is complicated to tackle in comparison to hematological malignancies. And Christos nicely introduced also the commercial CAR T cell products, which now are targeting the CD19 and BCMA, so lineage markers, removing both healthy and malignant compartments of B cells. In solid tumors, unfortunately, the excision cannot be made as such, because if, let's say, a non-vital organ is compromised, then surgery would become the most viable option. So when targeting solid tumors, we have to also think about these issues. Now, on the right, I just posted some of the issues facing in solid tumors. So there's the antigen dilemma. Of course, we're talking always about a single target, but most likely it's going to be a more heterogeneous tumor tissue. And also the T cell fitness can influence the CAR T cell efficacy, as well as the homing and penetration and the tumor microenvironment as a whole. More specifically in this talk, I will talk about the potential antigen dilemma, and we are thinking of tackling this by using adapter CAR T cells. As Chiara also introduced very briefly in our talk, we're using the anti-fluorescein single-chain V. So this is a CAR T cell that is specific for fluorescein, and we use small molecule bispecific adapters to guide our CAR T cell towards known tumor-associated antigens. And in this study, which I'm illustrating now, we published in October 2024, we are looking in somatostatin 2 receptor, which is overexpressed in many neuroendocrine tumors as well as other tissues. And the reason why we are targeting this is I want to focus on SSTR2, as I mentioned, is expressed in both breast, pituitary adenocarcinomas, in neuroendocrine, but also in meningioma and melanoma. We are also leading a parallel study in meningioma tissues, and hopefully we can share this data soon. Now, as I explained, it's made of 3 elements. So the first thing we wanted to validate was the interaction of our small molecule with the target of interest. And to do this, we ran flow cytometry. So you can see on the left, we simply titrated our bispecific adapter molecule on our tumor cells to see when we will reach saturation and to see an interaction between the small molecule and the tumor cells. Importantly, and I stress this every time we speak, we also, when we give this talk, I like to focus also on the type of interaction, because as we look into the design of the small molecule, we really wanted to have a non-internalizing interaction, because of course we do not want to downregulate the protein we are targeting. So we performed confocal microscopy to show that actually when we have an antagonistic interaction, we do not have internalization of our SSTR2 favoring CAR T cell targeting. And we also screened the octofluor, so this is the molecule we're bringing forward, in paraganglioma patient samples and also healthy pancreas, where we know the SSTR2 is expressed on pancreatic isolates. And we can nicely show that the octofluor can bind to SSTR2 on both healthy and malignant tissues. Now, when looking into the experiments to run the adapter CAR T cells in vitro, we looked into titrating as we did similarly with the flow cytometry before, but we titrated the octofluor and we ran cytotoxicity assays for 24, 48, and 72 hours. And you can see the results displayed here in the bar graphs. Very simply, we can show that from 30 femtomolar to 3 micromolar, we have a diverse population of tumor cell killing. And this is true because we have, of course, when you have a low presence of octofluor, so low concentration, we do not favor the immunological synapse formation. And what we can see in our data, and is also shown by the team in UPenn, that there is really a therapeutic window we can see in the in vitros where the adapter concentration is optimal to favor CAR T cell killing. And then we decided to move, once we could see that there was an efficacy in vitro and we really wanted to focus on more in vivo application, we decided to understand better what is the dosage we should give to then favor this this CAR T cell mediated tumor lysis. So in the model I'm showing here, we subcut implanted BON1 SSTR2 expressing cells in NSG mice, and we waited 14 days for the tumor to develop. The tumor developed to a size of 18 millimeter cubes, where we then started the injections of adapter CAR T cells and the octofluor. As indicated also in the slides, we gave the octofluor every 12 hours IV, and this was continued for 2 weeks, and we gave the dosages indicated here, so 12.5 nanomole, 2.5 nanomole, and 0.5 nanomole. And by simply looking at the tumor volume, we could really see that there was a beneficial effect of actually reducing the octofluor amount and dosage. And by actually saturating the system and giving 12.5 nanomole, we completely abrogated the anti-tumor effect. And once we wanted really to look into more the activation phases of our CARs. So how do our CARs respond when we give the octofluor? And we looked into, as shown here, similar model as illustrated before. So we had the subcutaneous implantation and then waited 14 days and we did the injections for 1 week. And at the end of the 1 week, we did terminal analysis on blood, spleen, and tumor tissues. And what we can nicely show here is that we have a really strong activation of our CAR T cells in the tumor site. So this is where the SSTR2 is expressed. And we see some recycling potentially of CD25 positive cells in the spleen when given the octofluor. And when we focus on the tumor and spleen tissues, we can really see that there's also a polarization of CD8 CAR T cells within the tumor lesions, also suggesting what often we've seen also in previous presentations, that the CD8s are really pushing the response towards the tumor cell lysis. And another aspect we wanted to show, because we keep on mentioning that there's this on-off tumor efficacy, and we wanted to demonstrate this in vivo. So what we showed is the exact same model as before, where we injected subcutaneously bone marrow SSTR2, waited 14 days, and then injected our CARs with the molecule. And what we showed is that when we administered for 4 days the octofluor, and then when we halted the administration in one group and kept on administrating the octofluor in another, we can show that from day 17, where we can see, for example, by monitoring interferon gamma in the blood serum of mice, we can show that the plasma content, or the picogram per ml, increases. But then when we keep on administrating and look at day 21, we can see that this continuous expression of interferon gamma increases. Whereas in the group that did not receive the octofluor for the past 4 days, this dose, of course, was reduced. And this does indicate that we can optimally activate and not our CAR T cells upon administration of the octofluor. And lastly, we wanted to focus more on the in vivo efficacy. So we decided to do a long-term therapy where we injected the octofluor for 3 weeks. And we wanted to show also how the dosage, as indicated by our previous in vitro data, how this is really a key aspect in also the speed and efficacy of our CAR T cells. So we kept the 2.5 and 0.5 nanomole dosage of octofluor, and we can show that actually having a lower dose of octofluor, we can see a better efficacy in vivo in comparison to the 2.5 nanomolar, where we could see a delay in tumor eradication. And that actually already brings me to the end of the talk. I would like, of course, to thank Professor Manz for the supervision and the guidance throughout this project. I presented on all of the lab members in the Manz lab. Of course, I'd like to thank also Professor Dario Neri for the help in synthesizing and procuring the octofluor and the collaboration throughout the years. And of course, Professor Chiara Magnani for the support and help, and all of our collaborators and our funding bodies for the project. Thank you so much for your attention.
Thank you, Christian. So last question before lunch.
Not that many for lunch.
Everyone is hungry.
Just one quick question, Chris. Thanks a lot for the talk. I think this is a beautiful platform and super scalable as well. Do you think, and in what setting, you could perform an analysis with, let's say, a commercially available bispecific T-cell engager to compare with your system and benchmark it?
So you would compare, for example, anti-SSTR2 CD3 as an example?
Yes.
Yeah, I mean, I think it's very much a key aspect, because when we bring this forward, we say of multi-targeting, then when you compare bispecifics, you can say, okay, but I can also give a second bispecific targeting. I mean, the advantage here, I could say, the immediate one would be that here we have second generation CAR T cells, so we have a 4-1BB intracellular, so you have also the co-stimulatory advantage that you may not have with the bispecific. I think the jury is still out on this, on whether this is a key effect, but looking at the direction, of course, we see where bispecifics now are looking also in the engagement of 4-1BB-L and C28. I think this is a key aspect which may favor, let's say, more CAR T-cell based systems. Absolutely, but it's a key aspect.
So, ladies and gentlemen, we start at 1 o'clock sharp, please, because we are a little bit delayed. Welcome to the third session. It's cellular immunotherapy number 2. We have some changes. I try to manage it because we have planes and trains that leave. So we will start with Nico Lachmann. He's from Hannover Medical School, focusing on iPSCs and macrophages. And the stage is yours.
Yeah, thank you very much for the kind introduction. And As you can see the title, so I also try to stick to the title, but I slightly changed it. We are switching completely gears now, and I'm not talking about CAR or T cells, and I'm particularly interested in macrophages, and I have to disclose also that I'm not a cancer lab, although we have very much techniques which very much fit to the cancer context, and I would like to give you some examples in my lab what we are doing in the scope of macrophages and also oncology. This is my disclosure slide, and I told you that I'm not a cancer lab, and my lab is in particular interested in diseases where macrophages are not working properly. And we are doing this, and these are mostly rare diseases in children. And we use actually those patients and to use then different kind of stem cells, for example, induced pluripotent stem cells, but also blood stem cells, in order to derive then macrophages and different immune cells, as shown here on the slide. And then to understand, okay, what's going on in those patients? So what's basically the cause why macrophages are dysfunctional? And then to develop new treatment approaches where we either use drugs to enhance or make the cells better, or we use the cells as a cell-based treatment approach in order to treat then the disease. And given the fact that we use macrophages, as mentioned before, this was then why different people approached me also, well, can you use those cells and maybe also in the cancer applications? So before I go on, and this is a very important slide for me because this is actually my motivation, why I'm very much interested in paving the way to use macrophages as a cell-based treatment approach, because James Till and Ernest McCulloch have basically paved the way for all cell-based treatment approaches. And I think this is why I put this slide here to many students and to you also to basically remind you that all goes back to those famous scientists here. And although they have identified the blood stem cells back at the time, the reason why bone marrow transplantation and or blood stem cell transplantation is very much effective goes back to the fact that they can self-maintain their cell pool in the bone and can make all the different immune cells lifelong. But and I would not to go into much detail here, and this is a very busy slide. This actually, the feature of blood stem cells, that they can maintain their cell pool, give rise to different immune cells. Well, actually, this is also true for macrophages. As mentioned, macrophages can be found in different tissues, also cancer. Yeah, and here those cells have a very profound tissue plasticity, and especially also the macrophages shown here in the different tissues, they also have a kind of self-renewal capacity, which then gives me, or why I say, well, macrophages have stem cell features, and this is the reason why we actually use macrophages as a cell-based treatment approach, as a lifelong treatment for those patients. But I will not show any data on this because this audience here is more interested in the cancer context. And here I think it's also very much important to mention that macrophages can maintain different stages of activation. As you know, they can be anti, pro-inflammation, and all the different spectra in between. So it's kind of a rainbow of different stages of activation, what macrophages in different diseases can acquire. And this is very much important, as I mentioned before, for the onset and progression of different diseases, but also in the context of oncology, because you then have the ability actually to target the function or to retarget actually the function of those cells. And now going closer towards the field of oncology, yeah, and this is just an example here, very prominent example published last month, where macrophages can be used as a CAR-Macrophage-based treatment approach in order to counteract solid malignancy. But the question is, where do we get those cells from? In comparison to CAR T cells can be nicely expanded, and we also heard in the morning about the manufacturing scheme. Macrophages are post-mitotic cells, so that means you have hardly any options to expand those cells. And that means that you have to think about different options where you can derive those cells. And I put here 3 different sources where macrophages or monocytes can be obtained from, and then down the road further enhanced by the introduction, for example, of CARs, and then to use those CAR-Macrophages then to counteract certain diseases. So although I showed you here that there are different options, all of those different options here have benefits and also drawbacks. And my lab is in particular interested, as mentioned before, in the use of iPS-derived macrophages, because we can maintain those cells lifelong in the lab, and we are, as I mentioned before, interested in the manufacturing of macrophages and different kind of immune cells, as I will show you in the following slides, and then in particular be interested in the large-scale production of those cells, because scalable production lays then the foundation to translate those cell types then also into patients. And once we have achieved the kind of upscaling, we can then further enhance those cells, as mentioned before, with CARs, for example, or preloading, and then to apply those cells in different diseases. And to guide you through, this is also a very busy slide here, but this is the fundament for the next slides, where I would like to show you how we make those cells. So we always start with induced pluripotent stem cells, as shown here, and then we make so-called EBs. These are spheres, cell spheres, and then those EBs are then subjected into a new medium. And as shown here, those cell spheres, we term then hemanoids. This is an artificial wording of hema for blood and oid for organoid. And the reason why we call them hemanoids goes back to the fact that if you add defined cytokines, and in this context, MCSF, these hemanoids are continuously producing macrophages, and you can have a cell factory which 24/7 over half a year produces cells which then can be used in order to be applied for different disease modeling concepts, and also for novel cell-based treatment approaches. So this scheme here I have shown is basically what we're doing in a small scale. Yeah, small scale means, for example, a 6-well dish, and 1 well of a 6-well dish gives around 500,000 cells per week. This is enough for, let's say, PhD students and for postdocs, but this is not very much clinically, or this is not in a clinical scale. And that's why, so the same scheme here, iPS, EBs, hemanoids, and then the production has been then applied to scalable systems, as shown here. So it's the same scheme where we start with iPS cultivation, EB formation, and then we put those spheres, and it actually looks like a snow globe. And I will show you a video where those EBs then are able to produce macrophages, and those cells can be harvested in a weekly fashion. This is also shown here. So harvest number 1, 2, 3 means week 1, week 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on. And here I can nicely show that independent of the week we get those cells, they have nice morphology on bright field, on cytospin, and they also express nicely the typical macrophage markers. And this video, as shown here, this is the snow globe I told you, yeah. And the snow globe is then those snowflakes are the EBs which are producing the macrophages in a scalable manner. And the smallest device we have can make 1 to 3 times 10 power 8 cells in a weekly fashion. And this is the smallest device. Yeah, and if you have a nice PI which has money, this allows them also for further upscaling and also for optimization, which we are currently doing in the lab. But not every lab has this know-how of large-scale production, and that's why we have been, let's say, inventing or repurposing intermediate-scale production, as shown here, So this is not a falcon tube. It looks like a falcon tube, yeah, but it's basically a 50 ml bioreactor, which actually can also be used in order to make those snowflakes as shown in the large scale. Yeah, and this is a very nice technique where actually this kind of small scale to large scale, now let's say a 50 ml intermediate scale production can be transported and can be used in order to bridge academia to industry. And as shown here also in this video, those snowflakes are the EBs, and once they settle down, the macrophages stay in the supernatant. You just take the medium, put new on top, production goes on, and then you have a weekly production of those cells, which can nicely be used for different purposes. So the quality is quite bad here, but this is basically, again, only to show the manufacturing scheme. What's more important is to show you the function of those cells and how we can make those cells and to apply those cells. As shown here, so we are starting with pluripotent cells. That's very much important, yeah? And down in the differentiation towards macrophages, pluripotency markers go down and early blood cell markers appear before also when we basically harvest the cells are nicely CD45 positive, which is, in this case here, the macrophages. And if we analyze those cells in more detail, this is actually a harvest from the serotube, and we perform logistic regression. Here, the bioinformatical approach shows us that, yes, indeed, the transcriptional profile of the cells which we make are very much overlapping to macrophages, and most importantly, also markers which are associated with macrophages are present on those cells, telling us that indeed we can make macrophages. Most importantly, and now slightly switching towards cancer, what's very much important is also, do the cells maintain the plasticity? So do those cells have the ability to switch towards pro and anti-inflammation? And to make it short, yes, they do that. So we polarize those cells here with different cytokines. There are different approaches out there how you can do that, and the cells can respond to these the cells can respond to those stimuli and can secrete pro or anti-inflammatory cytokines. So we have now a nice technique where we can have either a small scale or an intermediate scale or a large scale platform, which actually serves as the basis for the manufacturing of those cells. So I don't show data here, but this technique, as shown for the production of macrophages, if you just switch the cytokines, can also make platelets, red blood cells, DCs, as we had in the talk before, for example, and also NK cells, where those cells can also be used maybe in the frame of anti-cancer treatments. But now I would like to switch gears a bit and give you some glimpse of where those cells can be used maybe to counteract cancer. And here there are different approaches, as mentioned before, how those cells can maybe further equipped. And this can be, as I mentioned before, also with CARs, for example. There are certain drugs which can be used in order to load the cells, and then the macrophages migrate towards the site of the cancer and then releases the drug on demand at the site of the disease, let's say, for example, in a backpack approach. But I would like to give you some examples for CARs. So before I go on, what's very much important is to understand also the function of, in particular, tumor-associated macrophages, especially in the solid tumor bed, where they basically interfere with the tumor microenvironment, and where they activate actually, for example, T cells or interfere with T cells. This is one very much important aspect, whereas those cells, as we also heard in the talks before, have the ability of phagocytosis. So in principle, we have two different aspects how those cells can interfere with different, or how those cells can interfere and work in the tumor microenvironment. And that's very much important because you then can nicely design approaches in order to counteract the function. Yeah. So if we know phagocytosis is very much important, then you can design CARs, and this is one example here where you can increase the phagocytic function of those cells. Yeah. Whereas if we know, okay, if we would like to enhance also the engagement to the tumor microenvironment, then you can maybe use CARs, which have the ability to induce the secretion of cytokines, which interfere then with NK cells and also the T cells. So it's a toolbox. Yeah? So it's not only the cytotoxic effect of T cells, where macrophages have dual functions. Yeah, and based on the design of the CAR, you can basically skew the function towards the need of a certain certain tumor indication. And these are two examples for the function and for the response. And there's one paper which comes out soon where you combine both. As I mentioned, macrophages are kind of very plastic, and you can do much more and equip those cells with features where they maybe also secrete where they secrete cytokines, and at the same time you enhance the phagocytic function of those cells. So those are three examples, and what we have done, and this is a proof of concept study where we also combined actually a CD19 CAR with macrophages, and here in particular induced pluripotent stem cells. It doesn't make so much sense in the context of CD19, but as I mentioned, it's a proof of concept approach. And we used lentiviral vectors where we where we have basically applied those vectors and have transduced induced pluripotent stem cells and have manufactured, as I've shown you before, macrophages in a continuous fashion. And this is shown here. So the iPS cells, so the starting material, are nicely transduced. The EBs are also nicely expressed GFP, and also the CAR-iMax. And this can be also seen here, that the overexpression of the CAR has no detrimental effect on the production of those cells. So if we then use the CAR-iMax in combination, for example, with a T-cell line, Reddy, also in a very CAR-dependent approach, as shown here in this dark orange bar, the CAR-iMac can nicely phagocytose, and here this is a confocal picture, can nicely phagocytose these Reddy cells and do secrete pro-inflammatory cytokine in a co-culture CAR-dependent fashion. But this is a CAR cell line, or this is a Reddy's cell line, and we actually used, and this is hardly seen here, so we used actually different cell lines which have different expression of CD19 on their surface. And as you can see here, only the cells, or only the target cells which have a high expression of CD19 are indeed phagocytosed. And if we have a low or intermediate CD19 expression, the CARs cannot do the job. So nicely showing also the specificity of our approach. This is a cell line, but we also use actually ALL samples from patients. And here I would like to highlight that we have 4 different patients which have high blast or CD19 positive blast in the blood, and whereas patient 5 has very low amount of cells, and this can be also seen in the response of the CAR-iMac. So actually, CAR-iMac only respond to CD19 positive blast cells in those patients, whereas if we have CD19 low amount of CD19 cells in this blood, the CAR-iMac do not do the job, further proving that our CAR approach is very much specific to CD19. The last slide I would like to share is how do those cells engage actually the CD19 positive cells, and as shown here, this is a single cell SEC analysis where we actually combine 3 different samples, and the first important information is that independent, if we put CAR-iMacs either on Raji cells or on patient cells, the CAR-iMac respawn in the same way. So no matter if it's a cell line or patient cells, the cells engage the CD19 positive cells in the same way, get induced towards a poor inflammatory profile, as shown, for example, here. So these are the patient cells, these are the cell line cells, and indeed, the CAR-iMac react in the same way, no matter if it's a cell line or patient cell. Furthermore, it's a time-dependent process. So as shown here in an RNA velocity, so a time-dependent phagocytosis, the cells are being activated from a neutral towards a poor inflammatory state, also indicating those different processes of engagement, phagocytosis, and killing of cells. By this, I looked on the time and I promised to stay in time. I skipped the slide here. This is my last one. And I would like to thank all the people in my lab, all the different partners, because in particular, working with induced pluripotent stem cells, we requires a team. I thank all funding agencies and, of course, you for your attention. Thank you very much.
Thank you very much. Fantastic talk. Any questions from the audience?
Very nice data. Thank you for the nice presentation. What was the target to effector ratio in your phagocytosis assay with the Raji and the Dowdy?
Yeah, so we actually had done, so I only showed you one plot here. We have done 1-to-1, 2-to-1, and also 5-to-1. Yeah, and we nicely can see the more cells you add, the more they also phagocytose. In this data here, I think it was 1-to-1, if I'm not completely mistaken.
Okay, no, it's very nice. I'm also working on the macrophages, and we also see target antigen expression is really crucial. And I'd like to talk to you later on in the coffee break. Thank you.
Thank you. Sorry for the presentation. I had a quick question. You've shown the data on use human iPSCs for the generation of the macrophages. I was wondering if you can, or if you try to do the same with murine
mouse. Forget it.
Okay.
We started that in 2010. It works, but the economic approaches or the economic burden is too much in order to get enough cells which can be used in order to make any in vivo studies, for example. So it works in principle, but at least in our hands, we have stopped that because the efficiency is quite low.
Because for the CD19 macrophages, it would be also interesting if you include, like in an immunocompetent setting, it might be also eliciting the adaptive.
100%.
But of course.
Yeah. As I said, 15 years back, we started that and we stopped that.
Thank you.
Very, very nice presentation. Something that I quite never understood about the concept of CAR-Macrophages is whether these cells can actually transmigrate a vascular endothelium, because whenever it's T cells or NK, we know these cells are equipped to transmigrate, but macrophages are not. They are tissue resident cells. It's essentially like a fibroblast in essence. So what's the evidence for these macrophages to be able to cross endothelial barriers and transmigrate to tumors?
Well, as far as I know, it's basically the as for every other cell type, the hemotactic signaling. One thing which is very much important to mention also is there are different approaches, and for example, from the Beefy lab, stem cell transplantation, where monocytes move into the brain, for example, to make microglia cells. We are interested very much in pulmonary diseases also, where actually the cells coming from blood stem cells go to monocyte, move to the lung, and then migrate through the epithelium into the bronchoalveolar space.
Those are monocytes indeed.
Well,
so monocytes can transmit.
True.
Macrophages cannot.
That's true, and this is why different people are more thinking about not applying maybe macrophages, rather monocytes, to have maybe a better phenotypic and better function of those cells. We will see if this holds true.
Thank you very much. Unfortunately, I also have many questions, but we have to move on. Thank you very much for this great presentation. Safe flight home. Our next speaker is Evelyn Ulrich. She's heading the Division of Experimental Immunology and Cell Therapy at the Children's University Hospital in Frankfurt. Your talk is about NK cells and CAR, and the stage is yours.
Thank you very much for this kind introduction and for the invitation to Zurich. I really enjoy this symposium very much. And now we come from macrophage to natural killer cells. Here are my disclosures. And I think I can save some time and don't have to go into detail about the history, the long story coming from immune cell discovery to a sort of personalized engineering with CAR in the last decade. So, however, as you know, we have FDA-approved CAR T-cell therapeutics. There are some limitations that have been addressed by previous speakers so far. So, CAR T-cells are almost always still patient individual products from autologous T-cells, sometimes from cancer patients that have exhausted immune cell populations. And there are some side effects we have to tackle. In addition, limited successes so far reported for solid tumors. And I want to emphasize on a sort of possibility to have off-the-shelf immune cell products engineered with CAR receptors. So meaning natural killer cells or the cell line NK92. All these cell populations have also been reported to be capable to undergo engineering to express CARs on the surface. And a potential advantage of CAR natural killer cells would be then that beyond the CAR-mediated cytotoxicity, in cases of antigen loss, natural killer cell will still have an intrinsic capability to recognize malignantly transformed cells, as NK cells also recognize virus-infected cells, and to mediate cytotoxicity by these additionally intrinsic mechanisms. We try to understand these mechanisms. We try to improve NK cells, to engineer NK cells. And one of the best examples so far is the clinical trial performed at MD Anderson Cancer Center, improving CD19 CAR-NK cell therapy with a CAR receptor for natural killer cell that also provides, and that I want to highlight, IL-15 expression to have an autocrine loop for persistence of natural killer cells. These clinical trials showed a 1-year overall survival in the first patients that have been treated of 68%, with long persistence in peripheral blood, especially of high functionally IL-15 expressing NK cells. However, CAR T cells are broadly available now in clinical trials, at least, as you can see here, and CAR-NK cells have still a delay coming to translation into clinical applications. So in Europe, there's one single NK cell study with CAR-engineered NK92 cells in the Goethe University, led by Joachim Steinbach and Michael Burger, based on a CAR from Winfried Wells. Why is that the case? I believe we have this delay because it was so difficult to engineer with lentiviral transduction or alpha retroviral or gamma retroviral transduction natural killer cells, because these cells are prone to be infected by virus. So the VSVG pseudotype lentivirus did not efficiently transduce NK cells. We discovered then that another pseudotyping helps to transduce NK cells. It's RD114, and others reported baboon pseudotyping. And since then, these first clinical trials, or the preclinical approach mainly, and this 1 to 3 clinical trials so far have been possible. However, we are also focusing on non-viral genetic engineering of natural killer cells in my group, because I believe it would be an important tool in case that we have a stable modification using transposon technology that has also been mentioned already beforehand from Chiara and Magnani. And so for CAR T cell generation, and we use that for CAR-NK cell generation. So I don't have to introduce that technology. It's that we use Sleeping Beauty mRNA transposase and the mini circle DNA providing the CAR information. It's based on ancient DNA that has been discovered from fish that has this transposase activity. And we reach using that and applying that with improved protocols for natural killer cells, a stable CAR expression of above 40%. Here I show you some first published CD19 proof of principle data for this stable CD19 CAR expression. What's very important is that the biosafety has been addressed by insertional analysis, but also whole sequencing analysis, and we say safe integration of Sleeping Beauty 100 transduced NK cells compared to the lentiviral transduced NK cell preparations. We see an increase in cytotoxic functionality that is significant compared to non-transduced NK cells. What's always important to us is to have a look especially at this low E/T ratio, 1:1, because that is mainly what we expect. We need to have a high cytotoxicity at low E/T ratios, as it's so the physiological condition that will be met in the body. And here you see an in vivo proof of concept study with 9/6 CD19 expressing cells that induce leukemia in the tumor cell in the mice NSG xenograft model. However, we always wait 4 days that we have engraftment of the leukemic cells in a homogeneous manner in all these mice treated, and then we start treatment on day 4 with CAR-NK cell injection. We need some IL-2 because in these CARs is no IL-15 producing gene sequence. And you see, however, that we have a great delay in the proliferation of these leukemic cells in these mice. In recent ongoing studies, we now want to unravel the cytotoxic mechanisms mediated by Sleeping Beauty-derived CAR-NK cells. We work with different CAR constructs. Here you see examples from my PhD student, Alina. She used a PCASPR system in cooperation with Markus group from Hamburg, where the FRET-based system leads to emission of green and red signal. So for viable cancer cells, yellow signal. And once caspase is present, the caspase cleavage site disrupts the 2 emissions, and an apoptotic cell become green. And you see here how an NK cell that's here traced with a bluish fluorescence and has a red CAR staining attacks the viable leukemic cells and leads to apoptosis induction in one cell, in the next cell, in the neighboring cell. So we really can visualize now in a high throughput screen with AI-based models also the capability for serial killing of CAR-transduced NK cells. You see especially that the dynamics is faster of this killing capability. And we have more apoptosis induction, and we have an increase in this high serial killing capacity of these immune cells. We will further investigate which subset of transduced NK cells is the best killer cell. What's very important to us is to go towards translation, and here we had done primary ALL blasts from different risk groups, and we produced CAR-iNKT cells from 3 different donors. And you see that we always have an increase, a significant increase even, in the killing capacity once the non-transduced NK cells are engineered to express the CAR. And with that in mind, now we tackle more harder to treat cancer, such as acute myeloid leukemia, especially as we also know due to the expression of antigens that's also on healthy hematopoietic cells. And we hope that the natural killer cells can distinguish between self and non-self, or malignant cells and healthy cells. And Navid Alving, a PhD student in my group, has shown that the lentiviral transduction with a baboon pseudotype CD33 CAR vector works very well with 50% CAR expression in the NK cell preparation, with increase in vitro cytotoxicity, but also high increase in the in vivo cytotoxicity. But what I want to emphasize is that we still have a progression of AML in these mice. So just using a CD33 CAR-NK cell preparation is not sufficient to cure the mice. And however, we in parallel already start to go towards GMP-based preparation of these CD33 CAR-NK cell products. With help of Miltenyi, we use this Prodigy-based production system. That's a half-automatized manufacturing procedure. And I go quickly through these slides, but want to inform you that I think for all the additional data that I will show, we already have the protocols now to make a large-scale production or manufacturing of these CAR-NK cells, and the capability in vivo cytotoxicity of Prodigy-derived CAR-NK cells is comparable to that comes from the small-scale CAR-NK production. That's a good news. But as mentioned, we have to improve these CAR-NK cells. And we have learned that immune checkpoints keep in check NK cells, as you know, from T cells for PD-1, PD-ligand 1, or TIGIT as immune checkpoint inhibitor pathways. And in NK cells, one of the most important immune checkpoints is HLA-E-mediated inhibition of NK cells by the inhibitory NK receptor NKG2A. And we have developed CRISPR-based RNP protocols to disrupt CAR-1 gene expression to reduce the NKG2A level on the surface of NK cells. And then we combined it after the transduction with the CAR. We apply the RNP-based CRISPR disruption of the CAR-1, and then we have CD33 NKG2A low expressing CAR-NK cells. And with that, I want to show you that we have an additional increase in cytotoxic capacity in a 4-hour co-incubation assay, and using IncuCyte technology, we really see how fast these dual engineered NK cells kill. Once a tumor cell that's labeled with GFP becomes death, annexine gives them a red signal, and you see we have this change of the GFP expressing viable tumor cells into red death leukemic cells at only 24 hours after starting this co-culture experiment. It also works again with primary material. We have AML blasts from patients with different risk profile. Even high molecular risk groups respond in vitro in our short-term cytotox assay on this CAR-NK preparations. And very often we see that this dual modification has, again, a higher impact and efficiency against these tumor cells. We wanted to know how does it look like in an OCI AML2 NSG SGM3 mouse model. So here we have this xenograft model, but we use this mice that are transgenic for cytokines, human cytokines that support engraftment of AML. So it's even more challenging. Again, we wait until we have signal in all the mice that shows that AML has engrafted. Then we start treatment with the CAR-NK cell preparation and also the dual modified CAR knockout NK cells. And what was very interesting for us was that even if we had still a progression of AML, the bone marrow was free in most of the mice treated with this dual modified NK cells, in contrast to only the single modified, either knockout NK or CD33 CAR-NK cell preparations. And then we went on and we performed a re-challenge experiment with engraftment of this bone marrow from the differently treated animals into another set of NSGM3 mice, and indeed we had no remaining or upcoming AML disease in the mice that have been treated by this dual modified NK cell preparation. So what we believe is very interesting and encouraging, because probably also we tackle the AML stem cells in the bone marrow by this type of treatment. Even if I have to admit, we now perform also PDX models, it's here still a xenograft setting. In parallel, now we address in-depth analysis on single cell level. You can have a look on the expressed gene and proteomics profile that are published. So I will not report on this in detail, but we gain insights into understanding the way of action of this dual and modified and very efficient CAR-NK cell preparations. And what's interesting, because has also been previously mentioned, is that interferon gamma plays a key role for those CAR-NK cell preparations that are most efficient in vivo. And I'm very happy that we had the chance to build up a DKTK-funded innovation program, and now also developing another type of targeting of AML, addressing CLEC12A as a target that's not on hematopoietic stem cells, but also on leukemic stem cells and AML blasts. And you see here the consortium. And in parallel, we had a cooperation with preclinics, and we immunized llama to get VLP or use a VLP-based immunization. And this is the llama Serafina that helped us to generate nanobodies that via yeast surface display technology, we could sort out of this immunization procedure in the llama. We have high affinity binders that are VHH-based short nanobodies, and designed CAR constructs that are more appropriate, as we believe, than the general CARs that are used to generate CAR T cells. So we adapted the CAR design for natural killer cells. Now, with this VHH domain, so for instance, we optimize the CAR sequence and transgene expression. We use now the VHH-based dual CAR system and another hinge, and included also IL-15 into the vector design. And you see first in vitro assays also with low E/T ratio that we call it V4, because it was the fourth clone that we identified from the yeast screen. This VHH-based CLEC12A targeting CAR is more efficient than and single chain variable fragment based CAR coming from therapeutic antibody. And we have very recent unpublished results that also these CARs highly efficient in vivo application in the mentioned NSG-SGM3 xenograft model with OCI AML2. And you see again, we have promising results with mice that are free of AML in their bone marrow. And that's always the most important sign of therapy response in this model so far. And for sure, we come back to the CRISPR editing, and we have some hints that for sure also this VHH-based CAR can be combined with a knockout of inhibitory receptors in the NK cells, which again leads to more prominent killing capacity. And in this national strategy for gene cell therapy, supported by SPARC and B.A.G, we now want to go the next step towards GMP production of this dual modified CLAD12A CAR-iNKT for clinical application. I started with these insights in how few is the number of CAR-iNKT cell trials so far in Europe and Germany. It's a long way to clinical application. I'm happy to work with very engaged colleagues together also in different consortia. And I hope we will find toolboxes and technologies that will help to accelerate translation towards clinical application. And I want to thank my team members. You see them here in Frankfurt at mine, and highlighted in red are those people from them. I showed some data today. We team up also with people working with AI applied technologies from Freiburg, and I think this is very promising. I also thank all cooperation partners and you for your attention.
Thank you very much for this wonderful talk. Great data. Any questions?
Yeah, thank you for your great presentation. I was wondering, because you mentioned that the NK cells, they have the ability to also differentiate between malignant and non-malignant cells, whether you have any evidence that the CD33 approach would be sparing normal myeloid cells that express CD33?
Yeah, unfortunately, it always depends the level of antigen expression. And in case of CD33, also hematopoietic cells will be recognized by then CAR-iNKT cells. I just referred to the natural possibility that in terms of antigen loss, an NK cell will sense, for instance, the downregulation of HLA-E on tumor cells, and also other signals, on this balance of activating and inhibitory receptors. We, for instance, depending on, there's a donor variability, depending from which donor we have an NK cell preparation, we have another level of intrinsic killing capacity against AML blasts from different patients. And sometimes also just an unmodified NK cells kill 30% of a high-risk AML patient blast population, but only 30%. Yeah.
Thanks. Very nice presentation. I have two questions. One about the short movie that you showed, where the CAR T cell or the NK cell was interaction with tumor cells, and what I saw was that there was not a clear polarization of the CAR towards the, let's say, the membrane where there was interaction with the tumor cell. So is the reason why that wasn't the case is that that's different for T cells, because I know in T cells that happens, but can you say something about that? Was that just by looking at it?
Yeah. We also are wondering about that, and we are currently investigating this pattern that we often see. Yeah, sometimes we have the impression the CAR expression is at the site where really the contact at the synapse is built with the tumor cell, but it does not seem to be necessary.
No, it doesn't.
Probably it's also then mediated by fast ligand or other receptors that are present on NK cells in their recognition of tumor cells, and also the high amount of perforin runtime that's released. Yeah.
And the second question was regarding the IL-15. Was that membrane tethered IL-15 or not? Because
we tested different ones, membrane bound and the sushi IL-15, and finally we ended up with soluble IL-15.
Okay.
So I have one quick question. So when you compare it, or what makes the difference? Is it the cytokines, or is it like the knockout, or can you compare the effects regarding the toxicity and survival of your cells?
For the efficacy in vivo, you mean? Yeah, I think it's also the persistence of the cell and the activation status. So we really need activation of the natural killer receptors in addition to the CAR expression. That's our hint we have.
Thank you very much. So we continue. Thank you.
Thank you.
Our next speaker is Helmut Sali. He's a medical director of translational unit immunology in Tübingen. Actually, he's my mentor, my former mentor in Tübingen. Friend. Okay. And he's the academic father of my wife, actually. That's the most important thing here. And welcome. And great physician, great research. And now we will talk about bispecific antibodies in solid tumors. I'm very happy that you are here today.
Yeah. Thank you, Dominic. Good to see you again. And thank you also to Chiara and you for moving me ahead in the schedule, because unexpectedly I have to leave early. But now Markus has asked me to report about our data and concepts on bispecific antibodies in solid tumors. And as you can see, I did not dare to change the title, except that I left out the prostate carcinoma because I would like to show some data also in other cancer entities. So in our view, there is 3 major challenges that prevent thorough therapeutic breakthrough of bispecifics in solid tumors. And that is accessibility, sustainability, and specificity of the induced immune response. So what does that mean? Accessibility. I mean, when we talk about solid tumors, the T cells must leave the bloodstream to enter the tumor site, which they frequently do not do. And that's a problem. If they do not go where the tumor is, then they cannot kill the tumor cells. Then the second issue is lack of sustainability. What does that mean? Most bispecific antibodies available today stimulate T cells via CD3, and that is a problem because physiologically T cells require 2 signals to become fully activated. A first signal via the T cell receptor CD3 complex, and then second so-called co-stimulatory signals, prototypically CD28. And the importance of co-stimulatory signals is highlighted by the CAR-T cells, because CAR-T cells became effective only when the signaling domain contained not only CD3 signaling motifs, but also CD28 or 4-1BB co-stimulatory motif. So if you stimulate T cells only via CD3, they become anergic, they exhaust, and they lose their functional and proliferative capacity, which is a problem when you want to combat high tumor burden, where the T cells must proliferate and expand. And then finally, there is the lack of specificity. Bluntly said, I underestimated how few good tumor antigens there are, because most tumor antigens are also expressed on healthy tissue. And now, when you target the antigen that is expressed on the tumor cell, no, that was not the wrong button. That was. Is that the right pointing thing? Let's forget it. You will manage like this. So if you target antigen expressed on the tumor cells, then you get the desired activity. But the antigen is also expressed on healthy tissue. And there you get undesired activity, meaning not only that the tissue is damaged, the healthy tissue, but also you get on-target off-tumor T-cell activation. And that limits applicable doses. And if you cannot apply enough drug, then you limit efficacy. So what do we think can be done about these 3 challenges? How to improve on accessibility of tumors? We think that a remedy would be to target antigens that are not only expressed on the tumor cells, but also on healthy tissue. And there are, not only on tumor cells, but also on tumor vessels or the microenvironment. And if you do so, then the targeting of the tumor vessels, for example, would facilitate influx of the T cells by opening up the endothelial barrier, may also deprive the tumor of blood supply, and then the T cells can enter and mediate their effects. And as a matter of fact, there are a couple of such dual targets. I've listed here the ones about which I will talk today. There is PSMA, which you may know to be highly expressed in prostate carcinoma, but it's also highly expressed on the tumor vessels in a plethora of different cancer entities. Then there is endoglin, which is highly expressed on tumor vessels, but also on some tumor cells. And then there is my presently favorite target, B7H3 or CD276, which is rather tumor specifically expressed in a whole variety of different cancers and also in tumor vessels and also microenvironment. So, and we always generate target binders, proprietary target binders for these antigens to select the best ones. I'll come to that later. But the first construct we developed following this dual targeting was an antibody that we call CC1 directed to PSMA. It contains a PSMA binder with particular extended reactivity, not going into detail, and UCHD1 based CD3 binder. And this construct was generated and preclinically characterized. The data are published. Only one example, this construct eradicates concentration dependently large established 4 to 8 millimeter flank tumors in NSG mice adoptively transferred with human PBMCs. And this antibody was then GMP produced with public funding, and we evaluated it in a first-in-human trial in patients with metastatic prostate cancer. And this trial now has two sections, one intravenous, one subcutaneous. First, we started intravenously, and we applied the antibody in continuous infusion over 7 days and followed by a 2-week infusion-free period. And during the 7 days of treatment, we had daily increasing doses, intra-individual dose escalation with the intent to enable early patients included in the trial also to benefit from meaningful drug doses. And then, as I said, 2 weeks infusion-free period, and I can already say, afterwards, you're always smarter. Now I would not do it like this again, but I nevertheless would share what we saw. We also applied prophylactic tocilizumab to be able to apply high doses of the antibody. So what we saw, without going into detail, is we saw the expected lymphocyte drop that you see with bispecific antibodies. We saw some CRS never exceeding grade 2, and the other effects can be largely attributed to tocilizumab application like liver enzyme elevation and short-term neutropenia, etc. And what we also observed in the patients treated with the target dose is that all but one of these heavily pretreated patients showed a PSA response, and PSA is the only therapy-relevant tumor marker. So we were quite happy, but we saw that these responses were rather short-lived. So when we applied the antibody, the PSA went down, but then we withdrew the antibody and the PSA went up again. So what we did with the patients was something like this. And this is obviously not what you want. Why is this? And I like these data. So, first I have to say, now we said, okay, let's improve kinetics. And now we are evaluating the antibody as subcutaneous application once weekly, and we achieve continuous drug levels now over the whole time. No time to share the data, maybe another time. What I want to share is some data which look very simple, but I think they have never been shown or observed like this, because they document indeed CD3-induced T-cell exhaustion directly out of patients, so ex vivo. So what did we do? We simply took blood from the patients prior to antibody infusion and then after antibody infusion. And then we evaluated the cells in our in vitro assays. And as you can see, now how do I manage with the pointer? Or you have to do it. So what you can see is that when we treat the patients, or when we use the cells prior to treatment, then you can potently stimulate them with our CC1 antibody. But after therapy, after the 7 days, cells do not respond anymore. And this also holds true for the effector functions. The cells do not proliferate anymore after they have been exposed to longer CD3 signals alone. And they also do not kill anymore. And that's a problem. And then that forced us to conclude that antibodies directed to CD3 alone may be insufficient to combat higher tumor burden. What can we do about it? Now, we had very favorable safety data and quite good efficacy data, and they constituted the basis that the regulatory authority allowed us to conduct a trial in biochemical recurrence of prostate cancer. So when you're diagnosed with prostate cancer, you get definitely, if it's not metastasized, you get surgery or definitive radiotherapy. And then there is no prostate tissue in your body left. And then you're watched. And then the PSA levels very frequently, in 50% of cases, start to rise again before you see some tumor manifestation. That is called a biochemical relapse or recurrence, because where would the PSA come from except from tumor? And then this is a difficult situation. If you wait, then metastatic disease develops. Otherwise, you get anti-hormonal treatment. And let me just say, men don't want anti-hormonal treatment. So we reasoned that would be a situation with low tumor burden where the short-term activation with our antibody might be sufficient. And we are conducting this trial presently as first line of treatment, which we like a lot because immunotherapy has to come early, and we are very happy that we can do this. We apply the antibody twice weekly as short-term infusion over 3 weeks, and then 1 week infusion-free period. We are presently at the 6th cohort, and now we reach really meaningful drug doses. We have so far no problems with the application, even without tocilizumab. Grade 2 CRS and all the others grade 1 CRS and no other relevant side effects. And when I come back, maybe another time I can show you what the efficacy results are. What I want to talk about now mainly is what do we do with a higher tumor burden? And with this, we need to talk about this distinguished gentleman here, whom you can see playing Frisbee in California in the 80s. This is Gundram Jung, my mentor, and he's not only my academic father, but also the academic father of all the bispecific concepts that I'm talking about here today. Because besides playing Frisbee, he already published his first paper on bispecific antibodies in the late 80s. And he also, when I was a young physician, started my clinical education, was conducting a clinical trial evaluating CD28 bispecifics together with CD3 bispecifics. Why? Very simple. As I said, T cells require 2 signals to become fully activated. A signal 1 via T cell receptor CD3 and the second co-stimulatory signal, for example, via CD28. And this may serve to overcome the second big issue, namely to sustain efficacy. And as a matter of fact, we think that by combining 2 different antibody constructs, we may overcome all 3 challenges. How does that work? I already introduced that we think we need to target antigens that are not only expressed on tumor cells, but also on tumor vessels or microenvironment to overcome the problem of accessibility. And we use a construct like our CC1 antibody that provides a signal 1. And then we combine this now with another antibody that provides the signal 2, then we can mimic physiological T cell activation and sustain efficacy. But this also holds another advantage, because if these 2 constructs are functionally intertwined, the bispecific co-stimulator, or short BICO as we call them, does not mediate effects on its own. It has no effect whatsoever alone. So if you now combine them and use these constructs directed against 2 different targets with non-overlapping expression in healthy tissue, then you can increase specificity, meaning target 1 is expressed on the tumor and on healthy liver, and target 2 is expressed on the very same tumor and on healthy gut, but only when you have both antigens in close proximity, you get the sustained T cell activation and increased specificity. So, of course, we have a plethora of data that confirm this concept, which I cannot all show for reasons of time, but a very simple experiment that also proves one very important aspect when it comes to CD28 is this one here. And the important aspect is target restriction. If you hear CD28, the older ones of you will immediately think, oh, De Janeiro, the catastrophic incidents where healthy volunteers received a super agonistic CD28 antibody and did run into some problems, let me say. So what does that mean? The CD28 stimulation must only occur if the target antigen is bound. So how do we prove this? We use target cells that both express PSMA, prostate carcinoma cells, but one does not express endogline as target for our first BICO, and now I try again with the pointer, and the other one does express. So what do we see? When we add low level of our CC1 antibody alone, you see some T cell activation. And when you add the BICO in this setting without its target antigen, then nothing happens. And also the BICO alone has no effect, meaning, or when we look at the cells now that express the target antigen, then you see that addition of the BICO to the CC1, CD3 antibody alone largely increases T cell activation. And that means it acts in a target-restricted manner. What it also does, it induces pronounced proliferation of the important T cell subsets, namely the effector and central memory T cells that you need to combat high tumor burden. And this is by far better than what you can achieve with a CD3 antibody alone. And finally, of course, we evaluated these constructs or this combination in our mouse model where with animals bearing large established flank tumors. And you see when we use a very low concentration of the CD3 antibody that has no major effect in these animals, and notably the CD3 antibodies can cause side effects. CD28 constructs, if made properly, can alone not cause side effects because they do not affect T cells without signal 1. So what you can see with very low concentrations of the CD3 antibody, we can, when we add the BICO, eradicate established tumors. And now, again, I say these data are very simple, but simple things are beautiful. Now, if we take the cells of our patients of our clinical trial again, that were exhausted, that did not respond to the CD3 stimulation anymore. And if we add the BICO in the experimental setting, then we see that the BICO can not only prevent T cell exhaustion, it can also break the tolerance and lift the activity to a higher level than CD3 alone. And this again translates into efficacy with regard to proliferation and then also killing here on the right side. And we think that this constitutes a solid basis for the evaluation of this combination in advanced prostate carcinoma, which we will start within this year. And we will start this, I will not go into detail with the trial. Of course, 2 compounds is always complicated, but we have safety data on the single agent, so we will be fast, and the authorities agreed to this complex trial. Production of 2 bispecific antibodies is more expensive, but we are very happy that the Bundesagentur für Sprunginnovation, Federal Agency for Disruptive Innovation, is supporting our work here with 45 million. And this also led to the foundation of a spin-off company, which would be my disclosure, where we pursue the concept to use 2 targets, 2 signals to make truly effective bispecific antibody therapy. And with this, I would switch to my favorite antigen, namely B7H3 CD276. As I said, this antigen is highly overexpressed in multiple cancers on the tumor cells and in the microenvironment. And to target this antigen, we again made a panel of different target antibodies and then made bispecific constructs. One directed a membrane against the membrane proximal epitope of the B7H3 molecule, and one against the distal epitope of the molecule. And when we compared them with regard to efficacy, we saw that the constructs that target the membrane proximal epitope achieve much better T cell activation and also much better long-term killing. The blue line is the membrane proximal binder here. So it's long-term killing assays. So we say this construct targeted to the membrane proximal epitope has superior activity. But when we looked at cytokines, we see that this construct also induces much more cytokines. And cytokines are a critical thing. You need them to some extent, but they also cause side effects. And it's important to consider in this context how physiological T cell activation differs from bispecific CD3 antibody-induced T cell activation. Affinity of physiological T cell activation is low. That of most bispecific antibodies is high. With regard to cytotoxicity, both are equally efficient, but with regard to induction of cytokines, bispecific antibodies cause much more cytokine release, and this will translate ultimately in CRS and thus side effects. So the aim would be to reduce the cytokines while retaining a desired anti-tumor activity, and this can be achieved by CD3 affinity attenuation. And following this reasoning, we generated an additional construct that contains a 100-fold affinity reduced CD3 binder. And when we use this construct, you see here it binds much less. That's a lower curve to CD3 on the T cells. And when we compare this functionally, this is the data I showed before with the high affinity binders. And we see that the construct containing the low CD3 binder is equally efficient, but induces as desired much less cytokines. So displays a favorable ratio of efficacy and cytokine release, and was thus termed CC3 as our lead candidate, was further evaluated preclinically in a metastasis model. And as you can see, it eradicates lung metastases in the animals, and also again in our established tumor model in these humanized mice, I would say, but Markus would not agree. So in the mice adoptively transferred with human effector cells, then it eradicates the established tumors. And this construct is presently undergoing evaluation in a clinical trial where we treat patients with colorectal cancer, breast cancer, and sarcoma. We apply the antibody as 3-hour infusion. It also contains a half-life elongating mutation. So we apply it once weekly. Plan 16 applications, innovative, as I think, trial design, where we first use accelerated single patient titration. Now we are in the rule-based 3+3 standard titration. We have reached the target dose meanwhile, and we have just been granted an amendment to further increase the dose because it's well tolerated. We also see the first signs of efficacy. But again, also these I have to report another time because I have again to state, you remember, we still believe that we need the twice concept, 2 targets, 2 signals, a combination of 2 bispecific antibodies. And the combination partner for that, what we call CC3, our CD276 CD3 antibody, is what we call BICO3. It's a bispecific co-stimulator directed to FAP, fibroblast activating protein. Why is that? One needs to consider that the tumor stroma consists in 20 to 90% of fibroblasts. So that is a high proportion of the tumor mass, and fibroblasts, tumor-associated fibroblasts associated with a disease progression. But the beautiful thing about these fibroblasts is that they are not transformed cells. So they show no clonal outgrowth, they show less antigen loss, and so we think they are nice targets. The problem is that now when we apply this combination, we target antigens that are expressed on 2 different cell types. And it was important to ascertain that the synapse formation, etc., works also in this setting. But long story short, it does, and I can only show on this last slide that it works. Again, in our animal model, we can eradicate these tumors with the combination of our antibodies. And we will start, the antibodies are produced meanwhile, and we expect to start the clinical trial next year, this combination trial, and that is what I'm really looking for. So, of course, many people have contributed to this, and I have named only a precious few. And of these, I would like to specifically thank Jonas Heitmann, who is the guy who always has to translate what we do into regulatory authority-conform clinical trials. And then, of course, there's Gundram Jung, whom I acknowledged before, my mentor and the father of all these antibodies. Then there are the institutions which funded these projects. In particular, we are happy with Sprint. That gives you some air to breathe in the future. And you, I think, for your attention. And Jonas would be mad at me if I would not show this last slide. This is the presently actively recruiting clinical trials in our department. Thank you.
Thank you very much, Helmut. We have time for one pressing question.
Oh, yeah, okay. Thanks, that was great. What's the off rate for your CD28 and CD3 antibodies?
CD20, which antibody?
The anti-CD28 and anti-CD3. What's the off rate?
So if you would want to know the exact number, I have to admit I forgot. We need to ask Gundram, but we choose them to have a high affinity and to stick to these things very long. It's a very long rate. They need to be there for long because they kind of need to sit there and wait that the other cells pass by, particularly also for other combinatorial approaches.
Okay, thank you very much. For the sake of time, we continue. Our next and last speaker of this session is Heinz Leubli. He's a medical oncologist of the University of Basel, and his talk will be about adoptive cell therapy with tumor-infiltrating lymphocytes.
Thank you very much for the invitation. And we go back a bit in time because these are not constructed cells. So as you will see, this is a therapy that has been long used in some centers, and we have one of the promoter here, John Haanen, who really helped to push it forward in Europe, actually. I will come to that in a few seconds. So I would like to start with a patient that we have seen a few years ago, and this is a classical patient that would receive tumor-infiltrating lymphocyte treatment nowadays. So this was a 62-year patient that was sent from Bern to us to evaluate for a TIL treatment, and he had multiple treatments, including several lines of immunotherapy, checkpoint inhibitors, pembrolizumab, and ipilimumab, and nivolumab, and even experimental targeted therapy. He had some lesions in the lung, in the liver, but also subcutaneous lesions that were previously treated. So he came for this tumor-infiltrating lymphocyte treatment, and if we look how this works, as I said, the cells are not changed. So we take lymphocytes that are in the tumor, excise them, or the tumor is excised by the surgeon, obviously not by an oncologist like me. And then the GMP team cultivates these tumor pieces that are cut into smaller pieces in high levels of interleukin-2. So the basis for this treatment obviously was to find interleukin-2, and shortly after interleukin-2 was found, this treatment was already propagated about 40 years ago. So after this time, so this takes about, depends how fast the cells grow, this takes about 2 weeks. And then we have an intermediate product that we can freeze down. So this treatment is called pre-REP because REP stands for Rapid Expansion Protocol. And then we expand the cells with additional stimuli, including interleukin-2 feeder cells, allogenic feeder cells, and CD3 polyclonal stimulation. And then we have a TIL product, usually between 50 and 200 billion T cells, CD3 T cells, that are then given back to the patient. And obviously the basis, this is from an analysis that has been done by a PhD student in my lab. So the basis of this is that we have T cells that recognize the tumors in this product. So for example, here, this is shown that when you co-cultivate TIL product with autologous tumor material, you see an upregulation of interferon gamma and TNF-alpha. And then you give this product back to the patient that is lymphodepleted, similarly as in patients that receive CAR-T cells with fludarabine and cyclophosphamide. So the patient comes 1 week before we treat the patient to the ward, and we apply this chemotherapy over 5 days. And then the patient receives the treatment together with IL-2. And this IL-2 can be given in different forms. In most of the cases now, it has been given by a high-dose interleukin-2 doses every 600,000 units per kilogram every 8 hours. So this patient came to us for a trial, and here we had used a bit lower IL-2 dose. Yes, you can see it. And had then received directly nivolumab because the question of the trial was to combine TIL treatment with PD-1 treatment directly. The patient had a very good regression of his subcutaneous lesions, also lung and liver lesions. And this is also another patient. This is just to show how it works, just a very impressive patient, young 34-year-old patient with many bone metastasis, which was recently treated, and she had undergone very clear remission, metabolic remission. It was recently also shown by a group in Denmark that the metabolic remission also predicts long-term response to this treatment. So the treatment really works, as I have shown with this patient. And as I said, this is an old treatment, so it has been promoted at the NIH in Bethesda many years ago by Steve Rosenberg and his team. And this is from the first publication in 1988, so it's really a long time ago. And he said that you can administer large numbers of T cells. You can also select for T cells that recognize the tumor if you want, and you can activate the T cells externally. You can also select subpopulation, and you could also manipulate the host before you give them back. What you can also do nowadays with the genetic methods we have, you can maybe in the future also manipulate those T cells genetically before you give them back. And this is from the NIH experience. And as you can see, about 30% of these patients, and these are years, not months, so these are most of the patients pre-checkpoint inhibitor area patients, in melanoma patients. And what you can see, about 30% have long-term remissions, and all the patients that underwent complete remissions, or most of the patients had long-term remissions. So this is also showing that patients that have not responded well to other treatments can still be cured, even in a metastatic setting, with patients with immunogenic tumors. So this is then summarizing all these non-randomized trials that are also randomized and non-randomized trials, what meta-analysis that was recently performed by the group of George Kukos in Lausanne, showing that you have about this 30% long-term remissions over several years or months here. And what you can also see is that it doesn't matter if the patient has been pre-treated with PD-1 blockade or non-PD-1 or is PD-1 blockade naive. So this shows that this treatment works also in patients that have received PD-1 blockade. And then, as I mentioned before, I think this was very fundamental for moving this field forward, also to get now regulatory approvals in different countries, was this academic trial that was run by John Haanen's group and also Ingmar Iswaner's group in the Netherlands and Denmark, randomizing in a phase 3 trial for 84 patients in each group, comparing second-line ipilimumab versus TIL therapy in patients with metastatic melanoma that progressed after PD-1 blockade. And this is clearly showing that here in the progression-free survival, which was presented at ESMO some years ago and afterwards in the New England, that this is an effective treatment that is better than checkpoint inhibitor therapy alone. And as you can also see here from the response rate, there were many more patients having good responses. And I think it's also, again, to say in melanoma physicians that treat melanoma patients, we know that probably patients receiving or undergoing complete remissions, they will have a long-term survival, probably are cured from this disease. So I think 20% is quite a good number in patients that are resistant to classical checkpoint inhibitor therapy. And obviously, if there is something working, there's also a company coming Lifileucel, as you know, is now approved in the US as a TIL treatment, which is a commercial product produced by Iovance, but it's actually following, and I think it's also providing this to many patients. I think it's a very important approach as well to distribute this TIL therapy to many different patients. And this is from the phase 2 trial that was run by Iovance, also showing about 20 to 30% long-term progression-free survival in these patients. And many of those patients had previous checkpoint inhibitor therapy. And this is not only restricted to melanoma. Obviously, this is also working in many other tumors where you have tumor-specific T cells. So, I mean, as I said, the basis are tumor-specific T cells. So, for example, here in a phase 2 trial also run by Iovance, you see patients treated in second line with non-small cell lung cancer. And you can see that even in this difficult to treat patient population, you have long-term remissions. And also this is from a population in cervical cancer, which is virus-driven. disease. Also here you see that patients respond to that. So obviously for most of the hematologists here, that's not difficult treatment because you know how to use high dose chemotherapy, you know what's happening afterwards. For an oncologist like me, it was something to learn. Recently there was very nice review coming out on the different side effects of this treatment. Obviously you have the side effects of cyclophosphamide, you have the immunotherapy, but also have the interleukin-2. And in this nice review you can also see how this is going over time. And obviously we have the neutropenia that is quite short-lived usually, and this is easy treatable or can be easily supported. And also in our department, one of the most feared side effects is obviously this interleukin-2 effects, where we also had some patients that had very bad oxygenation and had to go to the ICU due to respiratory failures. As I said, we have run this trial as a first feasibility trial in Basel because we set up the TIL program about 2018, also with the help of John Haanen's group, but also George Kukos and the MD Anderson. We were able to treat 9 patients in this first trial, as I said, combination with nivolumab. And we also had quite heavily pre-treated patients, I have to say. So many of them had 4 or 5 pre-treatments, not only second-line patients. Nevertheless, we had a few responders. So the first patient here I've shown you, this was the patient that responded quite a long time. Nevertheless, he had a relapse. After 2 years, we have several patients that had only oligometastatic progression that could be treated, and some of them are still living. So to come back to this patient, and I think also from this trial we can learn how we can improve TIL therapy. So this patient, as I said, he had this nice regression on the TIL after TIL treatment. And what we have found then when we took the relapsed lesions or the lesions from the lung that were shown at the progression, we could see that the melanoma didn't look the same anymore already histologically, and we went back to the primary tumor and we found a biclonal disease. And this is actually something that we have seen now several times, that if you have some lesions or some clonal heterogeneity of the melanoma cells, that if we take only one lesion, we might select resistant clones by using these TILs. We also have shown this here, showing the relationship between the different tumors, but also here, if we look here at the TCR clones that invaded here on the right side, the progressing metastasis, we could show that here that most of the clones were not reacting to the same antigens, obviously, anymore, because the TCRs were different than what we found in the original lesion and also in the lesion that were responding to the TIL treatment, showing that we immunoselected the resistant melanoma clones. So also what could be so of course it's a treatment that works, but we still have a lot space to improvement. So to understand the resistance mechanisms, we have also done quite large analysis on the different steps of production, but also after the treatment in our study. And we also have performed, as I have shown before, these reactivity assays, and then we have analyzed sequenced the clones that are reactive to have the TCR sequences. And what we could then do is then track the tumor reactive clones throughout the treatment. So we had here during the production, we can see when we look at the CD8 T cells, and we could track the clones throughout the production. What we can see is that the CD8 T cells shift quite a lot from this exhausted phenotype to an activated phenotype. But especially in patients that didn't respond, it was quite disappointing that once they're back in the tumor microenvironment, they look very similar phenotypically by single cell sequencing as they were before. So we do actually this whole treatment, we put them back, and once they're back in the tumor, they look very similar, showing that probably also tumor microenvironmental factor play an important role here. But what was quite interesting as well was that we found in the blood, in the CD4 compartment, regulatory T cells, which is not so surprising. We used a lower amount of interleukin-2, which stimulates preferentially regulatory T cells. But what we have seen is also that they were not transferred, these regulatory T cells, but with, here there's some, what happens sometimes with if you're not using the the same computer model, but maybe you have to believe now that what we found here is that in the non-responders, what we found actually is that we have seen many more Th17 also in the CD8 TC17 cells compared to the responders. You can say now these are very low numbers, I agree with that, but what we also did is we looked also at the recently published paper from the Kukos lab, but also another lab, and we could reproduce actually this Th17 signature in different populations. And we have then also shown in a mouse model now that if you transfer or co-transfer Th17 cells we can see that the tumors grow bigger, but we also induce regulatory T cells in the peripheral blood in this mouse model. At the moment, we don't exactly understand how this works, but we're investigating this in vitro and in the mouse model that we established. So having these regulatory T cells, obviously our next trial, or having this association between non-response and regulatory T cells, our next trial aimed to reduce the proliferation of regulatory T cells. And we teamed up with a company called Anaveon that has licensed the product that was actually also found here in the University of Zurich by Onur Boymann's lab originally, which shields the binding position of IL-2 to the alpha receptor CD25 of interleukin-2 of the interleukin-2 receptor, and preferentially stimulates CD8 and NK cells. So what we are now running is this base TIL3M trial, which combines here TIL treatment together with this product. And what's also good is that the stability is much better because you have an antibody here, so you have to dose it only twice, not several times like the high dose interleukin-2. This is from the first patient. He had some response here. We also didn't see regulatory T cells in this first patient. Unfortunately, if we looked by flow after in the other patients, it was not true. In the non-responders, we always have found regulatory T cells in the blood, actually. I'm not sure if it's really only this interleukin-2 which plays a role, but we will see. This is an ongoing trial. So basically, to improve, I think, TIL therapy, there are two things we have to do. So one is obviously to select the patients properly. So this is also a nice review, which was recently published to show which factors are already known to influence TIL efficacy. So one obviously is how many T cells you have, how many antigens you have in the tumor, but on the other side also how you treat the patients. And I think at the moment we're probably mainly, and also after some discussions with the groups that have more experience than we have, we're selecting mainly about patients that have not so rapid progression. So we tend to have patients that have a slow progression, to have also some time, probably patients that also responded to immunotherapy before. And what we also have found, and this we had to learn, we had some patients with brain metastasis because we didn't exclude them initially. And I think also there is a time issue. If you have big brain metastasis, the prognosis is so bad that we usually now exclude those patients from the treatment. Obviously, biologically, we can, and this is also from the group of John Haanen, you could change, I think the citation is wrong, I have to say. I think what you could do, obviously, you can work on the product. You can also genetically modify, for example, the product. And this is something which has been tried, not so successfully so far, but I think this could be something really interesting in the future if this is a more common therapy. And what you could also do, as we started to do, is to change combination partners, but maybe also change one thing which is also tested now, for example, in CAR T-cell treatment, to change the conditioning regimen or the preparation lymphodepleting regimen. These are things we really can work on. And yeah, let's hope that we can treat many more patients with this treatment. So I think to summarize, I have shown you that adoptive cell therapy with tumor-infiltrating lymphocyte works. It has received FDA approval. We will see what happens in Europe. Obviously, in the Netherlands and in Denmark, TIL therapy is already available. In Switzerland, we're fighting a bit. And other indications are following, so we have to get used to this treatment as physicians. Also, we will see those patients more commonly on our wards, and I think next generation TILs and combination therapies could overcome resistance to TIL therapy. So I would like to thank all the people involved in Basel within the newly founded University Center for Cell Therapies, and obviously all the people in my group who helped to analyze and produce the products. Thank you very much.
Thank you very much, Heinz. I think it's an important therapy, and we already discussed the hurdles a lot of times. Any other question?
Thank you. Very interesting work. The first question, quick. Why IL-2 is needed? Injection, IL-2 injection, because you have already expanded so much in vitro. Yes, the T cells. Why do you need inject IL-2 into patients? Because the most likely toxicity comes from IL-2 injection. Yes.
I mean, it has been tested also different dosings of interleukin-2. Of course, the side effects are much stronger the higher you dose interleukin-2. And it has been shown that it is needed. I mean, there's meta-analysis now showing that the high-dose interleukin-2 patients actually, they're doing much better. So I think we're really going back now from de-escalating the interleukin doses at the moment to even use all, for most of the patients, high-dose interleukin-2. It seems like that they need that to expand and also to proliferate in the patients. John, you can add.
No, it's always a difficult question to answer because there are very few patients treated without interleukin-2, and it seems that the responses are worse compared to the ones treated with IL-2. But once you have a program that works, it's very difficult to leave out essential components, right? I have a question. I think very nice presentation about the Th17 cells. So have you looked whether these were bystander T cells by the TCRs, or were these tumor reactive T cells?
So we have looked at that, and there are bystander T cells.
Great talk. So Heinz, the problem, as with other therapies, is that you need to take out cells, you need to expand, etc. And in theory, you would say these cells do get signal 1. The TCR interacts with peptide MHC, and for some reason they don't act sufficiently. There might be checkpoint inhibition that can be dealt with checkpoint inhibitors. And then we talked about signal 3, right, cytokines. So would there be a theory to deliver local co-stimulation to get the TILs at site going?
I mean, obviously this has been tried with stimulating molecules. At the moment, I think I agree. I mean, it would be much more feasible to have proteins that you can deliver. Or are you talking about combination of TIL therapy, or rather just instead of doing the whole
instead of taking them out and
sure. I mean, I agree. I mean, it's a lot of
I mean, they see signal 1. They lack signal 2 and signal 3 at site, obviously.
I mean, obviously, many of the immunotherapeutics are approaching this, right? I mean, there's this co-stimulatory molecules like CD28 that have been tried for 1BB, that have been tried. I mean, many of those trials failed, at least in solid tumors so far. Also, cytokines have been or are under investigation. I agree. I mean, if you could stimulate the T cells in situ instead of taking them out, it would be much easier. Of course, I mean, on the other side, you have better control how you manipulate the cells. And I think maybe it's going to be a combination of both. Or once you take them out, you genetically modify them. This would be also an option. I think this has not been tried so much because I think the whole process is quite cumbersome, and to add a genetic modification to this process, I think it's even additionally GMP level difficult.
Thank you.
Yeah, so if there are no further questions, I suggest to close the session and we have a coffee break.
Pleasure to introduce the next speaker, so Marjolein van Egmond. It comes from the Netherlands, is a professor in oncology and inflammation at the University Medical Center in Amsterdam with a research focus on innate immune cells and antibodies. And you're presenting today, if the title has not changed, on engaging neutrophils for cancer immunotherapy. Looking forward to your talk.
Thank you very much. Now, the title hasn't really changed because that is exactly what we would like to do. We like to include neutrophils in anti-cancer therapy. And you might, oh, these are my disclosures. So, but you might wonder why. So when we started this work, basically at that time, neutrophils were sort of non-entities in cancer. And now they even have bad publicity because now it's kind of considered that neutrophils, they are immune suppressive. So they can suppress T cells. They can be involved in metastasis induction. So why would we then want to target neutrophils? That is because all these negative function of neutrophils in cancer is in the absence of antibodies. Because with an antibody that targets a tumor-associated antigen, neutrophils can effectively kill tumor cells because neutrophils express Fc receptors and these antibodies form a bridge between the neutrophil and the cancer cell. And one big advantage of neutrophils is their sheer number. So approximately already like 60 to 70% of the circulating white blood cells are neutrophils. And this is only the tip of the iceberg. So if you treat these patients with GCSF, so very simple injection, no complicated isolation or culturing, but you give them GCSF, you will mobilize an enormous army of neutrophils from the bone marrow. And where it was previously thought that neutrophils were a kind of boring cell because they would go into the tissue, kill a bacteria, and then die, it now actually becomes clear that they can secrete a lot of chemokines and cytokines that will also influence other immune cells. So in this way, we would like to generate kind of an in situ vaccination because we know they can activate and attract dendritic cells, and hopefully in this way, also other immune cells can then do their function. So these are a couple of examples of antibodies that are currently used in the clinic, and they have one thing in common. So even if they all target different antigens, they're all of the IgG isotype. So what happens if you add neutrophils to tumor cells with an IgG? So in green, you see the A431 cells. So they have a very high expression of EGFR. In red, the neutrophils, and this is in the presence of cetuximab, which is an anti-EGFR antibody, an IgG antibody that is used in the clinic to treat advanced colorectal cancer or head and neck cancer. And you can see that the neutrophils, they make a valiant effort. So they bind to the tumor cells. But this is a recording of 40 minutes. And in this 40 minutes, really nothing has happened. In fact, it reminds me a little bit of this. And sorry, we were not able to test the sound. So either there's no sound, it's moderate, or, well, you are probably awake. So but it does remind me of this. So these are some mostly men reenacting a battle. And as you can see, there is an attack. It's not very effective. Might even say maybe a little bit embarrassing. Because if you are going to unleash what is in essence the biggest army of the immune system, wouldn't you rather have something like this? And this is exactly what happens if you change cetuximab into an IgA. So with just changing the isotype, you get this phenomenal effect. So we always also get a question about, okay, these were healthy donor neutrophils. How about neutrophils from patients? So that's shown over here. Let me see if this works. Well, so it's, what's the, which one? I don't know. Oh, well, I'll put it, yes. Okay, I'll do it like this. Yes, yes. So this is an ADCC experiment where we used neutrophils from patients with colorectal cancer in 3 different concentrations of antibodies. As you can see that if you increase the concentration, you get more killing. especially in the case of the IgA cetuximab version. And this almost reaches the maximum amount that you would get if you use neutrophils from healthy donors. So that's shown in this dotted line. So with IgG, you get far less killing. And also this is what you approximately will get with healthy donor neutrophils, but you see you don't reach that. So especially, again, in the case if you use IgA anti-EGFR, you get very nice killing, even with the neutrophils from the patients, which might be immunosuppressive in the absence of the antibody, but with the antibody, they are really effective. So we did a lot of different assays. I'm not going to show all of these. And we compared a lot of different things by activating neutrophils, either via IgG or IgA. So there were some functions where we did not find any differences, like phagocytosis or the release of reactive oxygen species or neutrophil extracellular traps. But if you looked to the signaling, so the calcium release, tyrosine phosphorylation, you saw much more activation by cross-linking the IgA receptor, so that's Fc alpha receptor. We also saw more release of chemokines, cytokines, metabolites. As I've shown you, you see a lot more killing of tumor cells. And very importantly, IgA induces neutrophil recruitment because if you cross-link Fc alpha receptor, you get release of L2B4. That's a very potent chemoattractant, and that's why you get the swarming of the neutrophils. So of course you would like to test this in vivo, but there are some major challenges involved. The first challenge is already that we all know that mice are not men, and that particularly holds true for Fc alpha receptor, because even though mice do have IgA, they do not have a homologue of Fc alpha receptor. That part was actually solved quite a long time ago when I was a PhD student in the lab of Jan van der Winkel, and we generated an Fc alpha receptor transgenic mouse model that expresses human Fc alpha receptor on mouse neutrophils. But this is only half of the system, because human Fc alpha receptor only binds human IgA. So you also have to have your source of human IgA. And this is what happens. So this is a B16 melanoma model, a liver metastasis model in this case. So the cells express GP75. There's an antibody that's called TA99 that binds to GP75. And if you inject human IgG1, you see that over time, the antibody accumulates in the tumor. And also, if you look in the circulation, so for about 8 days, you have relatively stable titers of the IgG1 in the circulation. After these 8 days, mice will develop MAHAs, so the mouse anti-human antibodies, and then they will clear really fast. But you have a window of about 8 days. So if you inject human IgA in a mouse, you can see that already after 8 hours, almost everything is gone. This is presumably due to the very high glycosylation of IgA, and that the liver, the glycopeptide receptor, will just filter the IgA. But that means that the IgA doesn't even have the time to reach the tumor and accumulate in the tumor. So that's a major challenge. So on the one hand, IgA is a very potent stimulus to activate neutrophils. You get all kinds of pro-inflammatory action. You get a lot of killing. And very importantly, you get migration of neutrophils, but the short half-life is really an issue. So that's why we turned to bispecific antibodies. So these are basically IgG1 molecules that on one arm can bind a tumor target. So this can be EGFR, HER2, or GP75. The other arm targets the IgA receptor, Fc alpha receptor. So this will initiate recruitment of the neutrophils. But it is still an IgG, so it can interact with FcRn, so it gives a normal half-life, and it can also still interact with Fc gamma receptors on NK cells and macrophages. So, of course, we wanted to test this, and we decided to make it a little bit more difficult for the neutrophils. So in the previous experiment, we basically coat the tumor cells on a plate, and then we add the neutrophils, and just by gravity, they will fall down and come in contact with the tumor cells. But in this case, we use a three-dimensional culture system. So we embed a spheroid in a collagen gel. There's no direct contact between the neutrophils and the tumor cells. And then we can add the bispecific or not. So on the left, this is what happens if you do not add any antibody. So after about 4 or 5 hours, you see some brave explorers. So there are some neutrophils that reach the tumor spheroid, but nothing really happens. So if we add the bispecific, and I have to say, I'm a great fan of neutrophils, but even I had to blink a couple of times because this is what happens. So this is actually quite spectacular. So, and also if you look to the expansion of the spheroid, you can really see that in the absence of the bispecific, over time, you really can see that the spheroid is sort of invading into the collagen gel. So they really, the tumors are maybe growing, they're spreading out. And this does not happen, or maybe a little bit, but far less if you've added the neutrophils and the bispecifics. So you can really see that the spheroids sort of stay more or less the same and do not spread out. So of course, this is a difficult system to really determine if the tumor cells have been killed. So we are going to also do this in a longer period and see if the tumor cells grow out again. But it looks like a lot of things have happened. So we also looked at its interaction with dendritic cells. So neutrophils have a very special way of killing. So basically they rip off little pieces of the tumor cells. So that's referred to as trochocytosis. And if you rip off enough of these parts, then the tumor cannot maintain homeostasis and they will die. So you get a lot of spillage of tumor antigens, we think. So then we think that this might be taken up by dendritic cells, which then might mature and then ultimately activate T cells. So in the first step, we looked at this trochocytosis of the neutrophils. As you can imagine, it's quite difficult to see because in minutes, basically the tumor cells are covered by neutrophils. But if you look very careful at the edges, you can really see that these neutrophils are ripping off these green particles. So they're ripping off parts of the tumor cells. And again, if you quantify this, you see that it happens quite a lot in the presence of IgA or the bispecific, but not as much in the presence of the IgG cetuximab. So indeed, you get a lot of this trochocytosis. Then we also did the experiment in the presence of dendritic cells. So the tumor cells are labeled in green. The dendritic cells are labeled in red, and the neutrophils are not labeled. And this is in the absence of antibodies, so you can clearly see 3 distinct populations. But if you add the bispecific, you see that the tumor cells are gone. The neutrophils have now become positive because they've taken up all these particles of the tumor cells. And you can also see that the dendritic cells have taken up a lot of these particles. Then we looked at some maturation markers. So you see upregulation of CD83 and CD86. We did not see any difference in HLA-DR, but we saw that DC sign is downregulated, but is consistent with a more mature phenotype of the dendritic cells. So if you combine all this data, then basically if you look for IgA, you get a lot of migration of neutrophils, very much killing, but IgA cannot activate NK cells because NK cells do not express Fc-alpha receptor. You get poor antibody-dependent phagocytosis by macrophages because macrophages have a very low expression of Fc-alpha receptor, and you have very poor half-life. On the other hand, IgG can very effectively activate NK cells and macrophages, has good half-life, but is very poor in activating neutrophils. And you also do not get this migration. And the bispecific basically combines all these features in one molecule. Of course, then the key question is, does it work in vivo? So at least if you inject this in these mice, it has similar half-life compared to the IgG, so that works. So in this case, we use a subcutaneous model of this B16 model. In the middle are the individual mice, and these are the averages. So if you do not treat the mice, or you just give PBS, you see that the subcutaneous tumor grows out. If you treat with an IgG, you see that there is reduction of tumor development, which is the same if you use the bispecific in a non-transgenic animal. So these mice do not express Fc-alpha receptor, but that antibody is an IgG, so then it really acts as an IgG. And if you then treat transgenic mice, mice with Fc alpha receptor, now you see the biggest reduction. I have to say this was a prevention model, because if you have a large subcutaneous tumor, neutrophils have very much difficulties getting in. So then we switched to an intraperitoneal model. So for this we used a colon carcinoma model. And again, we use antibodies or bispecific antibodies that are based on cetuximab. And unfortunately, murine EGFR does not bind cetuximab. But if you change it at 6 locations, it does. So these cells have been transfected with a murine EGFR that has been adapted to bind cetuximab. And then we inject these tumor cells, let them grow out for a week, and then we treat for another week. And you can see that the mice that were treated with the bispecific have less a tumor. We also performed this experiment in collaboration with Mara Steinkamp in the US. She has a humanized PDX model of ovarian carcinoma. So in this case, the target is HER2. So what they did is that they give the spheroids of this PDX9. Then for a couple of days, especially in the total of 3 weeks, the mice are treated. And just before the first treatment, so day 6, and halfway through, they inject a transferrin probe, so you can visualize the outgrowth of the peritoneal metastasis. And again, what you can see that, of course, before the treatment, all the mice have developed tumors. And after treatment, not all mice, but some mice really seem to have cleared the tumors. And you can quantify this, unfortunately. So as the imaging was done after the 4th treatment and not at the end point. So by the end point, we had to sacrifice some mice because of humane endpoints. So that's why we have less mice to analyze. But you can see is that 3 out of 4 mice that were remaining, that were treated with an isotype control, had still growth or increased development of tumors, whereas 4 mice out of the 5 that were treated with the bispecific now have less tumors than they started. So we think this is very promising. One thing is it's not perfect yet, so we're working on that, but you have to realize that doing work with neutrophils in mice is extremely difficult, because mice have about 7% of circulating neutrophils, whereas humans have 70, so 10 times more circulating neutrophils. And even in these humanized mice, if you're lucky, you can reconstitute up to 25%. You never reach the 70% that you have in humans. So we think that by treating 1 week, knowing that these mice have so limited numbers of neutrophils, this is actually quite encouraging. Obviously, there's still a long way to go, but we are very hopeful. We also analyzed the immune cells in the tumor and the ascites fluid. And interestingly enough, we did not find any differences in the tumor. But we did see some slight changes in the ascites fluid. So CD11B is a myeloid marker with macrophages and neutrophils. So we saw a slight increase in the mice that were treated with the bispecific. And we also saw a slight increase of the neutrophils in the ascites of treated mice. So it may not seem much, but if you correlate the number of the neutrophils in the ascites with the size of the tumor, you can see that if there are a lot of neutrophils in the ascites after treatment, you have very small tumors. And if the mice did not have a lot of neutrophils in the ascites, you have much bigger tumors. So we really think that the neutrophils play a very important role in killing or getting these tumors smaller. But it does seem that they're not actually coming from inside of the tumor, because we did not find any differences in the tumor, but it looks like the metastases that are in the peritoneal cavity are attacked from the outside. So basically from the neutrophils in the ascites that will then attack the peritoneal metastases that are sort of stuck in all the organs. So, of course, if you would have been able to solve the problem with IgA, then probably IgA would be a very good molecule. But for now, we chose to go for the bispecifics. because they combine all the features of IgG and IgA. We have normal half-life, and if you treat mice with a bispecific, you get reduced tumor development in vivo. And this is my team, so I'd like to acknowledge some people. Sharon did the beautiful movies with the spheroids. Celine and Gizem did the interperitoneal models and were helped by Mandy and Richard. And I also especially like to acknowledge Kester Viersson from Sanquin, because he helps us with the development of all these molecules, and Mara Steinkamp because of her IP metastasis model. Thank you for your attention.
Thank you very much for the great talk. An interesting concept. Are there questions? Christian?
Thank you so much. It was very, very interesting. I was fascinated. So you mentioned that there might be a T cell involvement, like from monocytes, so from the neutrophils, then the dendritics, then potentially T cells. In your models, did you try running depletion studies on like CD8, CD4s, NKs, and seeing whether the adaptive immunity is also involved in the tumor response?
Not yet. So the difficulties with these models. So first of all, there were short models. So they grow out very aggressively. So we have like a window of 2 to 3 weeks. So there is not a lot of time for the T cells to get involved, but it's definitely something that we're working on to see if we can get better models that we can sort of extend for a longer period. And then, of course, we will definitely be interested to see that. So what we did do, we have mice that have green fluorescent neutrophils and red fluorescent dendritic cells. So if you look at tumors that were treated with the bispecific in these mice, you really see an increase of both the neutrophils and dendritic cells. Maybe with a little bit of wishful thinking, they might be in contact with each other. So it's definitely something that we will now go into much detail to look into that, yes.
Thank you. Another question over there, John Hahn.
Very nice presentation. When I see the enormous influx of these neutrophils coming in, and I think about with my head on T cells, I would be extremely afraid of cytokine release syndrome. Is that something that can be expected from such a massive attack of neutrophils?
I'm sorry, can you repeat it again? I couldn't hear.
So if you look, for instance, for T cells, if that would happen in patients, you would be afraid of a severe cytokine release syndrome.
Yes.
Now, this massive attack by the neutrophils, I'm sure, releases also a lot of cytokines, chemokines. So are you afraid of the inducing a lot of cytokine release syndromes in these patients?
Possibly, yes. So in essence, of course, there was only one way to find out. But this idea was also partly sort of adopted from our work on autoimmunity. So we've seen similar phenomena, at least in the mice with rheumatoid arthritis and also ulcerative colitis. So in ulcerative colitis, you see a lot of neutrophil influx and a lot of tissue damage. And I'm sure that these patients don't feel great, but they don't have cytokine storm. So hopefully if you I would therefore definitely go for a solid tumor and not for a leukemic tumor, because I can imagine if you systemically activate all your neutrophils, that would be really dangerous. But as long as it's contained somewhere, we hope that this won't occur. But I would definitely always recommend starting with a very low dose, just to be sure. Yes.
One question.
Thank you very much for a great talk. I have one question. Does granulocyte colony stimulating factor work with mice? And if yes, did you try that? And did that have any influence?
Yes, it does work, and sometimes it's helpful, but you will again never go on beyond 25 to 30%. So you somehow you don't go over that number. And actually, for the PDX model, it was not necessary because the tumor cells themselves secrete GCSF. So there you actually did see there was like 25, 30% of neutrophils in the circulation, because normally it's only like 2 or 3. So you do see an increase, but again, you never go beyond 30%. Yeah.
One more question, Markus?
I was just wondering whether, following what John said, like, do you see kind of a cold abscess formation where formerly a tumor has been?
Yeah, no, we haven't seen it yet, but as I say, in mice, it's really not as effective as in humans, or at least with human cells. We have a blister model in ears of the mice, and then we actually do see that they develop big ear blisters. So it may not be completely harmless. But I think that for all cancer therapy, of course, there are major side effects. And unfortunately, and that's, I think, with all these therapies, you have to go through the whole process, and then there's only one way to find out. So, yeah.
Many thanks for the talk. And then we move on to the next speaker.
Yes, it works. So it's wonderful to be here with you today. So thank you, the organizer, really, for inviting me to talk today. And I'm going to talk about iNKT cells. So we are going back a bit to CAR-immunotherapy again. Clearly, this is an audience that doesn't really require that much introduction about CAR-immunotherapy. Let's see if I can make it work. Does it work? So hopefully, 1 second. Yes. Now it should work. Yes. So we are all aware about the unprecedented responses that CAR-immunotherapy has induced in relapsed or in refractory B-cell malignancies. And clearly, I don't need to show how a CAR works, but the point here I would like to emphasize that currently approved approaches uses T-cell and reprogram autologous from the patient. So reprogram autologous T cell, they are engineered with a new CAR that can specifically recognize this antigen and given back to patient to specifically attack their tumors. So very excellent responses, but also ongoing challenges, particularly if we think about solid tumors. And this is just to summarize some of the challenges. So we know that autologous T cells are very often dysfunctional because of prior chemotherapy or also ongoing disease. If we wanted to use donor T cells, we heard about graft-versus-host disease. So naturally, T cells are associated with an unacceptable risk of GVHD as well as rejection. Whether autologous or allogenic, they do require deep lymphodepletion prior to infusion so they can engraft, proliferate, and persist. And still there is a microenvironment barrier we heard about. And this is why I wanted to find an alternative approach focused on a new cellular platform that ideally should be from a healthy donor. Healthy cells are usually more functional. Ideally, we would like these cells, though, free from any risk of GVHD and rejection, and we would like them to play with the immune system in the recipient without requiring to eliminate these cells. And obviously, we want them to home to tissues and attack the microenvironment. And so this is why I started to get interest into iNKT cells, which are invariant natural killer T cells. And you may be familiar with them if you are a transplant hematologist or been working in a transplant setting. Now, the reason is that they are not natural killer, but they do express natural killer receptors. And they are still a very special subset of T cells in that they express a T cell receptor, which is characterized by an invariant alpha chain. And this invariant alpha chain is specifically recognizing a monomorphic MHC molecule, which is called CD1D and presents glycolipid compounds. So because of this monomorphic nature of both the alpha chain and CD1D, iNKT can be taken from any individual in this room and given to another person without requiring any editing and without causing GVHD. And actually what we learned from Dominic Schneiderwind in 2014 and 15 is that iNKTs from donors can actually suppress alloreactive cells, including those that cause GVHD. But at the same time, they play the orchestrator of the immune system. So while suppressing the harmful cells that cause GVHD, they can activate the those cells which are required to protect our patients from pathogens and tumors. And finally, they can also play as effectors, so they can directly kill CD1D expressing cells, which are in the microenvironment, are actually those responsible for immunosuppression also of our CAR-T therapies. And they can, of course, also kill tumors that express CD1D. On top of that, we can engineer them. So now you can see we can create a multi-specific platform where one single cell can use 3 weapons to attack tumors: NK receptors, the T-cell receptor, and the chimeric antigen receptor. So what happens? These are the cells. They are a very rare subset in the blood. But still we can purify and engineer them to create a large-scale CAR immunotherapy. And when we culture them, we target. This is the scenario. If we use CAR-iNKTs, for example, as again targets like these B cells that express both the CAR target and CD1D, CAR-iNKT can use 2 weapons, the CAR and the TCR, to attack the target, whereas the CAR-T only can use a CAR. So what happens is that if we use a very short 3-hour cytotoxicity assay, already we can see that these cells kill faster compared to a monospecific approach. And even more, if we pulse the cells with a lipid, which is a lipid presented by CD1D, in just 3 hours, the majority of our targets has gone compared to CAR-T cells. And this is a short-term effect that we can also see when we use primary cells from our refractory B lymphoma patients. And in the longer term, you see here up to 3 months after infusion of CAR-iNKT cells, there was a prolonged survival for those mice that were receiving CAR-iNKT in a model that was using a target expressing both CD1D and CD19. So a very promising platform, of course, but I've also learned there are some challenges about that. And in addition to the low frequency, of course, heterogeneity is another problem. And this is really affecting several aspects of biology of iNKTs. First of all, the expansion. If we choose the wrong donor to generate CAR-iNKTs, we end up with a very poor product. And also cytotoxicity. There are donors that respond very well in 3 hours, but others that do not. And the same for cytokine profiles, where not only the effector cytokines required for anti-tumor activity, but also the immunomodulatory cytokines, including IL-4, which is key to suppress the alloreactive cells, may be less produced by some donors. So the question I asked myself is whether by using the wrong donor, I may risk to lose my patients, either because they just failed their therapy or because allogenic CAR-iNKT are rejected. So my goal for my postdoc was really to identify a method to select optimal donors as a key really point for success with CAR-iNKTs. And the idea is that we really want to show every time we use allogenic CAR-iNKT, consistent responses and durable persistence. So the first question is whether we can actually predict optimal donors. And if we do so, can we use then unedited iNKT cells without any editing to promote persistence and therefore prevent relapse? You can see the value of using an unedited approach where we can further streamline manufacturing, getting this off-the-shelf cell therapy with a very low cost and create backups and stocks of these cells for any patient. And so the choice of the model in this case is very key. The reason being that mice are inbred, their genetic variability is lower compared to humans. So clearly, if we want to perform a study on heterogeneity, mice may be not ideal in an immune competent setting. And the other point is that they use different alpha and beta chains compared to human, which leads to different functionality. So these mice studies may not be necessarily faithfully recapitulating what we see in humans. Very surprisingly, non-human primates as well are not ideal. The reason being that also in a resting phase, non-human primate iNKTs are suppressive, so we cannot really use for cancer studies. And this is why I asked myself whether I could use DOG, which is a very unusual, but if we want the first historical model where allogenic stem cell transplant was established in chronic myeloid leukemias. And I come from London, where chronic myeloid leukemia was a big fame target for us. So why dogs? Because dogs are companion dogs who live in the same environment. They are exposed like us to the same antigens, dangers, bacteria, virus. So their immune system, surprisingly, is very much similar to what we have. It's an experienced immune system, and they develop spontaneous tumors like us. Surprisingly, osteosarcoma is exactly what we see in pediatric patients. Glioblastoma is very similar. Lymphomas, diffuse large B-cell lymphoma are exactly the same. And they can also receive similar clinical care. Same chemotherapy, same cancer immunotherapies, and now they're also enrolled in CAR-T trials. So for me, it was a fantastic opportunity when I went to Penn, because I saw that as an ideal parallel patient population where I could test my CAR-iNKT cells and ideally advance my best iNKT cells to my human patients. So where to start? The point is that when I started, no one knew whether dogs have iNKTs and whether they are functional in dogs. So I started with a prediction based on a blood study, where the alignment between human and canine predicted iNKT TCR alpha chain suggested that these chains are actually identical in their rearrangement. So I've used human CD1D tetramers, so a human reagent, as a probe to identify canine iNKT cells among canine PBMCs. And what I found was this, a population very much reminiscent to the very rare population we see in human. I look at the immunophenotype, same phenotype compared to mice with a notable CD8 subset, which is key in human to deliver cytotoxic effects. And so I sorted, purified these cells, performed single sequencing, and at that point I had my demonstration. These canine cells are actually the iNKTs we're looking for with exactly the same invariant alpha chain we see in humans. And not only the alpha, but also look at this beta chain. They just use the same gene B25, which is a striking difference compared to mice. They use a variety of beta chains, and this is why their biology is so different. Whereas in this case, I was very much anticipating also similar biology in dog and human iNKTs. And last, of course, I done some single cell studies. So I taken the freshly isolated iNKTs, and you see here the subsets, which are very much overlapping, where the canine and human subsets seem to be basically the same, with very similar frequency and largely clustering away from those we see in the human T and NK cells. So takeaway message from this, we've identified a very much conserved subset in human and dogs. And this is suggesting that we can actually now use canine iNKT cells to perform translational anti-cancer studies that could potentially inform human trials. So my job was to expand canine iNKTs and start to produce canine cellular therapy products. And as you can see here, the expandability is excellent. Within 3 weeks, you can get in certain donors up to 1 million-fold increase. And so then I went back to my question. Can I predict in these products my optimal donor? So I applied single-cell RNA-seq again, this time in my expanded cells. And without going too much into the details, I started to identify signatures, including central memory and exhaustion signatures, which are so key to predict a very powerful cellular immunotherapy. These are violin plots showing an array of 200 genes that are differentially expressed across 4 different cellular products. And as you can see here, the profile in DOG3 was probably more favorable, suggesting a better functionality as well. In vitro assays, and I'm showing again a 3-hour in vitro killing assay, again, DOG3 was the one performing better. So my idea here was that probably by using a single cell approach, I can identify signature that will allow me to predict the best donor. And to validate that, first of all, I've taken this raw data from a trial performed in Baylor. That was actually the only trial present at that time. We're talking about 2020. kids with neuroblastoma treated with GD2 autologous CAR-iNKT cells. So I've taken their single-cell RNA-seq analysis, I've applied the same analysis, and clearly you can say that there is some differential expression as well. Differences are milder, which is expected, probably is an effect of chemotherapy in these autologous cells, but still significant. So based on my approach, I would also anticipate that patient 3 had a cellular product with an anticipated higher anti-tumor effect. And if we look at their clinical trial data, patient 3 was characterized by a better proliferation of CAR-iNKT in the peripheral blood, and that was also associated with a greater tumor reduction, a kid that actually achieved a secondary complete remission. So I was confident at this time that I can actually take my donor 3 K9 iNKTs without any editing. I've taken 400 million cells. These were stocked in the liquid nitrogen. So I've taken them, thawed them, and transferred into 2 30-kilo size dogs. So this is pretty much a pediatric size for these recipients, meaning that my approach could be potentially merely translated into a clinical trial. And these were mismatched dogs. So what I observed is that there was no toxicity, no CRS, and of course no GVHD, and there were signs of sustained activation in these dogs, where there were some lymphoid activated cells within 7 days, where some of them were cytotoxic subpopulations, including NK cells. Other were immunomodulatory cells, including Tregs, that we know again from Dominique's studies, they're so key to mediate allo-suppressive effect by iNKT cells. So they are required. And therefore, the next question is whether, in fact, my donor iNKT cells were still there. And what I've done, I've leveraged my sex mismatch in this model, where the donor cells were female, so I could monitor EXIST, which is a non-coding RNA only expressed in female, but not in male recipients. And so I've done it by PCR, digital PCR, and nearly 4 months later, I could still identify EXIST in my recipients. And the same also by MHC profiling, I could observe that the DRB1 as a polymorphism, that was a polymorphism only present in my donor, was still there also in my recipients. So take-home message here is that there is an opportunity, if we use donor iNKT therapies, to also be very selective and pick only the optimal donors to ensure maximum therapeutic effect. And actually, there is opportunity also for using them as an unedited cellular platform. So next, and I will go very quickly here, we are obviously working on an off-the-shelf CAR-iNKT approach, where we are starting again with a comparative approach. And I'm using here a cross-reactive human canine CAR construct. Why so? Because obviously if I can use the same construct in my dogs, then immediately I can use them in my patients, and that's possible also because of the identity and conservation of the targets in human and dogs. So I can use the same CAR construct. And what I'm doing here, for example, I'm using an anti-IL-13 receptor alpha-2 or anti-B7-H3 approach. The transduction efficiency of iNKT, regardless of the species, is very high. And now this is allowing me to consider a broad target, tumor targets for my studies. I'm starting now with glioblastoma and osteosarcomas. And without going into the details of the dissection, mechanistic dissection of these therapies, I'm just going to show you an approach that allows me to see how these cells are active also against the microenvironment in vitro. So to do so, I'm using organoids from patients. These patients are glioblastoma patients undergoing surgery. We get the tumors, we develop these organoids in vitro that include both the cancer cells, but also the microenvironment expressed in CD1D. And this is just an example of what we see when we use just conventional T cell. Usually T cells, and this is well published by my collaborator, remain at the periphery of the organoid. There is clearly some death associated to the presence of T cells, but the core remains basically intact because of anoxia and because of the microenvironment. And this is just to show you at a higher magnification, vibrant activity really at the periphery, very little happening in the core. So what happens when we use iNKTs? The anticipation will be that at the very least, we see something happening at the periphery with some death associated where iNKT goes. But actually, I can see a pronounced migration of iNKT, which is known. Again, they are tissue-resident T cells. They can migrate and overcome anoxia. They can overcome tumor microenvironment. And this is also associated with cell death, which is including both, in pink here, microenvironment cells, but also primary glioblastoma cells. So in parallel, we are also conducting in vivo studies. And here I'm showing you a comparative approach that will allow me in vivo to establish an equivalence between my canine iNKTs, CAR-iNKTs, and human CAR-iNKTs. So in that case, again, I can show that whatever I see with canine CAR-iNKTs later on in dogs can be very much anticipated to be predictive of outcomes in humans. So the outline is very easy. I use either human or canine tumor cells, which are expressing, in this case, B7-H3, and they're also engineered with luciferase so that I can monitor tumor burden by bioluminescence in vivo. In this case, I'm showing you osteosarcoma. So I engraft NSG mice, immunodeficient, with my tumor cells, with the goal of identifying also intrinsic properties of my CAR-iNKT cells that can lead to anti-tumor effect in vivo. And I treat with mice with either iNKTs engineered with an irrelevant CAR, in this case an anti-CD19 CAR, or CAR B7-H3 iNKTs, and then monitor them over time up to 3 or 4 months. And what I'm seeing here, I'm showing here the results from the canine part of my studies. Compared to background, clearly when these mice do not receive treatment, these tumors are very aggressive, rapidly progress with lung metastasis. If we treat with iNKTs and an irrelevant CAR, there is some control of disease, but certainly the disease is very much controlled in the presence of CAR B7-H3 iNKTs that push back the tumor to a background level. And in a longer term monitoring, you see here great tumor progression. We're actually losing mice already after 3 weeks, and still a deep remission in CAR B7-H3 iNKT treated recipient with a trend to rebound in tumor growth in the ones that receive just iNKTs. So this is clearly supporting now our next trial. We're actually already screening canine patients. I'm very excited about that. I'm working in a hospital, which is very much reminding me of my pediatric patients. And we are actually screening these patients. We are also I already identified an ideal donor for me, and the first patient will be treated in March 18. And this is obviously preliminary on my next human trial using allogenic CAR-iNKT for the treatment of pediatric patients with B7-H3 positive sarcomas. This is a protocol I've already drafted, discussed with experts also within SITC, and we hope to file the IND very soon next year. And with this, I would like to thank all my collaborators at PenVet, Penn, the Children's Hospital, and NIH, where we are conducting also spatial studies to better characterize our tumors, all my funding agency, and I will be happy to take any question.
Many thanks. Exciting data. Are there questions? I can start in the meantime. There's one. So for your clinical trials, will you administer the cells systemically or locally?
Yes, IV. And this is based on the fact that it can migrate really into tissue very easily. It will be a single infusion without prior lymphodepleting regimen. Yes.
Thank you. Then the question there.
Thank you. Very nice data. I have a question regarding the specificity of this NKT cells. So you are injecting huge amount of NKTs. Is there any issue that they will get activated by CD1D endogenous?
That's a great question. So the answer is no. And I've shown you here 400 million cells because I want it to be in the middle based on what we have from clinical data using both CAR-iNKTs and iNKT without any other engineering. So the first trial was published in melanoma, autologous iNKTs without any CAR engineering. In that trial, they used 1 billion iNKT without any toxicity. So even if they can recognize CD1D, this is not leading to any autoimmunity or systemic toxicity. In the CAR-iNKT trial I've shown you from neuroblastoma patients, they started with 3 million CAR-iNKTs and they got the complete remission with a low dose. So whether we use low or high, it doesn't really seem to be that relevant in terms of toxicity, and efficacy is as well good. I guess also it's a matter of the donor. As I said, if I use probably a poor donor with just 3 million cells, I wouldn't see that much. And in that case, I want it to be in the middle. So this is why I use 400 million.
Thank you. Thank you. Other questions? So you use a normal lentiviral vector for transduction?
I can use both. Currently, I'm using a retroviral vector because K9 like more a retroviral vector, but lenti works as well. Yes.
Great. If there are no further questions, then thank you very much again, and we move on to the next speaker. So it's a great pleasure to introduce the next speaker, Andreas Plückthun. He's a professor of biochemistry here at the University of Zurich, has done really groundbreaking work in many areas, particularly protein engineering, and will talk today about precision gene delivery for tumor therapy. Many thanks for looking forward.
Is it on now? Okay, very good. Yeah, first of all, thanks to Markus. It's a great pleasure to be here. We are a protein engineering lab, as you have just heard. And I want to talk today about a platform we've been developing over the last few years that has the aim of increasing the therapeutic window. So we are developing systems for precision gene delivery to target particular cell types in the body, especially to deliver those proteins which be systemically toxic, and also where we would be able to produce multiple factors simultaneously to really optimize efficiency. And we're using engineered proteins both for achieving this and that the targets, that the payloads can be engineered proteins. So this is a tumor, which of course consists of many different cell types. And as we'll see as we go along the talk, we will target more or less all of these cell types individually. Let's see whether I can make the pointer bigger. Where is this in Windows? Here we go. No. Here we go. There we go. Okay, very good. So we'll target tumor cells. We'll target immune cells. I won't talk about the targeting of blood vessels, which we're working on, which I think is making very exciting progress, as well as cancer-associated fibroblasts. So what we're actually doing is to install a factory. We use non-replicative virus-like particles that really create a biofactory in the various cell types that we can design, and to secrete proteins that only have to then diffuse locally. So we can call this the factory behind the enemy lines. And this can be, why is it not moving? This can be therapeutic antibodies, fusion proteins, that only need to diffuse locally. And most interestingly is probably cytokines, and especially those that would be systemically toxic, because that way we can actually design the tumor microenvironment. We don't have to suffer with what we have. That's the idea. So what do we use as a basis? We're using virus-like particles that are based on adenovirus. So why would we use adenovirus as a basis? And why would we use a non-replicative virus-like particles when most people that work in a similar field use adeno-associated virus? The answer is very clear. AAV is too small. We don't have enough coding capacity. So we can engineer a lot of things, but we cannot make AAV much bigger. And so therefore we decided to use adenovirus as a basis, which you can already see from the picture, has a coding capacity of 35 to 36 kilobases. That's a lot. You can code a lot of genes in there. So we have to solve three problems now. We have to make the virus non-replicative and free of any viral genes. We have to make the virus target the cells of interest, and we have to shield the virus from interaction with blood components and non-specific interactions with cells, especially detargeting it from the liver. And I'll lead you through all of these 3 points. So the first thing is, how do we make the virus non-replicative and free of any viral genes? So this is what we want. We want to have these 36 kilobases with whatever genes we encode there and have no viral genes at all. And so these can encode, for example, the genes for antibodies, cytokines that would be secreted to give this paracrine delivery. We have also used prime editors and gene insertions for gene editing and writing. I won't have time to talk about this today, but we're working very closely with my colleague Gerald Schwank on that. We can, of course, encode regulatable promoters, regulatory circuits together with the above. And we have developed a minimal replication system that actually allows the genome to be replicated without making new particles, just increasing the gene dosage in the infected cell. So that has been actually very useful. And of course, in principle, we can encode any cytoplasmic protein or membrane protein there. So this is important because we have so much space that we don't have to use any suboptimal small constructs or minimized proteins when we use prime editors or gene editors. But as I said, I don't have time to talk about this today. So in order to do this, we need a second genome, and that is the wild-type genome, which is a very complicated machinery with 46 genes. And this machinery is needed to really package this. And so the key thing is that if we put both of these genomes in the same cell that has a CRE recombinase, this encapsulation signal is flanked by LOXP site and gets spliced out. This has a wild type encapsulation signal, a little piece of DNA. So what actually happens is this makes all the proteins in the right amounts at the right time to actually drive the machinery, but it is this genome which is packaged. And so I don't have time to show you this, but we have optimized this to the sub-ppm level of contamination. So the next issue is, how do we target the cells of interest and how do we do this selectively? So this is what the particles look like. They're icosahedrons, and they have a fiber and fiber knot at each of the 5-fold axes. So we have used this and have used our DARPin technology, a scaffold we've developed more than 20 years ago, have, I think, by now selected over 600 against 600 different targets. And we can make extremely specific proteins that you can see here on the left-hand side that have an extremely complementary surface to whatever target we want. And so we have used the knob of the at the fiber as a target, created DARPin, solved the structure, determined that the C termini are close to each other, and actually designed a trimeric protein to make this a clamp, to really bind very, very tightly. We can then add a linker to this, just glycine serine linker, put another DARPin here that now gives the cell specificity. So each of these knobs has now 3 binding sites to a cell that we like. The cool thing about this is we make these in E. coli that trimerize by themselves. We just add it to the virus particles and purify it away. And so we measured just quite a number of years ago already that they stay on the particles for 10 days. And so thereby we can use the same payload and change the specificity very rapidly, determine different receptors for uptake, determine different epitopes. And this has been immensely useful because it's very fast. The third thing we had to solve is to shield the virus from interaction with blood components and non-specific interactions with cells. And so for this purpose, we actually used antibody technology. This is actually where my lab comes from, and used a single-chain antibody against the hexon protein, trimerized this as well, and actually humanized it at the same time, and more or less covered the whole particle, as you can see here. This is a cryo-electron microscopy done by my colleague Ohad Medalia. And so that actually shields this thing from uptake by receptors, especially in the liver. This has been immensely important to detarget it from the liver. All right, to actually show you what it does to the immune system, I can show you this experiment. This is now the virus with just the adapter that I explained. And that's the virus-like particles with its shield and the adapter. What we're doing now is to use neutralizing antibodies. First, if we have no neutralizing antibodies, the shield really does not significantly inhibit uptake through the receptor. If we titrate in neutralizing antibodies, that's the black bars, you can see this goes down dramatically, that's a log scale, whereas if we have the shield on, this is much, much less. So now let's just take a very simple experiment first, which is to now see, does it matter if we co-express several things? And so in this particular experiment, we used the murine antibody against PD-1, the murine IL-12, and the murine IL-2, and also put all three things together in the same genome. And what one can immediately appreciate is that if they all have their own effects, but having all together really leads to a tumor reduction in this B16 model that overexpresses HER2 that we use as a targeting. Now you could argue, why don't you just mix three viruses at the same gene dosage? You can do this, which is the yellow line here. But if we put everything in the same particle, it's still better. And our interpretation of this is that every infection event now guarantees that all three proteins are expressed. All right. We have also a very nice collaboration with Reddy and Heinz Leubli, who spoke earlier today, on doing something similar with the overexpression of IL-12 and with or without CCL5 in the tumor. And the cool thing is, this is an explanation of how the anti-PD-1 resistance could be overcome. This is the work of Nicole Kirchhammer. I don't have time to really highlight this very interesting paper of hers, but I want to show just one, I think, a cool snippet from this. So these are of course mouse experiments, but what was also done in the paper is to take primary tumors from patients, and some of them actually are active and produce interferon gamma, but some are not. If we treat these with the virus-like particles that express IL-12, they're all good. They all become active. So one can overcome this resistance from patients. I think this is a very encouraging way of perhaps addressing this. So let me just go a little bit more mechanistically, and we have a very nice collaboration with Viviana Gardinaro's lab at Caltech, and have done there what is called the clarity process. What this is, to take tumors out of the mouse and treat them first with formaldehyde and acrylamide and make a massive cross-linking so that all the proteins are cross-linked to a mesh. Then we use detergents at very high concentrations to remove all the lipids. And then we add a solution to match the refractive index. And so this makes the tumor completely transparent. And what I'll show you now is microscopy pictures, light microscopy done in 3 dimensions using these transparent tumors. So what we'll see now is the vasculature in red that has been stained by lectin. We'll see the transduced cells because they make GFP. And we used, in this case, trastuzumab as a model antibody to see where it is produced and how this works. So here we go. So what you can see immediately is that the virus-like particles have left the vasculature and have infected the cells. The antibodies are still in the cell and being produced and have left the system. And we can really see that this is actually produced within the tumor. The tumor is essentially destroyed from within. This is not a video generation. This is true microscopy done in 3 dimensions, just rendered that you can see the cell surfaces. So I think this is pretty encouraging. So this is just a very, very short summary. At time 0, you have an intact vasculature. At 11 days, one can see actually holes in the vasculature and infected cells that have taken up the virus-like particles, produce in this case GFP, an antibody. And this is after 2 months. So the system keeps going for 2 months at least, where now the tumor is full of holes, but full of antibodies that has been produced from the inside. So then we went on, and there's actually another very nice collaboration with Heinz Leuble's group. We then made a more aggressive molecule, in this case, a bispecific T-cell engager. But to engage the tumor cell, we actually used the DARPin. The reason why we did that is because we have no purification, right? The stuff is produced from the cell and has to be used as is. So in other words, there must be no misfolding. And so therefore, this is why we use DARPins, because they're extremely robust and there's no domain mispairing as there is in single chain tandem constructs. So here we go. If we take these proteins as proteins, there's a small reduction in growth. This is the typical concentration that has been used in the literature. But if we produce this in situ by the system that I just showed you, there's a significant reduction. We didn't get all of them, but about half of the mice have a complete remission. So this is actually not so bad for the first shot. Okay, so this was targeting tumor cells. We have also targeted stromal cells. We have targeted the vasculature. This is work that is still ongoing and look actually quite good. But I want to show you then also immune cells. Regarding the stromal cells, we have used them also as a production site to produce the therapeutics from the stromal cells and let them diffuse to the tumor cells such that the tumor cells sort of destroy themselves and destroy the factory. This actually has been a very useful approach. I think it was pointed out before that human carcinomas are full of fibroblasts, murine are not. This is important to remember. So if you want to read more about this, this has been published 2 years ago. Let's come to immune cells now. And this is an idea that, of course, has come in to many people. We have heard some very interesting work before. Would it be possible to make CAR cells in vivo? In other words, replace the personalized fermenter by a syringe. So in order to tackle this, we used our technology and have to somehow specifically attack T cells. And for this purpose, we actually use this time a mixture of 3 adapters: anti-CD3, anti-CD28, and the IL-2 receptor, using IL-2 as the protein itself. And what actually turns out, each of these themselves has some effect of uptake. However, by far the best is to mix all 3. We then changed the ratio, and it's actually interesting. The best ratio is 1 to 1 to 1, because one can calculate this maximizes the likelihood that each particle get at least 1 receptor of each type. Okay, and I skip all this in vitro stuff and directly go to in vivo experiments that we get did together with my colleague Christian Manz. And the first experiment is a sort of more simplistic model, which is an intraperitoneal approach, where we first come with human T cells and then these particles that are loaded with these three adapters. And the good thing in this system is that the different T cell subtypes are transduced, CD4 and CD8 to about the same level. Whether in this system you can also pre-activate the T cells. Actually, we found out that's actually not needed because this combination of CD328, as was very nicely explained earlier today, and IL-3 is actually doing this already. And we see a very similar subtype activation. This is preferentially hitting T cells. Since we have the IL-2 receptor, we also hit B cells, but no NKs and no monocytes. But of course, one could leave out the IL-2 if this was an effect. I think the more interesting question, the model, is this model, also from Christian's lab, the CD4 humanized mice that now express the human immune system, if you will, where we only have to now come with the T cells. And the very encouraging result is that we do hit the T cells in vivo in this system, and we can count the viral genomes, we can use IRFP, and we see a selectivity from T to B cells. Of course, the levels have to be improved, but nonetheless, I think it's a foot in the door and a very encouraging way of approaching this. The last thing I want to share with you is to hit dendritic cells. And the way we did this is to use, again, a specialized adapter, which hits DC sign on the dendritic cell, or CD209, and we use the peptide that binds to the integrin CD11C. And we used this time a different molecular design where we fused the peptide to the adapter that binds to CD209. And so thereby you also get an avidity effect in a very local manner. So the very good thing is this was done to target it to draining lymph nodes. We get essentially nothing in the liver, nothing in the spleen, and a very, very nice enrichment in lymph nodes. Hopefully this paper is going to come out very, very shortly. And so then we thought, okay, let's do an experiment to see whether something like this could be used as a tumor vaccine. We used a B16 over model where we engineered the vaccine as a fusion to the invariant chain. And I think this is the key thing, because we have so much space on the system, we co-expressed either IL-2V or IL-21. And we could use an extremely low number of viral particles, so virus-like particles, 10 to the 7. And the very encouraging results are summarized here. And I don't want to go through every bar, but just put the highlights out. So this is the healthy control, which you can see in lung weight is very low, obviously. It has no metastatic foci and has a very low melanin content. So this is a melanin model looking for foci in the lung. Okay, and if we go now to the maximum system, which is the co-expression of IL-2V or IL-21 in this system, we can see that we basically return to baseline. That we have the lung weight practically identical to the control. We have an extremely small number of metastatic foci, and we have an extremely low level of melanin content, which you can see here. So I think this is also very encouraging and sort of shows you that these things can go in many different directions. So to summarize, I think we are somewhat progressing with a precision gene delivery with engineered binding proteins. It has shown great promise for therapeutic protein production in situ, which I tried to show you. Cell engineering in vivo, CAR-T cells being an example, but also tumor immunization with the cytokine co-expression and gene editing using very large enzymes, which I didn't have time to show today. So I have the privilege to work with a phenomenal group and great collaborators. Some are here. And so I think I'll stop at this point, and I'm very happy to take questions.
Many thanks for the great talk and fascinating science. Are there questions? Yeah, there.
Thank you very much for the presentation. I was wondering in the part with the DARPins with the Bispecific anti-CD3, if you have some data about how much is produced and if there is some risk that you produce so much that some Bispecific will bind the T cells, some other will bind the tumors, and so you won't get them in contact. I don't know if you saw something like that.
I think it's a fair question. Would we sort of get the hook effect? Would we produce too much? This is something that probably, when this is really translated, will have to be titrated, and of course, under these conditions, there will be probably inducible and tissue-specific promoters. I'm not really sure that we are at such a level of being able to really inhibit the system of having so much. I mean, this is a bit hard to calculate. I mean, we have a production from these systems that is sort of, let's say, not so dissimilar from a CHO cell line, I would say. And whether that is enough or too much in a particular setting will probably have to be titrated. But I think in the real translated system, this will definitely be a regulatable system where this will be where this will be checked.
Thank you. Thanks. Other questions? Christian?
Thank you for the talk. It's very interesting. I had a quick question regarding the cytokine immunization. So when the tumor cells express the cytokines, and whether you saw in a CRS, like so a cytokine release where you could see also some toxicity, as of course cytokines in a recombinant or soluble form could cause
no, we didn't see them, but I think that's the decisive point, because we're making them locally. We're making them in the lymph node, and as I showed, they were really not found, and the particles were not found anywhere else. And so I think it's sort of basically, in the absolutely ideal world, the DCs should do this themselves anyway, but they don't. And so we produce the gene, but this is super local and doesn't really diffuse out very far. And I think that's the key. That's the key of this whole technology.
There was another question online in the chat. Somebody asked, in your opinion, what's the advantage for in vivo engineering of T cells to use DARPins instead of antibody domains? So you basically also can decorate the viral-like particles with antibody domains instead of DARPins.
Yeah, so this is really how to make the adapter in the first place. Yes, I think that is a very fair question, and there is an answer. Because what you're doing here is with this trimerized, maybe we can go, no, can I go back? No. I think it would be easier to show with a picture if we can go to that. Okay. There. Yes. So what you see here is this trimerizing unit, and that brings the three green DARPins very close together. Now they are extremely resistant against aggregation, so this stuff folds. Period. Whereas with antibodies, if you have three next to each other, especially in the single chain format, you would have a very high chance of getting aggregation, getting misfolding, and so on and so forth. So I think in most cases, they don't tolerate having, especially in the single chain format, having such a high local concentration. And so here, it's also the shape of the thing is just perfect for that. And I'm not saying it cannot be done, but I think it would be much harder.
Thank you very much. If there are no further questions, we move on, and thank you very much again. So great pleasure to introduce the next speaker, Christian Klein. He actually led for many, many years the cancer immunotherapy section at Roche and is now pioneering innovations at Curie Bio and will give us an overview about bispecific antibodies for cancer therapy. Looking forward to your talk.
Yeah, thank you very much, Markus, and to the organizers for the invitation. And I will give you today a more general overview about really the present and future of bispecific antibodies for cancer therapies, and a topic I think that has already been touched in many presentations here. So if you think about the development of antibody therapies, I think you see it here. In the past 20, 25 years, you saw that initially everything was about monoclonal antibodies. Then you saw the emergence of Fc-engineered antibodies that have enhanced ADCC function. And more recently, antibody-drug conjugates arrived in the clinic, as well as bispecific antibodies. And I think that's also a trend that is will further be strengthened. We are also seeing increasingly even multi-specific antibodies. And I think we will also see, in addition to cell therapies, approaches like Andreas just presented them. So if you look into bispecific antibodies into the clinic, you can see in hematological malignancies, the majority of those antibodies is really T-cell engagers and other engager classes, whereas in the solid tumor area, you have a lot of checkpoint inhibitory bispecifics, also T-cell engagers and other engagers. But as we already heard or have seen, it's more difficult to develop these agents for the therapy of solid tumors. And that's also reflected. The other aspect is that you increasingly see also bispecific antibodies in advanced clinical stages, so meaning either approved or in phase 2, phase 3 clinical trials. And I think that's also a difference from the past decade, where the majority of these antibodies was in preclinical stage or maybe phase 1 clinical trials. So that's the current list of approved bispecific antibodies. And you see in total, we have almost 20 bispecific antibodies approved. The majority of these in bispecifics for cancer therapies is T-cell engagers. As you can see it here, there's also a couple or increasing amount of receptor tyrosine kinase bispecifics coming up that partially also have enhanced ADCC function. And we also see a couple of dual checkpoint inhibitory bispecifics that are getting approved. So just very briefly, receptor tyrosine kinases, I think it's really a topic that's coming back. I think was very popular, let's say 15 years ago, but then after the wave of cancer immunotherapy, there has been less interest. But yeah, nowadays there's a couple of those dual receptor tyrosine kinase bispecific antibodies available for treatment of patients already. And as I mentioned, these antibodies also incorporate Fc engineering, meaning they are more potent in recruiting NK cells and macrophages for tumor cell killing. So the first example, targeting EGFR c-Met. There's also biparatopic antibodies targeting HER2, like zanidatamab, which is targeting 2 different non-competing epitopes on HER2 and has been approved in HER2 positive cancers. Most recently, we also saw an additional antibodies, inotuzumab targeting HER2 and HER3, again, with enhanced ADCC function to better engage the innate immune system. Another topic that has become very popular based on clinical data is checkpoint inhibitory bispecifics. You have here the PD-1 VEGF bispecific.
ivonescimab. There has been quite a buzz about this antibody because it's the first antibody that was superior to pembrolizumab in a head-to-head study in China so far, where it is approved. But currently, this antibody is tested in advanced clinical studies in the US and in Europe. And there's also additional bispecifics that block checkpoint inhibitors as well as angiogenesis simultaneously in clinical trials. And I think we will still have to see whether this really results ultimately in improved overall survival also in those patients, because so far we have seen that it prolongs the progression-free survival of patients. We are also seeing that in general, concepts that rely on dual targeting on the same cell become more popular, and that includes also what I call here as the so-called cis targeting. So that can be the co-inhibition of two checkpoint inhibitors on the same T cell. There's also approaches that can make use of increased internalization, for example, to get rid of checkpoint inhibitors on the cell surface. So this is a topic of protein degradation or antibody degraders. And you can also make use or modify post-translational modification, for example, by bringing a phosphatase in the vicinity of a checkpoint inhibitor, and by that mean blocking the inhibitory function of PD-1, for example. We ourselves, we were using this concept of cis targeting for the delivery of IL-2 to PD-1 positive T cells. As you know, the PD-1 positive T cells, that's the antigen-specific T cells. And when you deliver IL-2 to this to the PD-1 positive T cells, you can see that you have much higher potency, approximately 40 to 50-fold higher potency as compared to non-PD-1 expressing T cells, and that can really make a big difference. I think that's also something I really recommend if you want to target the IL-2 receptor on T cells, make use of the cis targeting, because otherwise you are not making or not targeting the right subset of antigen-specific and stem-like T cells. An alternative approach, very similar, relies here on a CD8 targeting antibody and an attenuated version of IL-2. And the authors here have shown that this molecule per se on non-CD8 expressing T cells has a very low potency of or IL-2 receptor agonism, whereas once it is targeted to CD8, it has a strongly enhanced activity on the CD8 positive T cells. And actually, this concept has now also shown first clinical proof of concept in the clinic by a Chinese company, Innovent, that has shown using such an PD-1 cis-targeted IL-2 variant activity both in CPI-resistant lung cancer patients and also in colorectal cancer patients, an indication that's typically known not to respond to checkpoint inhibition. And Innovent just has reported that they now initiated a pivotal study with this approach. So I think ultimately we will see that IL-2 or IL-2-based immunocytokines really will arrive in patients in the near future, because there has, as you know, there has been during the past 10 to 20 years a lot of clinical disappointments with IL-2-based therapies, but I think we are ultimately there. Another approach to make use of bispecificity to get higher tumor selectivity is in the context of antibody-drug conjugates. So really with the aim also to target these ADCs more specifically to cancer cells and basically reduce toxicities in normal tissues that also express these antigens. So that's also a big trend in the field making bispecific ADCs and increasingly also ADCs that make use of dual payloads so that you can also overcome resistance mechanisms to the payload of these ADCs. And yeah, as I already alluded to, you can get rid of the surface expression of receptors by targeting either an E3 ubiquitin ligase that is expressed on the cell surface, and that will then subsequently induce the degradation of a cell surface receptor. Can also be on T cells like PD-1, or you can use strongly internalizing receptors like the transferrin receptor to degrade also cell surface proteins, and this has been successfully applied to checkpoint inhibitory proteins like PD-1 on T cells or PD-L1 on tumor cells. And another approach that has also become very popular in the field more recently is the generation of surrogate cytokines that are antibody-based, typically single domain antibody based, and you can, as you know, many of these receptors are heterodimeric in nature, and as soon as you bring IL-2 receptor, beta and gamma receptors together, you can trigger the receptor signaling, and you can actually achieve this by making two single domain antibodies to these extracellular domains of the cytokine receptors, and by bringing them together, trigger the intracellular signaling. And of course, the amazing application of this is by doing so, you can now really also generate de novo cytokines that naturally do not exist and bring cytokine receptors together that you cannot achieve with a conventional cytokine. So with that, I want to switch gears to T-cell engaging bispecific antibodies, because that's something we have, or I have worked during the past 15 years. So and they represent really the biggest class of approved bispecific antibodies. As you may know, they bind with one arm, or as we heard already, to the T-cell receptor, and with the other arm to a cell surface antigen on the tumor cells. They can then, or that results then in the killing of the tumor cells, in the secretion of cytokines and T-cell proliferation. It also results in the upregulation of certain factors on T cells that then counteract this mechanism, and that is why co-stimulation and checkpoint inhibition in combination with T-cell engagers is very important in order to make really use or the maximal use of their efficacy. And we have also heard that, of course, the secretion of chemokines and cytokines results in infiltration of additional cells into that tumor that can be naive T cells, but it can also immune suppressive cells such as macrophages and neutrophils. So that's really important to consider when you think about combination therapies for T cell engaging bispecific antibodies. And when you look now here at the timeline, the first T cell engager was approved 2009. But subsequently withdrawn from the market. And then the first real T-cell engager, I would say, was blinatumomab, approved 2014. But now in the past 10 years, we have really seen many of these bispecifics approved, both for B and plasma cell malignancies on the one hand, but we also seeing now the first T-cell engagers arriving for the therapy of solid tumors. We have ourselves developed glofitamab or Columvi. That's really one bispecific antibody developed here in Zurich, and that is approved for third-line relapsed refractory DLBCL, and was recently also filed in combination with chemotherapy based on successful phase 3 clinical trial for second-line DLBCL. So when you look at the targets addressed by T-cell engagers, you will see that actually they have already many targets been tested in clinical trials. All of these targets have been tested in clinical trials. The ones in bold are the approved ones. So yeah, of those, so far only a few targets seem to be amenable: CD19, CD20, BCMA, GPRC5D for B and plasma cell malignancies, and DLL3 for solid tumors, as well as GP100 as a peptide MHC target. So tarlatamab is really the first T-cell engaging bispecific antibody that has shown consistent efficacy and safety in relapsed refractory small cell lung cancer patients, and has been approved based on those data. So that really shows you, so the response rate is, let's say, in the range of 40%. And that really shows you that, yeah, well, in fact, T-cell engagers can also be efficacious in solid tumors. They are not restricted to hematological tumors, but you need to have a selective antigens. And another example that where phase 1 clinical data were published recently is a STEAP1 T-cell engager from Xencor, Amgen, that showed objective responses in relapsed prostate cancer patients. I was also talking about TCR-based or T-cell engagers targeting peptide MHC as a target, and the first of those is tebentafusp, that targets a peptide derived from GP100 for uveal melanoma, and has been approved based on those data. And there have also been recently additional data from both Immatics and Immunocore for additional targets, including MAGE-A4 and PRAME, where also objective responses have been observed for these constructs that target peptide MHC complexes, so intracellular antigens. And we have heard today already about now the importance of co-stimulation. This is actually, of course, the consequence that the immune system, particular T-cell responses, are tightly regulated. And as a consequence of this, on the one hand, you find inhibitory receptors such as PD-1 upregulated on those cells, but you have also activating receptors. And actually, the immune system or T-cells require signals so that the immune response can continue. And we have used this for the development of so-called co-stimulatory bispecific antibodies. And there's different possibilities how you can do this. You can do this in trans by providing the co-stimulation as a consequence of binding to a target that is expressed on a cell in the vicinity, like FAP. Then we also heard about that already today. And then you can provide the co-stimulation either for an MHC peptide recognition or also in conjunction with a bispecific antibody, as you can see it here, both in the endogenous system and in the more synthetic immunity system. And as you have heard, the first boosting endogenous immune responses with co-stimulation has not been very successful in the clinic so far, and that's most likely consequence of a relatively inconsistent signal 1 in those patients treated, particularly CPI-resistant patients. But when you do it in combination with a T-cell engager that provides a very strong and consistent signal 1, there's very strong evidence that this also can work in the clinic. And one example here is CD19 4-1BB fusion protein that we have brought into clinical trials in combination with glofitamab. So you get here basically a signal 1 from glofitamab, and then by binding to CD19 on the same lymphoma cells or B cells in CIS, you provide the 4-1BB signal to those cells. And Roche has presented very promising data for this combination. It's currently tested in a randomized phase 2 clinical trial. And we hope that based on those data, it will then also advance to pivotal studies to enable or to support approval. Another co-stimulatory molecule that we have brought into the clinic was making use of CD28 as a co-stimulatory receptor in conjunction with CD19. And we have tested it here in humanized mice CD34 cord blood humanized. What you nicely see is that this antibody, as opposed to the T-Genero molecule that shows very strong cytokine release in humanized mice because it still binds to Fc gamma receptors, does not induce cytokine release because it does not have Fc function any longer, and by being monovalent is also not super agonist any longer. And when we then subsequently tested this in preclinical studies, we were asking ourselves what's really the optimal combination for a T-cell engaging bispecific. And you see glofitamab in this model has an initial response, but then tumors relapse. And when you combine it with CD19-41BBL, you can prolong the duration of the responses. Also, when you combine the CD19, CD28, but ultimately these tumors, they still relapse. But when you now treat first in the combination with CD28, which is kind of a very strong and early co-stimulator, and then followed by treatment with 41BBL, which is more important for the durability, then you can really get very long-term responses and induce the remission of all these tumors. So that ultimately, I think the ideal combination would really be the combination of CD28 and 41BB co-stimulation. Another approach to overcome resistance mechanisms is target two different antigens on the target cell, and there's a company from Lausanne. They made here tri-specific antibody that basically is targeting CD38 and BCMA. CD38 is relatively broadly expressed in the immune system, so therefore you have to make it more selective. And that by generating this bispecific antibody, they were really able to basically co-target CD38 and BCMA on multiple myeloma cell surface. And as you can see here, this approach is then superior in efficacy as compared to the combination of the BCMA T-cell engager teclistamab and the CD38 antibody daratumumab, and they have also presented recently promising clinical data on this. But the challenge with T-cell engagement remains really the availability of clean antigens, and for that purpose, different approaches have been chosen that I will show you now. So just one more thing regarding the co-stimulation. So you can also try to integrate this in one molecule. So this is a Claudin-6 targeted T-cell engager that has here a second arm that binds either to CD3 or to CD137/41BB. And by doing so, you can have a molecule that is really tri-functional. So it can induce T-cell receptor activation and at the same time also provide co-stimulation to the T-cells. Because, as I now alluded to already, there's not many tumor-specific antigens available for solid tumor therapy. One approach to overcome this is basically to make a molecule that is inactive in the periphery, and then by masking, so the CD3 arm is masked by a peptide in this case, and in the tumor microenvironment where a lot of proteases are found, this peptide is this linker is cleaved, the mask falls off, and the bispecific gets active in the tumor microenvironment. And indeed, Janux for this PSMA pro body has shown a clinical activity with a very favorable safety profile in prostate cancer patients. Very recently, another company that is pursuing a similar approach with a steric masking here. So you basically attach here polymer that restricts the access to the binding regions of the antibody in the periphery. And in the tumor microenvironment, by protease cleavage, this steric mask is cleaved off. They have also shown that you can target now antigens that with a conventional T-cell engager cannot be targeted, like HER2, because you will get too much on target of tumor toxicity in normal tissues. And another approach is to make use of the acidic pH in the tumor microenvironment. Here, a company, BioAtla, they have made antibodies that are only active in the acidic or at acidic pH, but not at physiological pH. And by doing so, you can make a T-cell engager that then is very specific for EpCAM, in this case, in the tumor microenvironment. And this molecule is currently also in clinical trials. And last but not least, you can also make use of assembly concepts. So you can cut a T-cell engager into half so that the CD3 arm is not active any longer. And only when these two bispecifics then come together in the tumor microenvironment, they can reform the CD3 arm and engage T cells locally in the tumor. We have published a very similar approach to this, which we called pro-drug activating chain exchange, which is very similar to this approach, but basically incorporates a domain that shields the antibodies so that you have better biophysical properties. So yeah, to wrap it up, I think, of course, we will in the future still see monoclonal antibodies in the clinical practice, both in cancer therapy and outside in non-cancer indications. We will be seeing more antibody drug conjugates, but the bispecific antibodies, they definitely represent the strongest growing class of therapeutic antibodies in preclinical and clinical development. And we are also seeing that these bispecifics, they are going beyond oncology. So we are seeing them increasingly in autoimmune diseases, in metabolic diseases, also in ophthalmology and in CNS diseases. Particularly now for bispecifics, yeah, we will be seeing more dual targeted bispecifics that may, to be more tumor selective. I mentioned dual CPI inhibitors and the CPI angiogenesis inhibitors. I think that have promising clinical data. And regarding the T-cell engagers, definitely also more and more combination therapies on the one hand with a standard of care. So that includes checkpoint inhibitors, chemotherapies, also combining T-cell engagers with antibody drug conjugates. But then also NME combination, that means also non-approved drugs like co-stimulatory bispecific antibodies, which I personally believe is probably the most promising approach for combination therapy with T-cell engagers. And then, yeah, really these novel approaches, bringing those into the clinic, tumor activation mechanisms, making use of dual targeting or antibody degraders and products, as well as cytokine mimetics and yeah, multi-specific antibodies. I think that's where the future will be in the field. And with that, yeah.
Thank you very much for the excellent overview. Are there questions? Jason.
Thanks a lot for the nice talk. So for sure, there are several bispecific T-cell engagers approved for hematological malignancies. And you have shown nicely today the data from Amgen for tarlatamab, which was recently approved for small cell lung cancer. But still, it joins a group of very small bispecific that are used for solid tumors. Do you think there would be a brighter future of this type of BiTEs used in solid malignancies with co-administration of, for example, cytokines or something to boost the activity and the strong immunosuppressive environment? Thank you.
Yeah, definitely. I think there's still a few target antigens available where T-cell engager can be efficacious. Well, I think I showed the STEAP1, also Claudin 18.2. There's some kind of neoantigens like EGFR-V3 and others where these approaches can be used. I think for the big indications, however, it will probably be required to make use of these tumor activation mechanisms, right? Like antigens like HER2, EpCAM, EGFR, you will not be able to target those with a T-cell engager because they are just too potent. But there is now possibilities by protease activation, pH activation, also to make the next generation of T-cell engagers in those fields. And I think there has been now first clinical data supporting that these concepts, they can work out. So I'm optimistic that in the next years we will see it. And I think as we heard before, when you then combine this with co-stimulation, of course, you have to make sure, or ideally you are using targets that have a non-overlapping normal tissue expression. I think that can then also enable you to get more tumor selectivity of the combination therapy.
Other questions? There's one more question.
Thank you very much for your nice presentation. I have two questions, one a curiosity. And the first one is, when you showed the trend of the TCE, it seemed like at the beginning they were producing or designing small molecules, like smaller antibodies, like tandem single chain Fvs. And then increasingly it seemed like they were going to more bigger antibodies. Is the reason, what do you think is the reason about stability or half-life? Half-life could be a positive or a negative thing. What are your thoughts?
Yeah, well, early on, I think there was there were some issues with the FC that resulted in toxicities. So that's why the first generation of T-cell engagers did not have an FC part. But then the problem was that they had very short half-life of, let's say, around 20 minutes, so that they had to be continuously administered to the patients, like Blincyto, and that's really why the field moved away. There's no solutions to basically get rid of the FC-mediated toxicities. And I think the surprising fact is that also if you now look at the doses at which these T-cell engagers are applied, right, they're much higher than, for example, the dose of Blincyto, which is still in the microgram per meter square range, but most of the today's T-cell engagers, they are in the 10, 20, 30 and higher milligram range, more or less almost like conventional antibodies. And I think that really shows you, I think you need these doses to really enable the antibody to go into the tumor microenvironment, right, to accumulate there and to mediate efficacy. And if you don't have an FC part, the antibody just does not have the time to even go to the tumor. That's why it's really important.
Thank you so much. And the second question I have is a bit more specific of one of the results you showed about the CD19, CD28. You showed that when you use CD19, CD28, eventually the tumor outgrew. Mm-hmm. But when you added after the CD20, the 4-1BB CD19, you didn't have this.
Yes.
Did you encounter T-cell exhaustion with the CD19, CD28, or what is your main explanation?
Yeah, I think it's, well, this is kind of resistant, or the relapse in this case is really driven on the T-cell side. And I think we still do not fully understand what's going on there, but it's kind of this, the exhaustion phenomenon, right, that you can delay when you do the co-stimulation with CD28. But ultimately, it's also known that for the more durable responses, you need the 4-1BB co-stimulation, and that's also what in natural T cells you have all of these pathways present. Or in the field of CAR T cells, you also see the differences regarding the persistence. So the CD28-based CAR T cells, they usually don't persist as long. They are more efficacious, or they produce more cytokines, but then they also disappear. Where in the 4-1BB, they have more of the durability. So I think that's very line in a way with the expectation, I would say.
Great. With this, many thanks for the presentation. And we move to the last short talk. Great pleasure to introduce the last speaker. So Mitchell Levesque is a professor here at the University of Zurich with a focus on dermato-oncology. And this is also his topic now. Mitch, we look very much forward to your talk.
Great. Thanks to the organizers for inviting me to this really stimulating symposium. So I actually made changes to my title and track changes here. So you can see that I'll discuss in the next 10 minutes predicting therapeutic responses to immunotherapy in melanoma and preventing IREs. And here are my disclosures. So we are used to getting good news from melanoma, and there have been a lot of failed trials recently, but the good news that was reported this year was that at least in the Checkmate 067 trial, the 10-year response for melanoma patients in stage 4 treated with immune checkpoint blockade is highly durable. So at 10 years, 52% of patients have melanoma-specific survival in the combination checkpoint blockade. Nivo alone, 44%, and Ipi alone, 23%. So this is on one side really great news that these responses are durable, but on the other side, it means that half the patients are not benefiting from this therapy. The second great news that came out last year in a very important landmark study from the group of Christian Blanc was that the Nadina trial reported that in stage 3 melanoma, that neoadjuvant setting versus adjuvant was highly beneficial. What this means in melanoma is that adjuvant setting in stage 3 means that you remove the tumor and then give checkpoint blockade. Neoadjuvant means that you keep the tumor in place, you treat with checkpoint blockade 2 cycles and then remove the tumor. And you see that there's substantial benefit as a result of this different therapy regimen. And so this is great news for stage 3 patients. But once again, the challenge in melanoma really is twofold, and that is predicting which patients are going to respond to which therapy. You can see that very clearly here. So which patients are going to actually respond to the single or double. This is clearly not known yet in melanoma. And also which patients are going to develop immune-related adverse events. You can see those IREs are very high, so up to 55% in the combination treatment in the first line stage 4 melanoma. And in stage 3 setting, it's also very high. And this is really important if you think about it, because a lot of these patients don't even need therapy. So a lot of these patients, up to 40%, actually will have event-free survival up to 1 year without any treatment. So the question is it worth exposing them to this potentially lethal and maybe even adverse events that last their entire lives if it's not necessary? So that's why biomarkers are still really critical to differentiate these two different things. So we started working on this already in 2018. And at that point, what we did is we just performed cytoff analysis in peripheral blood from patients receiving checkpoint blockade together with Burkhard Becker. And what we saw is that clearly patients who were future responders to therapy had very high levels of classical monocytes in their circulating blood. And this was predictive of both of progression-free survival at 6 months, but also at 12-month overall survival. So this seemed like a very good biomarker, but the criticism was always that we're only looking at peripheral blood. What's actually happening in the tumor? So the tumor is where the action is. So next we set up a study together with Marina Bakach at Roche, where we performed single-cell RNA sequencing in peripheral blood of patients at baseline and follow-up, as well as in tumors from these same patients. So these are matched tumors and PBMCs from patients receiving checkpoint blockade. And what you can see here, if you look at the tumor and the PBMCs, is that there's a clear separation between the genes expressed in tumor cells versus immune cells. Importantly, these are CD45 sorted cells. So we're just looking at the immune compartment here. We also looked at the melanoma compartment, which is a story that we're following up at the moment. But in this paper, at least we are just focused on CD45 positive cells. And so this is over 250,000 cells that we've analyzed in this patient cohort. And reassuringly, what you can see is if you compare the immune cells in the circulating blood versus in the tumors, that you see what you would expect in terms of classical monocytes being upregulated in the PBMCs versus tumors, as well as naive CD4s and CD56 DIM and K cells and other things. And the tumor, you see an enrichment for exhausted CD8s and activated NK cells, as well as DCs. So this makes a lot of sense. So the next question we asked is, of the patients then that responded to therapy, and this is either in the adjuvant or in the first line setting. So these patients that are responders to these to checkpoint blockade are in red. You can see that reassuringly that there was actually a increase in classical monocytes. So this could confirm our earlier paper in the future responders to checkpoint blockade. So the myeloid cells were highly enriched in future responders. So this is really just at baseline. And if we now look at the tumor, which we didn't do in the original paper, we can see also an enrichment for myeloid cells in the tumor of these patients. And there was a correlation between the enrichment of myeloid cells in responders, both in the blood and in the tumor. So this seems to be a strong pattern here. And the next thing that we did is we asked, well, since we have paired samples in the circulating blood and the tumor of these patients, can we understand how these myeloid cells are differentiating as they enter the tumor? And so what we did is it's called the velocity analysis. And this allows you to perform a kind of trajectory, a temporal analysis of how the cells are migrating. It's an inference, it's not a direct measurement, but an inference of how these cells are differentiating once they enter the tumor. You can see these are all the myeloid cells in the PBMCs, and these are now in the tumor itself. And what you can see is a strong difference between future responding patients and the non-responding patients in terms of the trajectory of these myeloid cells. So as the myeloid cells leave the blood and into the tumor, in a future responding patient, these then polarize into more of an M1 phenotype, and these are CXCL9 expressing macrophages. And we can actually chart the differentiation path of these myeloid cells as they enter the tumor. And then in the non-responding patients, these are differentiated more into an M2-like myeloid-derived suppressor cells or M2 macrophages. So we can see actually the trajectory of these cells as they leave the blood into the tumor, and that is strongly different between future responder, non-responders to checkpoint blockade. And if you look at individual activation markers, you can see in the two groups of responding patients here on the bottom, responder or tumor-free, you can see that a strong upregulation of activation markers in the macrophages of future responding patients and not in the non-responding patients. So with that, we identify potentially interesting targets that we're pursuing in terms of being able to polarize cells actively into a more immunogenic phenotype. And that's something that's currently ongoing. And then the second thing I want to tell you about, just to show you a highlight of another paper that we recently published in September, where we use a similar assay, so single cell sequencing, CyTOF, and also in this case spatial transcriptomics on a large cohort of melanoma patients. In this case, what we were trying to do is identify patients that would ultimately develop adverse events. And adverse events are very important because it could lead to treatment cessation, so that the patient no longer gets the drug, and also potential mortality and lifelong adverse events. So it's important to be able to identify the causal mechanisms of these and also which patients are going to respond. Other studies have done such work, but focusing on baseline turns out to not be very informative. And so what we did that was different than other studies is that we not only sampled patients at baseline, but also at the time of the adverse event. And not only did we sample the blood, but also the actual adverse events, so the immune-related colitis or the skin inflammation. And then we profiled these adverse events as well as the PBMCs. And then we have a very large validation cohort where we could show that these results were consistent. So I'm just going to show you one result from this study because it's published, and you can look up the other data that we generated. And what this shows is the spatial transcriptomics now. In the first column, these are normal skin samples, and the second is skin rash from the patients who developed inflammation as a result of checkpoint blockade. Here we have the normal colon, and here the colitis sample from the patients who developed immune-related colitis. And the pathways that came up as strikingly different between these samples in the spatial context was IL-17 signaling, interferon gamma signaling, and IL-6 signaling. You can see this dramatic upregulation in the colitis. And this is the quantitation here. So the skin rash and as well as the colitis have a strong upregulation of IL-17, interferon gamma, and IL-6. And then if we go back and look at the PBMCs, we can also see that this IL-17 is mostly coming from the CD4s, and most importantly, that it's not present at baseline. So it's not that you can take a baseline sample and predict which patients are going to respond, but it's only present at the time of the adverse event. And then the last thing I'll show you is because in Switzerland, we're allowed to do off-label treatments for a small number of patients that require such treatments. These patients, what we found is 2 patients with grade 3 immune-related adverse events as a result of the checkpoint blockade. And the medical authorities allowed us to treat them with an IL-17 targeting antibody, which is available for inflammatory skin disease like psoriasis, which we had available in the dermatology department. So we treated these 2 patients with grade 3 adverse events, and in both patients we could see a good resolution of the adverse events. and also reduction of IL-17 signaling in the blood of these patients. So this is not by any stretch of the imagination a proof of clinical validity, but rather I think it's a nice validation for hypotheses that need to be tested in an actual randomized clinical study. So with that, I'd like to summarize by just saying we could confirm the predictive value of classical monocytes in the blood of patients in not only in adjuvant, but also in first-line setting in checkpoint blockade response, as well as the macrophage T-cell interactions as playing either a pro-inflammatory or immunosuppressive role in checkpoint response. And that's something that we have to look at in terms of targeting and promoting better responses in the clinic. And then lastly, the role of IL-17, which, as we heard from the last session, is controversial. And so there's some complications to the role of IL-17 in immune response. And so there's something that needs to be looked at very carefully, but at least we could show it is certainly a mediator of adverse events in this setting. So with that, I'd like to thank my research group, the clinical team of Reinhard Dummer, especially the people who led the last Nature Cancer study, Florentia and Phil, and then the group of Roche, Marina Bakac and Ramona, who did the analysis that was shown in the med paper. So with that, I'd like to thank you for your attention.
Thank you very much, Mitch, for the excellent talk. Are there questions? Chiara?
Yes, thank you very much. I have a couple of questions. First of all, since you basically demonstrate that in responder you have polarization toward M1 macrophages, instead in non-responder you have the M2, there are studies that maybe also try to combine together with P1 and CTL4 also checkpoint inhibitor for macrophage of myeloid population. And instead, the second question is more on the IL-17 part. Are the patients that basically develop more toxicity also the ones that are responding more, or you see something that is like you can target just toxicity and this will not affect the response?
Yeah, it's a very good question. So in terms of the first question, I think it's still not shown that the myeloid-derived suppressor cells or the M2 macrophages are actually providing the suppressive role that's preventing a response, because it could be a correlation. It could be that it's already an immune suppressive microenvironment and that those cells polarize once they get there. So it's still not really clear to us what the functional role is of those cells. And that's something that has to be proven more formally. But I think that there are anyway clinical trials ongoing to try to target these cells. And I think that could be very promising, because it's not just our study, but other studies have shown that they're always correlated with poor response in some format. So I think it should be a very promising area for research, and I look forward to that. In terms of the second question, it's a very good one. It does seem to be the case that patients who have strong IREs at the beginning are the ones that have a sustained response. So you don't necessarily want to prevent that response from happening at the beginning. But if the patients then have to be taken off of the drug because of that IRE, then there's good rationale to try to at least reduce the inflammation, and then maybe potentially go back onto either a single agent or a different checkpoint. And as more therapies come online, I think it's going to be easier then to make those choices, because maybe they don't go back onto IPI, which is probably causing that response, but on some other checkpoint like anti-LAG3 or something like that. So I think that over time that this will be resolved. But it's certainly a good question, is how much does targeting the IRE itself prevent the response? And that's something that could be very dangerous in a randomized trial where you don't want to affect ultimately the patient's outcome.
Are there other questions? If not, then in the interest of time, many thanks, Mitch, for the talk. And I hand over to Markus.
Yes. So we come to the end of a very long day and symposium. I think we saw such a broad spectrum and so many various successes and advances that it's almost mind-blowing, but that's good news, and that's actually needed, because if you think of the cancer patients we need in advanced stages, although there are enormous progresses, there is a lot of room for improvement. So it's great to see all these fantastic signs ongoing. I would like to thank again the organizers, especially from the Comprehensive Cancer Center, Silke Uflacke. I think we should applaud. And again, I'd like to thank the sponsors. And most of all, I thank all the speakers that were giving really impressive talks. I thank all you for coming, for engaging in the discussions, and also the, I think, up to 97 online participants that were with us. So with that, I wish you a good evening and a good weekend.